Isn't it exciting to be in a new room with so many new things to figure out about?
Now the slides are here. Excellent. So I think we can start. Good morning, everyone, and a warm welcome to this morning's keynote lecture. It's a great pleasure to introduce you to Professor Linda Griffiths from the Massachusetts Institute of Technology. Linda Griffiths was trained in chemical engineering and is currently professor of biological engineering, MIT, where she also directs the Center for Genopathology Research. After establishing many design principles for biomaterials and scaffolds for regenerative medicine, including co-invention and commercialization of the 3D printing process in the 1980s, with early demonstration in liver and bone tissue engineering, she gradually shifted the focus of her lab toward building in vitro models of complex human diseases, especially those characterized by chronic inflammation and immune dysfunction. In the late 1990s and early 2000s, her lab developed and commercialized the liver chip, an early 3D perfused liver model. A current focus is improving the tissue engineering approaches for organs on chips, especially for complex epithelial tissues like the endometrium. She's a member of the National Academy of Engineering, the National Academy of Medicine, the American Academy of Arts and Sciences, and has received numerous other awards. Griffiths currently serves on the advisory board of the Society for Women's Health Research and has served on the advisory committee of the director of the National Institutes of Health. And the title of her talk is Deconstructing and Reconstructing the Patient. Linda, we're very much looking forward to your talk.
Thank you so much, Adrian. It's really, actually, let me have the mic. I'll just do it with the mic from here. The room is a little bit difficult with the way the lights are, so I'm going to stand over here. So it's great that you're all here so early on the last day. It's been a fantastic meeting, and kudos to the organizers for bringing us all together and to the NIH staff who helped foster this field through the Tissue Chips program. What I want to do today is share with you first a little bit of intro to some things that have gone on in lab related to the topic of this conference. That'll be somewhat of a review and survey more than new stuff on the organs on chips, but then really dive into something I think is super important for this field, which is conceptualizing the biology in patients, particularly for complex diseases, as complex diseases that involve inflammation are some of the hardest ones to develop drug. Who's staying here for the American Diabetes Association meeting that starts today? Okay, I see a couple people like me who'll be here. And I'm going to focus on a particular disease because I actually go in the operating room and collect samples from patients, and I go to many, many clinical meetings in that space and explain how we deconstruct the patient, and then of course come back to tissue engineering and organs on chips. So that's the talk today. So first, the little survey. I became involved in this field actually at its beginning. I had my roots in tissue engineering working with a transplant surgeon and Bob Langer. I was their first tissue engineering postdoc, and we focused on trying to build liver for regenerative medicine. But in the 90s, there was this aha moment that all of these things that patients were getting liver transplants for, we just didn't understand the disease very well. There was a complete dearth of models to explain human disease. So I shifted the focus of my lab toward building these kinds of in vitro models. And when Gail Naughton, one of the early tissue engineering pioneers, founder of the company Advanced Tissue Science, and I were asked to write a perspective for science on where the field of tissue engineering was going, we included in it that we really thought it would be in the future these model systems for complex diseases. So this is 20 years ago, and you always say in 20 years we're going to be, but here we are. In fact, this is a huge, huge, huge growing application in the field of tissue engineering. So at that time, we had developed a very simple 3D perfused model of liver involving a little silicon chip shown here on the left, deep reactive ion etching. You see cells into the scaffold. This is the basis of what CM Bio now has as a liver chip. There's a membrane on the bottom. The cells undergo morphogenesis. And in this initial iteration, they formed tissue-like structures. These happen to be rat cells. When we combined the different non-parenchymal cell types, you could see features of some of the sinusoidal, but in no way is this liver. The value of this arrangement, though, was the perfusion and so on allowed us to get good mixing and keep the tissue functional for weeks at a time. So it was a lot of interest, collaborations with University of Pittsburgh and others led to a lot of interest that led us to simplify the operation of the chip. Albert Waugh, who's here from NIH, struggled with this very first iteration on the left and published some very nice work with sinusoidal endothelial cells, but we translated it into a multi-well plate format. And critical to that was the development, together with my fabulous collaborator, Dave Trumper in mechanical engineering, of onboard microfluidic pumps that are pneumatically driven. So these are just little pneumatic pumps that circulate fluid around and around, driven by air and vacuum. So there's a membrane between the top and bottom, and the fluidic channels are machined in this case. And so this has now been commercialized, and you probably have seen several of the presentations by CN Bio in their booth and so on, and they continue to be a wonderful collaborator and supporter of our work, and we actually order liver chips now from them. So since the beginning of my work in this field, working with pharma, going back 20 years, working with Cindy Afshari when she was at Amgen, all the problems that were brought to us in liver involved some aspect of immunology, idiosyncratic toxicity, et cetera, et cetera. And this is just an example of a project we did with Amgen to try to dissect what happened, dissect a phenomenon observed when tocilizumab went into the clinic. And it was observed that patients taking small molecule drugs like statins, the metabolism changed so much that the FDA put a warning label on it about that. And so together with Amgen, we showed that you could recapitulate this phenotype seen in patients by creating an inflammatory situation in liver where if you had immune cells present, and in this case, Kupffer cells, and then relieved the inflammation with tocilizumab. So this could be recapitulated. And again, this isn't all the functions of liver, but it's an illustration of the value of these MPS systems. And so now we continue to apply these currently in a really lovely collaboration with Novo Nordisk to liver metabolic diseases and then to some other applications. So for example, here at the ADA meeting, Erin Tabonian, who presented a poster last night on work she's doing to continue work done by a postdoc, Jessica Ungerleider, is investigating in vitro metabolic disease models here showing that you can induce insulin resistance by hyperinsulinemia. As many of us have mentioned during this meeting, the use of insulin in cell culture is insanely hyperphysiologic for most cell culture. So that's an example. We, of course, were one of the groups that was involved in the big DARPA program, our group and Don Ingber's group. And we use this as an opportunity to expand the pumping technology to interconnect 10 organ systems together, living at one time in constant communication on a platform. So unlike the Emulate system where they pipette between, this one actually had interconnected living for a month. And Murat Chirit, who's here and now heads Javelin Biosystems, headed our translational systems pharmacology team, and he did all of the modeling for the pharmacokinetics such as shown here. So this was a demonstration project. DARPA let us continue demonstrating more biology. This just showed that you could do this, but then we applied the platform to some very interesting physiological situations involving gut and liver. And Martin Trapachar, who's very involved with this meeting, when he was a postdoc in the lab, led two really fun projects looking at gut-liver interactions and gut-liver-brain, specifically around how short-chain fatty acids may be influencing physiology in inflammatory situations. And I'm not going to show all the data, but the punchline in this story was that they're a double-edged sword in health. They can be very beneficial, but when there's inflammation, you can get a potentiation of certain deleterious effects. And you can read his work, and he's here at the meeting, and you can find him at the break if you'd like more on that. What's stimulated us since the DARPA project, though, is the need for much more sophisticated tissue engineering systems to put into these platforms. So we first showed, we extended our platform technology, again, not with great tissue engineering, but that's what I'll talk about mainly in the talk. We extended it to show that you could culture one of the most fastidious oxygen-sensitive microbes, Faecalibacterium prausnitzii. Sorry, I'm getting used to this. which is a super, super, super strict anaerobe. This organism is responsible for production of a lot of the short-chain fatty acids, particularly butyrate, in your lower GI tract, and it dies in the presence of oxygen. So this is a continuous culture, human colon monolayer here on a transwell, and the bacteria growing here. And this has also been published. So this demonstration showed us that we have the technologies to do this, but we have a monolayer here, so we need a much more sophisticated model. So what I'd like to do now is turn to ways that we think about deconstructing these complex diseases so that we can build MPSs that will help us discern the path forward for drug development. Who doesn't know about endometriosis? Oh my God, I'm so happy everybody knows, because 10 years ago, that wasn't the case. So it's a chronic inflammatory disease characterized by ectopic growth of endometrium. We don't know what causes it. It could be developmental. A lot of young girls have problems starting right when they start their periods, but others develop it later in life. There's many different forms the lesions take. Here's some in the abdominal wall, highly vascularized. This is invasion into the colon. The thing about them, they're beautiful cystic-like structures. They retain a monolayer epithelium, completely polarized. So it's not like a tumor where you get all jumbled cells that lack polarization. And of course, it can even invade through the diaphragm and get to the lung. And this is very serious. Patients really, really, really suffer. Super prevalent. And if there were good treatments for it for everybody, I wouldn't be talking to you here today, right? So about 30% of patients have invasive disease. This is a patient of my collaborator, Maurizio Brau in Sao Paulo. He had to take out 10 centimeters of this patient's colon because she couldn't poop. Okay, so this has many manifestations and has remained clinically very mysterious. The landscape is very murky because the symptoms don't correlate well with the staging of the disease. So a patient with advanced disease might have very few symptoms, and a patient with one lesion may be crippled in pain and unable to work during her menstrual cycle. Who has heard of adenomyosis, or who has not heard of adenomyosis? Okay, so adenomyosis is the sibling, if not the twin, of endometriosis. It's when you have these lesions growing in the muscle of the uterus, the myometrium. And so you can see, again, beautiful epithelial structures sitting right there in the muscle. And about 1 in 10 women are estimated to have adenomyosis. To give you a sense of research intensity in the field, let's compare we like to use PubMed as a surrogate for measuring research intensity in all research sectors, industry, and around the world. And if you look at Crohn's disease, which afflicts about 1% of the US population, there's over 60,000 papers published. Adenomyosis, which probably afflicts 10% of reproductive age women, if you normalize, it's probably afflicting at least twice as many US citizens as Crohn's, and yet there's only 3,000 papers, 5% of the research intensity. NIH only has 2 grants ever on adenomyosis, and one of them just started. So this is a really huge problem because these diseases are under the radar despite afflicting a huge percent of our population with crippling symptoms. People miss work because of this, seriously, and they can't get pregnant and all these other kind of things. So when we came into the field in 2009, I got pulled in by a gynecology surgeon, and for some personal reasons as well, including the need to be thinking about how we could apply engineering to women's diseases more prominently. What was striking to us is that the disease was treated as binary. You're a patient or you're not a patient, and you have stages that are staged according to disease burden. Stage 1 is a few lesions, stage 4 is a lot of lesions and endometriomas. But there was enormous heterogeneity in the clinical landscape in terms of symptom presentation, types of lesions, et cetera. And so we started to question whether the staging, which is just disease burden, really made sense. And so we promoted the idea that it's not one disease, that perhaps there are molecular subtypes that one could identify. And so we started out about almost 15 years ago now to deconstruct the patients. So to move beyond this idea that the lesion burden is a classification scheme. And a miracle happened right after we got a foundation. We started with a foundation grant of about $1 million. And right after that, I got breast cancer. And it was incredibly, incredibly illuminating, because within days of finding a tumor myself in Singapore, do self-exam, women, I was classified as having something called triple negative breast cancer. So the 3 molecular markers that were related to the mechanism, prognosis, and therapy for the disease immediately, based on clinical studies with thousands of patients. There was nothing like this in endometriosis. And even though there are not somatic mutations that are credible yet, we speculated that we might be able to find different subclasses of patients on a molecular basis. So how do we do that? And why do we do that? So to kind of orient you, in an era of personalized medicine where you might think every patient is different, and indeed every patient is different, there are probably subgroups of patients, maybe 5, 10, however many, such that you're not disease or not, you're not binary, but you can't possibly, with almost 200 million women around the world suffering from these diseases, you can't possibly have a different therapy for every patient. So we like to think of subgroups of patients that may be in the order of a dozen or so. And this is going to be a many decades long quest, of course, to figure this out. And so we embarked on what we call the systems biology of endometriosis, where we study the disease in the eutopic tissue, the endometrium, where you have epithelia communicating with stroma and immune cells, in the peritoneal cavity, where you have fluid that has about 1 million cells per mil, mostly immune cells, and obviously all their signaling molecules, and then the lesions themselves, which I'm showing here, are on the abdominal wall. And you can glean facets of disease processes from studying all of these tissues in a comprehensive way. And we've published a lot on this. I'm going to give you an overview of a couple of studies to give you a sense of how we approach this. So when we started, we had said, well, if there's not somatic mutations that you have in cancer, maybe we can glean insights by looking at cell-cell communication networks involved in the immune response and in the invasion response. And biologists had described a lot of inflammation markers in the peritoneal cavity where the action is, but they typically would focus on a single pathway or a favorite molecule like RANTES. But as engineers, we come in and think, how is the whole network operating, and how are networks communicating with other networks? So this idea that we can think about information flow in an ecumenical sense and measure activity across the entire network to discern its function and its dysfunction, that was new. So we developed a couple of studies probing the networks, and one was the cell-cell communication networks involved in inflammation in the peritoneal cavity. And the peritoneal fluid is super easy to harvest. We've taught surgeons all over the world how to do this because you can get it right at the beginning of surgery, and it's discarded tissue, so very easy IRB. You get the fluid and then you can separate out and save the cells to examine later, and then study the signaling molecules. And so we developed a way, and this was back, 10 years ago, it was hard to do quantitative Luminex. It's still hard to do quantitative Luminex. So we spent a lot of time developing a way to do Luminex to get quantitative information on about 50 of these cell-cell communication molecules between the immune cells. And then we did an unsupervised multivariate analysis mathematical method to ask the question, are there groups of patients in whom changes in the immune network are correlated? So in other words, if you might have patients in whom some things are up and some things are down, and these patients are changing together. So we used a kind of math. It's the same thing that Netflix has traditionally used to tell you what movies to watch. And it's unsupervised, meaning we didn't give it any clinical information. So we didn't say who's a patient and who's not. That doesn't mean it's hypothesis free, because we had a hypothesis about the communication networks. But we could then ask, are there groups of patients who are together who may have a common mechanism? Now, we did this because typical univariate analysis is not that informative. And this is a typical kind of result showing IL-8, which is always in most studies reported up in endometriosis patients in the peritoneal fluid. This is a log scale, and you can see the problem. Here's the controls, here's patients, and this is plotted 2 ways. These are patients who are taking a drug. And you can see the spread here. A huge fraction of our patients are overlapping with the controls, no matter how we plot it. Here's by disease stage, here's by menstrual cycle phase. So there's always this huge overlap, suggesting that there's some patients who are different. Another way to look at this, and this is a different study study, I'm not going to go into it, it's one we did recently. This happens to be endometrial biopsies. But again, you see the same overlap. This is IL-15 and endometrial biopsies. And this is all published. This has actually been published, sorry for the slide. But the one that came closest to significance shown here, so these are all the cytokines, IL-15, didn't turn out to be significant after we corrected for multiple hypotheses. But you see the problem here. Almost all of the data are overlapping with the controls. So now, though, if you go in and you add up all of the different cytokines, almost all of them have these features, but you can imagine that patients who are up here may have some other cytokines down here differing from controls, and now you get the power of multivariate analysis. So when we did that, we had an initial study with about 100 women, and we did this analysis, and what we found in about a third of the patients who had all the disease stages was a signature set of cytokines shown here that they all express, and none of the controls had this signature set. We've replicated this for the top 5 cytokines with Maurizio, and that paper hasn't been published yet because the postdoc left precipitously, and I've got to write the paper. But we could take these data and reverse engineer this immune network, because immune secretion profiles are beautifully documented in many databases. And we identified a particular state of macrophage in the peritoneal cavity of these women and then experimentally validated it. And we also predicted that Jun kinase was regulating production of these. And this was the first time that Jun kinase had been implicated. And in fact, we showed that Jun kinase was by inhibiting Jun kinase. And then we did a lot of other kinase inhibitors. And so this was very exciting, especially because we then also implicated Jun kinase in a companion study looking at invasion with a similar kind of network analysis. And this involves in vitro immune invasion assays. And of course, this has also been published, so you can go back and look at this with a lot of math in that. And this was in collaboration with Doug Laufenberger, our systems biology collaborator. Now, what was fascinating, when we published these papers, almost immediately, we learned that Jun kinase inhibitors had cured multiple patient populations of endometriosis. Woohoo! Okay, however, they were not human. And in fact, Steve Palmer, the lead author here, was at Merck, and he had gotten Merck to sponsor 2 preclinical studies that then Merck decided not to move forward into the clinic. They licensed it to Preglem, their molecule, because Jun kinase inhibitors were being developed for other applications. The clinical trial was unsuccessful for a lot of reasons, probably not having to do ultimately with whether inhibiting Jun kinase would actually help patients. Issues with the drug, issues with the specific composition of the drug, issues with the trial design, et cetera. And so in talking to a lot of people, pharma, we got to have better efficacy models. So that brings us to how we reconstruct the patients and the topic of this conference. Okay, so now we want to go in and we want to build avatars of the patients. We want to capture key disease phenotypes in vitro so that a company can feel confident that if we propose a mechanism because we went in and studied all these patients, or they propose a mechanism because they went in and studied patients, that we can test that mechanism in vitro. And we can also obviously use many of the approaches here at this meeting to look for toxicity and other effects, because our lab has shifted mainly to analysis of disease processes, and we're not really hugely involved in toxicity studies anymore. And so to go in and think about this, obviously the lesions are there. Patients with no lesions do not have these symptoms. So how can we think about what might we do for the lesions? And it's a dream someday that we would diagnose this at birth and prevent lesions from ever happening. But we started thinking that surgeons will take the lesions out, but there are micro lesions still left in these patients. And are there ways that we can affect their growth into these more macroscopic lesions that are causing the patient so much distress? So part of the lab, I have a really dedicated postdoc who's going to be joining the faculty at Tufts in August in bioengineering, Juan Necco, and in collaboration with a local company, did some whole mount tissue imaging using the clearing process and light chain imaging to characterize some of the features of the lesion. So this is an adenomyosis lesion in situ, and this is looking down inside the lesion. Recall that these are beautiful cystic-like structures with a monolayer epithelium. And so you can see gland-like structures pointing out here, here, here. These are incredibly innervated. They're incredibly vascularized. And what we are doing is saying, okay, if we're going to recreate these lesions, we're going to think about the parameter space that's biophysical and biochemical that direct the properties of these lesions, because some lesions are very close to peritoneal fluid, which has an insanely weird composition of hormones and et cetera, but they can then invade into muscle. How crazy is it? You never even see carcinoma in muscle, but these are in muscle. So a lot of interesting features about the environment. So we create a parameter space and we want to march through parameter space and understand how these cues are dictating function. So of course, we have to first build a tissue bank from biopsies from well-phenotyped patients and then rebuild the patient. So how are we doing this? We had to develop an enormous number of new tools. And I think these tools will be generally useful to the community, and we're trying to commercialize them right now. A huge innovation was not ours, it was Hans Cleaver's. You can make organoids, and everybody here knows about organoids. This is an intestinal organoid, but it changed our world when this technology was translated into the endometrial space by two different groups. And we work with the folks in this group here, Hillary Critchley and others, because now people all over the world use these approaches to make endometrial tissue banks. Before this, we would get endometrial biopsy samples and the epithelia had to be a one shot. You use it right away. But now we have a huge tissue bank with a lot of patients. So the general process when we started, of course, was to take the glands, isolate them, and build the organoids in matrigel droplet culture, as is standard, using the cocktails of growth factors. But as we know, matrigel is a huge problem for so many reasons. And we needed to get away from it. We also had a super practical reason for having to get away from matrigel. In the DARPA project that we had, tissues had to live for a month. The endometrium was one of our tissues, and there was no way you could keep a culture going for a whole month in matrigel because it disintegrates. So we had a very practical reason, which is that 28 days is a long time. So building on pioneering work by Jeff Hubble many decades ago that the universe has adopted, we did a very, very customized approach to using multi-arm PEG modified with cell interacting peptides, cross-linked with peptides. You combine these two precursors with cells, and we typically do a Michael addition, which is a small pH change, and you can create these beautiful gels that encapsulate cells very, very safely. And we published almost everything on this. You can find it online. So I'm going to do a whirlwind tour of it, but hopefully repeating some things you may have seen at some of our posters, but giving you enough information that you understand what we did. So a crucial thing for us, of course, is we want to build the entire lesion. And we actually also want to build the endometrium, because the endometrium is so fascinating, not just for recurrent pregnancy loss, but for regenerative medicine purposes, because it grows so incredibly beautifully without scarring. So what did we do that was special? The thing that our application drove was that we had to have epithelial cells be very happy, and they're very finicky. They want a basement membrane, and then stromal cells and immune cells at the very least, obviously endothelial cells and so on. So how can we build a matrix that satisfies all of these cells and is so-called one size fits all? What we did was, of course, cross-link things with a degradable cross-link so cells can remodel, and that is a very common approach. But we thought a lot about the particular adhesion ligands that went into this gel, and we envisioned it as you initially put cells, they're naked because they just got taken out of a tissue or they just got taken out of a cell culture expansion format. So they're naked, they have no ECM, they need some signals immediately when you put them in or on this gel. So we needed some minimal peptide adhesion ligands in order to engage integrins immediately. But then cells make their own matrix. So we have two ligands that we include, and this was based on a lot of engineering, including by Victor Hernandez-Gordilla, who's here. He's now at United Therapeutics, and I'll show you some of his work in a couple of slides. But we had an unusual RGD ligand. Now, a huge fraction of the biomaterials community makes materials with simple RGD ligands, or maybe a loopy RGD ligand, cyclic or something. But that's partly because almost all cultured cells used in the biomaterials community express alpha V beta 3. Either they're tumor cells or they've been cultured a long time and they evolved to interact with vitronectin, fibronectin, and serum. But most primary cells do not have alpha V beta 3. It's very rare in the body. It's in tumors, wound healing, and actually in the endometrium for a tiny window during implantation, but it's not a real physiological ligand. It's a kind of wonky integrin. So we include the synergy site from the ninth domain of fibronectin. So here's fibronectin, so that we get a broader RGD integrin binding set of integrins. And this works very well with primary cells that are not expressing alpha V beta 3. And then a motif from collagen that can interact with alpha-1 beta-1 and alpha-2 beta-1. But that's not all. So that's to get the cells going. But importantly, we then also add these peptides that bind to matrix that cells produce. And that's because PEG itself is anti-adhesive. It's a blank slate. If cells are making matrix, it's not going to stick to PEG very well. So we make it stick to PEG. And that seems to work very well. And we published a huge number of papers on this by now. And I encourage you to look them up if you're interested in this. But let me tell you what we're doing with it. So when we started this, of course, we became interested not just in the endometrium, which is a really fascinating mucosal barrier, but the intestine. And there were many reasons for this. One, of course, was our DARPA funding required that we build all these organs. But going forward, there's also other reasons to continue working in the intestine, and they have to do with the funding out there. You can almost open up your chute in your ceiling and get funding for intestine, but not so much the endometrium. So this is for the young people in the audience. But there's also fascinating intellectual reasons to compare these two together, in that in the gut, as we know, the stem cells reside in a crypt in a healthy normal homeostatic gut. In the endometrium, in contrast, you have the basalis, which is always there. The functionalis is shed every month in cycling women. But you have evidence from several groups, including a recent publication from the Cambridge group, that there are stem cells actually out here in the lumen. Okay, so they're getting rid of, maybe they're what's causing endometriosis, right? So there's super fascinating questions about structure and function in these two mucosal barriers that we're pursuing, because there is evidence of stem cells here. And so we continue to digest those, and I don't have time to go into all of that fascinating biology in this talk, because I want to tell you a little bit more about the matrix, which was developed by Victor. And he took the scanning, semi-empirical scan of parameter space, thinking about the nature of our chassis, meaning the PEG, thinking about the integrins that are in the crypt and how do we target them when they're naked so that they'll live and prosper, and then deposit matrix. And in doing this, he identified a composition of hydrogel, completely, totally synthetic. So you're not going to go in the supplemental information for this paper and find that we doped it with laminin. There is no doping of laminin in this, as there are in some other papers, and it's very confusing sometimes to figure that out. So this is completely synthetic. So here's cells growing in matrigel, and this was done multiple donors from the upper GI as well as the colon, that you can get clonal growth from single cells in this, not with the efficiency that you get in matrigel with this initial formulation, but we've done a lot of work since then. So a crucial thing was his figuring out that you needed active Gefoger. If you substitute this crucial residue in the motif with analog, you don't get growth of the organoids. And that's all been published. We're now exploiting some fascinating properties we observed with this to try to generate true mucosal barriers and barrier function by having fusion of the organoids with the surface. And this was a poster that Elise Gubbins in the lab presented last night. And again, this is one size fits all. So you can include stroma and immune cells, and they can the immune cells can traffic through this and so on. So let me finish up by explaining what we've done with the endometrium here, because I have plenty of time to finish what we do. Okay, so we want to come back to the endometrium, which again is really intellectually fascinating, because more than other tissues, it is dramatically, well, except for the ovary, of course, and other reproductive tissues, it's dramatically influenced by the cycles and hormones. So typically you start your period and the functionalis is shed, and then you're going to grow the endometrium back. This is driven initially by estrogen and what's called the proliferative phase. So there's this massive centimeter big growth of the endometrium. And then ovulation occurs and progesterone is produced by the corpus luteum. And you get a maturation of that endometrium, and the stromal cells, in the fibroblast in the endometrium, bifurcate into different populations. Some remain responsive to progesterone and they become epithelial-like. They make a basement membrane, they produce prolactin, they make gap junctions and cadherins, and they look very epithelial-like. And then another population of fibroblasts becomes acutely senescent and makes matrix and growth factors and so on. So some fascinating biology, and you retain along the way stem cells in that fibroblast population, as shown very beautifully by Carolyn Gargette's group in Australia. Okay, so you have the secretory phase, and if you do not have implantation of an embryo, progesterone falls precipitously, triggering a cascade of events that causes tissue breakdown. So you've got this whole approximately 28-day cycle, and it's a challenge in tissue engineering because it's a kind of long time to have the tissue be resonant. So So let me now take you through some things we're doing in tissue engineering and introduce you to one concept we use to characterize the phenotype. And so even in 2D culture on plastic, as shown here, these are stromal fibroblasts from the endometrium. They have a very unusual phenotype. They express huge levels of CD10, a cell surface endopeptidase, that's also expressed in the immune system. If you give them a progestin, they undergo this process called decidualization, in which they turn into those epithelial-like cells, and they make buckets and buckets of prolactin, a molecule involved in many physiological processes, and it's broken down to create submolecules that are very involved in regulating angiogenesis and endothelial function. Many uterine disorders, including endometriosis and adenomyosis, are characterized by something we call progesterone resistance, which is one of those syndrome things. It means that if you give these stromal fibroblasts progesterone, they're not going to do all the things they normally do, including they're not going to make prolactin. Okay, and so we can use that as a characteristic in vitro. And everything that I'm doing, this is all patient-derived cells. We no longer work with cell lines. Okay, so as one example, and this was all led by Juan Necco, who's transitioning to his own lab this summer. And let's focus first on those fibroblasts. And in 2D, I just showed you what they do with giving prolactin. So you have them in estrogen, you give them a progestin, and we're using methoxyprogesterone acetate because progesterone is metabolized very rapidly. We're transitioning to progesterone, and I can talk to people about that later if they want. But you can see that the bump up in prolactin production after a couple of weeks, which is a characteristic experimental protocol, is modest. But if we put them inside this 3D gel, the response to prolactin is progesterone is dramatically enhanced. And this is true across multiple passages, et cetera, et cetera. And I'm not going to show you the data, but if we culture these cells on this gel, and this gel is about 250 Pascal, 250, 300 Pascal, so similar to the endometrium. If we put cells on top of this gel, guess what they do? It's crazy. They act just like this. So even a soft gel does not generate this. So this is an area that we're still investigating. There is a paper in preparation. We're not fully understanding why the soft gel 2D culture doesn't help that much. Okay, but let's move to building co-cultures of the endometrium, because we really have enormous crosstalk driven by these hormone cycles, crosstalk between multiple cell types. Let's just first look at the epithelia and stroma. In fact, I'm only going to describe epithelia and stroma, even though we're doing a lot with the other cell types. So first you can combine the stroma and epithelia, and here we're just using organoid culture. So you could see the beautiful apical actin, which is how the polarization should be. And if we were to stain for basement membrane, you would see it there. But these are the stromal cells interacting with the epithelia. And if you go through the cycle, you can recapitulate features morphologically that you would see in vivo. Now, of course, we want to do disease modeling. So a simple first characterization is add an inflammation cue to the culture media, IL-1 beta. And this is on bioRxiv, and it's going to be submitted any day now in a highly revised form from what's there, but the data are still pretty similar. So if you add an inflammation cue, you immediately see that these cells flatten out. They get about half as tall. The stroma become activated, and they do a lot of crazy things. And this is true across the whole menstrual phase. And there's a lot of effects on proliferation and so on that are described in the online version. And of course, adding IL-1 beta will, if you add IL-1 beta to control co-cultures, they make in this gel, they make a lot of prolactin, but the controls make a lot of prolactin, but IL-1 beta blunts their ability to respond to progesterone. So this is the progesterone resistance that you see in patients. But importantly, the use of a synthetic hydrogel lets you to recapitulate features of the biophysical environment of the invasive lesions. So here we have a lesion. This is in the myometrium, and it is so crazy. These things are invading in the muscle. This is just wild. So this is the epithelia. This is by light sheet imaging that Juan did. And you can see these jagged edges invading into the muscle, and it is pervasive throughout the endometrium. Okay, and now if we create our gel to have the stiffness that I just showed you, but also to make a stiffer gel like that of the myometrium in an adenomyosis patient. And now we do these co-cultures. You can see these are beautiful, normal looking organoids, but in the stiff matrix, they take on features of the invasive phenotype. And there's some other behaviors that they engage in that are indicative of inflammatory just induced by the biophysical microenvironment. So now we have a number of tools to investigate this in a systematic way. One thing we're also doing is trying to compare to in vivo, and this is a little side story from a collaboration with Hilary Critchley's lab in Edinburgh. And Hilary Critchley is a clinician scientist. She's one of the leading lights in the world to investigate a phenomenon called heavy menstrual bleeding, in which in Europe and the UK, patients are giving a selective progesterone receptor modulator, an SPRM, and the common one is ulipristal acetate, okay? And so you can have progesterone as an agonist of the receptor, and mifepristone, also known as the abortion drug, is a complete antagonist, and UPA sits somewhere along here, so that it interacts with the PR, but in a weird way, such that in the intact endometrium, it downregulates proliferation and but causes enormous morphological changes in the endometrium. And in the US, there was concern that these changes might be associated with later development of endometrial cancer, which is on the rise in the US. Of course, endometrial cancer is hugely associated with obesity. So women in the US are not able to take this actually super effective, wonderful drug because of lingering questions about what it may be doing permanently to the physiology of the endometrium. And so what Alex Salova, one of Hilary's students, did in collaboration with our lab, she decided to, Hilary had a patient cohort who were taking the drug, and she could take endometrial biopsies from the patients before they took the drug, while they're taking the drug, and then at a defined time after they'd taken the drug. So this is a really valuable clinical cohort who had taken the drug and for whom we can not only study the molecular features of their endometria via the various omics, but we can also build the tissue banks and do the in vitro models and ask, are the in vitro models capturing the signatures that we see in vivo of these patients? And the huge question for us was, can you recapitulate, or can we understand more something about the proliferation of these cells and what is going on, because there's features of both proliferation and differentiation. And so could we illuminate things in a way that might get this drug more favorable light in the US? And so what Alex did in her thesis, it may sound simple, but it's really quite tricky to do these cultures, is ask, does the matrix composition affect the cellular response to this treatment in vitro? Okay, so she's going to look individual cells, stromal cells, organoids cultured separately and combined together and compare those. And she's going to do this in Matrigel and in our completely, totally 100% synthetic PEG gel. Okay, so here's a picture of them. Now, she won a big poster award at the Society for Reproductive Investigation in March, top 5 posters, and she got one of them, which was great because she had been a very not confident student going into the study, and she emerged very confident. This paper is, she just defended her thesis, and we're getting ready to submit this paper. So the punchline is here, and I'll walk you through this busy slide to tell you what the important thing is. Okay, so there was a lot of omics data from the patient samples in vivo from which we could glean that the proliferative pathways, the cyclins and all of these things were downregulated. So all the pathways related to proliferation were pretty much downregulated from the in vivo omics in the patient samples. So we wanted to ask we didn't have, enormous number of samples in vitro because this was all done during COVID. So we used a reverse phased protein array of 100 different antibodies, picking the particular genes from the in vivo omics data. And so these were mostly cell proliferation genes, shown here in red, and decidualization here in yellow, and other proliferations in green. Okay, so the punchline is here. Okay, so these are the cells treated with estrogen, so they should be proliferative here. Okay, so based on in vivo, these are proliferative. And we see that in our PEG-grown gels, the genes associated with proliferation are pretty much upregulated, they're red. But everything associated with proliferation is downregulated in the PEG gels. Okay, so these are co-cultures. Matrigel is a mess. Okay, it's a mess. You're not recapitulating what happens in vivo. And these are co-cultures. So an interpretation that we're continuing to develop is that in the PEG gel, there's no it's a low noise environment. Matrigel has all kind of signals in it. So if the epithelia and stroma are trying to talk to each other, it's really hard because there's a lot of stuff going on in the way. It's like trying to have a conversation if you're on the runway at Logan and jets are taking off. You got to really scream. Okay, so only a few things are going to get through. Okay, but here there's nothing in between. It's a quiet cafe, you know, cup of coffee, bottle of wine, whatever, and the cells can talk to each other perfectly. Okay, so that's where we are. And so she then went back and looked at some of these things in vivo, and you always have to go back and look at the protein level in vivo by IHC with different antibodies, et cetera, and showed that the things that we were seeing in vitro were recapitulated in vivo. So that's hopefully to be submitted soon. Some of the clinical part of that has already been published. Now, I'm not going to go into this story, but I just want you to know of the publication. We had another collaboration with Klaus Jorgensen's lab in the UK on pancreatic tumor where we grew stroma and immune. These are just some data from it. It's the same gel. And an important feature of this is his lab did this all on their own with our instructions, and they made the gel themselves. So it's a very easy to use gel. And Klaus had worked with another lab, Matthias Lutoff's lab, and could not get this the gel in that case to work, but found this one very easy to use. So now let me finish up with how we're modeling the birth of lesions, because what we really want to do is look at the lesions interacting with microvasculature. So taking a cue from the work of work like Chris Hughes has presented so beautifully over all the years at the NIH meetings, making microvasculature and microdevices, and then adding other tissue types to look at their interactions with the microvasculature. And of course, our big question is, we're giving patients a lot of drugs that manipulate hormones with the idea that somehow we are suppressing production of estradiol, that if you give a patient a menopause drug, you know, and this is a huge market for menopause drugs, that they're just going to shut off estrogen production. It turns out that's not the case. A lot of patients actually have progression. I actually had progression on a GnRH analog, and it was terrible because I had terrible menopause symptoms, but I grew a lot of lesions that were very painful. So we want to understand what's really going on in a lesion, because steroid hormone metabolism is really, really wonky. And so we are working with Roger Cam, who's local, who has technology similar to Chris Hughes. And you've probably seen a lot of Chris's work here at the meeting. It's incredibly beautiful work and very inspiring. And you know, this involves putting the endothelial cells and fibroblasts into a gel, typically a fibrin gel in the center of a device like this. They form networks, and then you can flow cells through these and look at recruitment and so on. Now, Roger's lab has used this for a lot of applications that are published, many, many scores of applications. For example, looking at how tumor cells extravasate into extracellular space. So the tumor cells here in green, neutrophils are helping them move out. Once they move out into that space, you can watch them grow, because these are devices you can do imaging in. And he has commercialized these devices. The AIM chips are available, and a lot of people use them. Their website is pretty helpful. And this is our student, Ellen Kahn, who gave a talk on Wednesday morning on how she's adapting some of these approaches to the endometrial models under our tissue chips U01. And this was her first attempt to recapitulate things that have been done in Roger's lab, building the vessels in the AIM chips and flowing monocytes through them. Okay, now for the lesion model, what she's done and what she presented at this meeting is there is a CAM lab iteration where devices that are now in a prototype stage in PDMS, you form the vascular bed and you leave these little holes and you can put tumors in. So we've been trying to build the endometriosis lesions in, and what Ellen presented was the vascularization into a proto-lesion from a preformed vascular bed. This is fibrin, and this is all PEG, because we have to use PEG to grow our lesions. So we're hot on making a synthetic matrix that can be vascularized, but for now, this is the mode we use. And there are ways you can use PDMS for these kinds of experiments with hormones, but you have to be very careful, because the hormones, you must saturate the devices with the hormones first, and you can't cycle anything in PDMS. So that, of course, has prompted us, and we're very grateful to NIH for 2 grants that support our work translating all of this into thermoplastic chips that have embedded pumps of the sort that drove our liver chip and drove the DARPA project. And this is not a story for today, but this is a photo of one of the prototypes. It's not the exact one we use, but we're using this for many applications in liver, gut, everything, and I'm going to be done in 1 minute. And so I'm finishing up. We actually are starting a consortium at MIT to translate these things with Roger and Ron Weiss. So if you're interested, if you're a pharma company and you're interested in learning more about that, you can ask me. But this is my last slide. We can finally test our Jun kinase inhibitors. We're teaming up with Steve Palmer, who went into academia. He's developed new variants of inhibitors. He's a really great medicinal chemist, and we're going to test these together in these new devices. And so that's it. Thanks very much. I'm around at the break if you want questions.
Exactly. It's a keynote lecture, so no questions, but please digest with her. This was wonderful. Give her another round of applause, please. I thought this was the great start into the last day of our conference. Thank you, Linda.
Thanks for having me. Yeah, great meeting. So let's get out there and cure endometriosis.
So, okay. So let's get started with the next set of presentations.
start to sit down. We need to start the next session. Thank you. Okay, I think we're gonna keep the intro short as Linda already had given us the wonderful intro to our next session, which is precision medicine and clinical trials planning on a chip. Clinical trials on the chip planning, sorry. We learn how to use microphysiological systems to deconstruct and reconstruct the patient. Now we are actually entering into the era of deconstructing and reconstructing the population, right? So really using the microphysiological system to better understand the enormous variation, disease phenotypes, and patients' drug responses. My name is Magdalena Cassandra, and together with Peter, we're going to be moderating this session.
Exactly. So welcome also from my side. My name is Peter Loske from the University of Tübingen. And without further ado, I have the honor of introducing our first speaker for this morning. It's Uwe Marx from Tissues, and he's going to be talking about organismoids.
Thank you very much. Good morning, ladies and gentlemen, and thank you, the organizers, for inviting me to that presentation and being able to introduce to you the organismoid concept we have. Anything I have to do? The green one.
We need help. It doesn't go forward.
It
Could you
doesn't switch to the next.
Okay.
Okay, here we are. Perfect. Thank you. An unprecedented level of knowledge about human biology, which accumulated at the beginning of the millennium, has enabled the dynamic development of microfluidic systems, as you all probably know. Now, Linda Griffiths' labs and Don Ingber's labs pioneered the single organ on a chip development, whilst Michael Schuler's lab and our labs at the Technische Universität Berlin pioneered the multi-organ chip developments as illustrated here. We at the TU Berlin set the concept for the creation of an in vitro equivalent of an individual's body on chips from 2007 onward. That is like this paper here. To develop the concept, I teamed up with a number of professors of different disciplines, and the first public exposure of our concept was an invited presentation at the Royal Society in London in 2009. Here is a key slide I used at that time to explain that the preclinical test dilemma can be solved ultimately if science can deliver a human mini organism on a microfluidic chip. Indeed, a 73-kilogram human body consists of a bit more than 10 to the power of 13 cells, representing 224 cell types, assembled into approximately 60 organs, and they all belong to the 10 biological systems which are highlighted here. If you supply 2,500 kilocalorie energy per day to such a body, you all know you can remain that body live and healthy over an entire lifespan. Now, the hypothesis is that the physiological integration of at least 10 key organs on the microfluidic chip enables a minimal self-contained organismal homeostasis. That's why a human body on a chip is now defined as a miniature, mindless, emotion-free, self-contained physiological in vitro equivalent of an individual, mature human body. To underline that, we coined the term organismoid for such an envisioned solution. Much like the term in vitro organoid now stands for a tiny self-organized in vitro replication of a single organ. We expect the first proof of concept to happen in the next decade. Now, three fundamental working principles underlay this vision. Firstly, all human organs are built up by multiple, identical, functionally self-reliant structural organoids. Secondly, such physiological organoids are evolutionarily conserved and subject to genetically encoded self-assembly. And thirdly, vascular perfusion, a connective tissue bed, and organ-specific stem cell niches are essential features of any of these miniature functional units in our body. Your today's breakfast has been absorbed by approximately 7 million intestinal villi, is currently metabolized by 1 million liver lobuli, and you will excrete waste products of your breakfast through 2 million kidney nephrons somewhere in the afternoon, hopefully. How small can we downscale a human body? We at the TU Berlin decided on a downscale factor of 100,000 and published that rationale in 2012. In this case, your body still consists of 10 liver lobuli on a chip, which allows 30% of them to be damaged and regenerated without the liver model losing its function. This number of liver organoids requires a corresponding number of functional units of the other at least 9 organs to emulate a minimal, functionally independent human body on a chip, as illustrated here. In 2014, our engineers translated that into a 10-organ chip design. And I will show you their video. Let's take a closer look at the human on a chip. This chip generation has an area of a standard glass microscopic slide, and it consists 4 layers: an adapter plate, a nutrition plate, an organ plate, and a sensor plate. Now, a common blood vasculature interconnects the different organ models, such as the intestine, the liver, and the pancreas. Nutrients are absorbed by the intestine and distributed by the circulatory system. Insulin and bile is produced by pancreas and liver respectively. Food which is not absorbed is excreted. The kidney filters waste, excess ions, and chemicals from the blood to form urine. Additional organ models on the chip include the lung, the bone marrow, skin, adipose tissues, brain, and testicles. Now, with that arrangement, substances can be applied intravenously by direct injection into the circulation. The transparent sensor plate allows the electrical coupling of organs and microscopic access. Now, if you assemble the entire system, substances can also be topically applied onto the skin. Oral uptake and inhalation can be emulated respectively. Finally, the chips can be run fully automated over extended periods of time by employing state-of-the-art robotics. Such organismoids are supposed to be sufficiently flexible for diverse disease modeling and to bear the potential of ultimately replacing animals for systemic substance testing. Michael Schuler's labs and Linda Griffith's labs evolved toward MPS, integrating 10 and more culture compartments in a single chip format, and Linda has shown some of the examples just recently. They each use different cell lines and cells to test their prototypes in a 1 to 4 week co-culture experiments. I have highlighted the remaining technical challenges which they and we as an entire community are still facing with MPS. And I don't want to go through all of them, but another organismoid-specific biological challenge became apparent. All organs should be derived from one donor for a functional, immunocompetent human body on a chip. Otherwise, rejection will occur in the systems. Therefore, for our first organismoid prototype experiments, we established the background to derive all cells and organoids from single donor iPS cell banks. This approach reflects the fourth principle of our organismoid concept, the accelerated terminal differentiation of premature organismoids enabled by in vitro training programs for organoids and enforced by organismoid-based on-chip crosstalk. In more detail, we aim to emulate the ontogeny in humans, here highlighted in yellow, shortened to few months in the generation of healthy adult body replicates physiologically functional for long periods. They can then be utilized to emulate acute, subchronic, or chronic disease periods of an individual to precisely select the best fitting therapies for that particular patient. In the multi-organ chip universe, we have first proofs for iPS cell-derived tissue maturation due to on-chip crosstalk for bone and liver models in the respective 4 organ papers of Jordana's group, which has been presented 2 days ago, and from our own research. So we established qualified, internationally registered and published 6 commercial integration-free iPS cell banks. Subsequently, robust differentiation protocols have been developed and generated for 10 types of cells and organoids from a single donor bank. Thank you. We integrated these into the organismoid prototype chip and evaluated the 7-day performance. Immunohistostaining and gene expression analysis for key parameters have been performed, indicating stable homeostasis. We were able to stably endothelialize all microfluidic channels involved in the chip. However, similar to Michael and Linda, we haven't reached the final organismoid level yet. I have summarized our remaining challenges here. The on-chip hematopoiesis in the bone marrow is still insufficient. The capillary endothelial network could not be fully interconnected within the organoids. Consequently, we are not yet able to produce whole blood on the chip and perfuse and circulate it into each of the organs. The differentiation of testicular and immune tissues is still ongoing. And finally, innervation and lymphatics should be considered. Now, our Humimic multi-organ chip platform currently consists of protocols to establish 16 human organ models, as you heard in some other presentations here. Based on our organismoid design, we reverse designed much simpler universal chips of the same standard geometry to host 1 or 2, 3 or 4 organ models of any lower level of biological complexity, using always the same benchtop and automated equipment. 22 assays have been established at different levels of qualification yet. And we aim to use this specific knowledge from that different assays here to generate at this lower level of biology the remaining biological information to solve the challenges. I highlighted the respective programs adding missing information for the organismoid solution here. As an example, we recently managed to improve our autologous 4 organ chip. Now here they downstairs, difficult to read, I think. But I will give you a short glance on that. This physiological-based pharmacokinetic compliant iPS-derived 4 organ chip has been published a couple of years ago, but it lacked an endothelial blood-brain barrier. Now we have improved its performance by adding a functional blood-brain endothelial barrier differentiated from the same donor. That was an internal proof that the maturation of such a barrier can be exercised within an acceptable time frame. The BBB-specific endothelia have been cultured at the opposite side of the porous transwell, as you see here, membrane of the neurosphere compartment. High tier values and biomarker analysis pointed to BBB-specific endothelial properties. We benchmarked the functionality of the barrier independently, applying 3 drugs which expose low, medium, and high BBB permeability respectively in humans. A single exposure regimen at day 5 of the co-culture of the BBB containing 4-organ chip has been performed and terminated at day 7 after 48 hours. Drug levels reached equilibrium within 48 hours in the 4 separated biological compartments of the chip: the intestine, the surrogate blood circulation, the brain, and the kidney circuit. Among others, the blood and brain data here shown with red and green arrows perfectly matched the behavior of those drugs in patients. They displayed low permeability for atenolol, medium permeation for carbamazepine, and free passage of verapamil at the end of the 2-day pharmacokinetic profile. These results added a functional blood-brain barrier to the autologous PBB key-compliant chip, and the human 4-organ model is now ready to support ADME profiling, PK studies, and toxicity assessment. It further enables disease modeling, including the immuno-oncology domain, at a new level of human biology due to the autologous nature of all organs. Okay, let's come back to the organismoid topic. How can they add value to precision medicine? Now, when doctors diagnose a tumor in patients, they eventually can use organismoids to select the most appropriate treatment plan for the patient if they have available MPS cell bank. Therefore, for example, a set of 24 identical healthy organismoids of that patient could be generated in qualified test facilities. Then a specific number of organismoids are loaded with patient tumors, and various treatment regimens proposed by the physicians are then tested in the Humimic Autolab. The earliest, most appropriate result triggers the transfer of that particular treatment regimen to the patient. This should result in a comparable curative effect for the patient at best. Now, such chip-based precision medicine is characterized by a number of outstanding new features. For the first time, biological replicates of patients' bodies are available, which we do not have in clinical trials. This is expected to significantly increase therapeutic precision. In addition, the healthy body controls will optimize monitoring of remission. Normally, you don't have healthy body controls. Access to every tissue in every organismoid during the tests will provide previously impossible insights into disease progression. The approach is also suitable for mimicking clinical trials in the preclinical setting. As I mentioned, the community expects to see the first proof of concept not earlier than in 2030. At the end of my presentation, I would like to be crystal clear with the limits of MPS-based platforms. Due to its miniature size, an organismoid on a 3 by 7 centimeter chip cannot properly emulate the fracture of a large bone or precisely recreate the impressive pumping performance of a 300-gram heart. By definition, it also cannot model disorders of consciousness and mental illnesses where the entire architecture of the 1.3 kilogram brain is required. Given all that presented information, I envision future MPS-based automated test centers to become the core solution for preclinically informed development of new drugs and advanced therapies. Such platforms will enable individual treatment schemes best fitting for individual state of the disease and at a particular moment of your life. Furthermore, you-on-a-chip solutions will be able to predict health-promoting diets and cosmetics on an ethnic, gender, and even individual level in the future. And there are other consequences beyond healthcare. I expect the laboratory animal studies to be cut by at least 75% through implementation of such robust solutions, and product development will be also optimized in the future. That said, I would like to thank our team for its incredible impact and thank our partners for running that technologies too. thank you for your attention.
All right, thank you very much for this presentation. Other questions? Please raise your hand. I'm going to run. Okay, well, everyone's thinking about a question. The scaling, do you apply that also to your media? Do you go from, I think, 5 liters of blood down to about 50 microliters of?
50 microliter, yeah. We are scaling down all biological parameters, more or less, and that allows us then also to scale to have a direct in vitro to in vivo extrapolation.
I have a simple question about the vasculature. Do you use tissue-specific vasculature for all the organs? Because I saw for the blood-brain barrier, I was wondering, you do the same for kidney and intestine and everything?
No, we are developing from IPS, we are differentiating the endothelial cells for a number of genetic processes, and then adding that, we were expecting that they become organ-specific in the organs, but for the blood-brain barrier, we need the external pushing them to the blood-brain barrier characteristics.
Thanks. And related to the same question, are you able also to modulate the shear stress in every tissue differently? It's like the vascular shear stress, it's physiologically relevant in each tissue. Thanks.
Self-organization and right microfluidic flows will do that. So the shear stress is dependent upon the fenestration of the endothelium in those organs where they are fenestrated, which are only few. And in all the others, the shear stress is defined by our flow, and that is a physiological pulsatile flow.
Hey, very nice talk. Have you managed to make all the media that you have serum-free at this point?
Yeah, we do have medium serum-free medium for the 4-organ chip, and we are, and for all 4 organs, important, and that has been shown by others too. Important is that you have the barriers in place, because the medium in the circulations are then other and start to become other than these in the apical part of the intestine or in the urine, definitely, and in the brain fluids. We add human serum sometimes, or human albumin to that media, but the system produce their own proteins by the liver.
Thank you. And for the scaling problem again, if you have drug concentrations or molecule concentration extremely low, how you'd solve that problem? Will you use like parallel organ on a chip units, or how that can possibly be solved?
Oh, very good question. We try to go exactly through how the patient is doing it. So if it has a three times a day tablet, we downscale this tablet to 100,000, the concentrations, and give it orally, like in this case, on the intestine. If you cannot make that concentration with 1 or 2 drops, you start to deliver the drops over 5 minutes, because in the intestine, you anyway have some time to get the drugs to the intestine. It's not a gastric, but a post-gastric application.
Thank you very much, Uwe, for a wonderful talk. Our next speaker, Adrian Roth from Roche, will tell us a little bit more about how to use human tissue models to support clinical development and personalized medicine.
Thank you very much. Thank you for the kind introduction and for giving me the opportunity to present here today at this meeting. So first of all, for those who may know me from meetings on the same around the same topic before the pandemic, probably as you can see from my current affiliation, I decided to switch roles after 15 years doing preclinical research and investing a lot of energy in building up predictive models, including MPS, to characterize drug candidates to enable entry into human testing. I changed roles and moved into clinical research, joining a team that is dealing with personalized healthcare approaches, and where we're also very interested to explore and see to what extent microphysiological systems could help improve clinical development and optimize the benefit-risk ratio for our patients and driving personalized medicine approaches. So that said, today you're not going to hear anything about predictive tools that I've been working on together with the team over the last years. And actually that's also not necessary, because there were a couple of great examples during this meeting from my former colleagues, both on posters as well as presentations looking into this. Rather, what I would like to do today is share with you some of the ideas and goals and concepts that we're currently working on, and also give you some examples what we have been initiating studies. Clinical development has a bit of a longer time span than preclinical development, so those studies have not read out yet, so I'm not able to share with you like ready data and results, but I hope you find kind of the ideas and the concepts relevant and interesting. So how do we move this forward? Maybe here? No, here? How does this work? Now it works. Okay, so very generally, the basic kind of difference how we look at the problem, what problem we are dealing with. So a lot of the work that's being done with microphysiological system in the preclinical discovery space is around having models in place that are able to test quite large numbers sometimes of different types of drug candidates in order to select the best ones, those with the best properties, the most promising ones to move forward. And you typically do that with a handful of different cell systems next, of course, to other types of tools, which aim to recapitulate what I would say is like the average human. Now, when you look at the situation in clinical development, it's actually a bit a reciprocal approach, quite different, because here you typically have one single candidate where you invest into and which you want to move through the whole stages of preclinical development. But you need to be able to reflect the huge diversity of patients that you typically have. So once you have decided on a candidate, you don't want to stop that. You want to see, okay, which are the patients that are at risk, which are the patients that benefit most, and how can you find that out? And this is where we would like to introduce patient-specific organoids, or organoids that represent a specific patient subpopulation. Can we use those to generate a patient-specific response and then aim for an individual treatment? So the graph that you can see here, I took that from a publication or like a leaflet around some basics of personalized medicine. So fortunately, it's not that dramatic that 80% is waste, but it can happen. But I would say our preclinical work is a bit more promising and successful. So it's certainly not 80%. But as you can see, then in an example that I will be giving just later on, we do have some areas where actually we're really struggling with relatively poor, relatively small fraction of patients that do respond well. So overall, what is our vision? So we really see MPS as a window into our patient's individual physiology for a more accurate prediction of patient safety. And we think this could nicely complement what's already there. So a lot is already being done in the personalized healthcare arena for clinical development. So looking into histopathology, there's a lot of genomic work being done. So we would like to combine that and thereby improve the patient experience. Minimize hospitalization is an important topic for us in clinical development. Of course, we would like to predict the response and identify those patients that would benefit most. So kind of both efficacy and safety aspects that we want to look at. We want to use MPS to develop tailored approaches to manage those toxicities. So rather than just kind of in the worst case giving up if you have severe safety signals or even fatalities, how can we predict it and then early on proactively manage those toxicities so that in the end a drug which may work for a large portion of the patient population can actually be given to patients who suffer from diseases. We also would like to use it to define optimal treatment regimens. So I'm particularly thinking there of combination approaches in oncology. To my knowledge, in our current portfolio, there's not a single agent where the intent is to use the drug as a single drug. So it's always combinations. how can we define the optimal combination approach? We think MPS can also really help us there. And ultimately also optimizing trial design. This is a bit of a stretch goal. I mean, there the data really have to look very, very good and promising. I would like to later on give you an example, like predicting the response. So there's one example that I will allude to just later on, predicting immune-related adverse events in trials. This is something that we're struggling with a lot in the immune oncology space. And also another example about how can we identify those patients at risk when we know already there seems to be some signals maybe in phase 1 studies or in early phase 2 trials. And we do see a couple of patients that show some signals. What can we do by virtue of using microphysiological systems to develop mitigation strategies once we go into really heavy, large, very expensive trials? So very generally, a few minutes just on personalized medicine. So what is this actually? What are we doing here to get insights into patient-level drivers of response to improve outcomes and reduce the burden of healthcare on society? So this has a couple of different elements. First of all, trying to understand what are those factors that impact an individual response. A lot has been done already about genetic drivers. HLA typing is something that has almost become a bit of a standard as soon as you have something popping up there looking into this, and I will allude to that a little bit later on. Ancestry is something that's coming up more and more, and I was very pleased to see that actually also part of the MPS community is starting to think about that. Is quite tricky actually also, how do you define that? So ethnicity, ancestry, and the genetics underlying that, these are difficult terms to precisely address, but is absolutely a need, and is coming more and more also as a regulatory request that we look into that. Of course, also underlying diseases, age groups is again an important topic that I hopefully will be able to talk about later on. We have some projects in place using MPS to try and address different age groups, which again is technically a bit difficult, but we hope to be able to have data there soon. Of course, also in clinical development, it's a lot about prediction, biomarkers. Can we use MPS also to develop new biomarkers next to genetic testing? Monitored in patients, this is a topic a bit apart from MTS. Digital tools for remote monitoring to reduce hospitalization is an important element there, and also help physicians then for post-marketing in clinical practice. How can we guide them by providing them with additional information from our labs and from our investigations? So precision medicine then makes use of a couple of key enablers, key technologies, as alluded to for patient profiling. Genetic testing is currently being done at Roche for about half of the patients, I would say. So this is typically included, and most of the patients actually agree to the consent of being genetically tested. And on a routine basis, next to the normal clinical chemistry, things like cytokines, chemokines are being measured for patients. So this is an important aspect. Then in order to validate our systems, and this we need data, we need real world data. This has been part from my preclinical presentations over all those years, access to patient samples, patient cells, but also the patient data. I still think this is something we haven't solved yet. It's not so easy to access this data. Tools like natural language processing is something that we currently apply to really leverage kind of historic data, patient records that are in PDF format at best, or hard copies. How can we leverage that to have more clinical data that we can use to build those systems? Digital tools, I mentioned, this is a bit of a different topic. And what we would like to introduce now is a new kid on the block for clinical development and personalized medicine, patient-derived tissue models. So can we use these to in vitro to mechanistically understand and predict individual patient response? Now to a case example, cancer immunotherapy is an important part of our portfolio. When this whole thing started, it was believed that large molecules, they typically don't show lots of side effects, and we had to learn over the years, including also many other companies, that this is actually not at all the case. There's very promising results, sometimes really close to cure. However, as of today, we have to realize that only quite a limited number of patients actually respond to immune therapies overall. We have a relatively high rate of adverse events, and we don't have really good tools to predict them. And even more interestingly, and I will allude to that later on as one of our examples, that was really a starting point for our group. It has been observed by a couple of groups that about 70% of patients that had to be discontinued due to adverse events actually showed the best treatment response, hinting towards a link between the occurrence and the risk for adverse events in the immune oncology space and efficacy. And so we thought about that and did some prior investigations and are currently using that example to see can we do more with that for a personalized healthcare approach. So just very quickly here, so here's the whole range of clinical toxicities, going back to that statement that we heard earlier on when I worked at Roche, also in preclinics. Yeah, antibody-based drugs, they're fine. They would not induce any clinical toxicities. This is actually not at all the case. It's quite wide here. So these are two famous prototypical checkpoint inhibitor drugs, and you can see like a wide range, both in grades and also types of organs addressed. So we do need tools to address that earlier on during stages of candidate characterization preclinically, but also those well-characterized molecules. Once we go into clinic, this is how it looks like. So we need better tools to manage that. So what has been done over the past years, including our team at Roche, we set out to build a suite of different types of models, typically consisting of primary human tissue, polarized where necessary, adding PBMCs in flow. All the tissue components are either donor matched in the best case, if possible, or at least HLA match. And so this is the basic simplified setup of those systems. That have been set up, and those systems then have been used to recreate the key processes that we're interested. So the drug-induced activation of immune cells, and then initiation of downstream events at the site of action, and then there really have something that to measurable at the site of action in order to take decisions based on that. So in order to achieve that, kind of three main aspects needed to be solved for this example. Key event of immune cell activation. As you can see here, an example of the recruitment of leukocytes following stimulation of an endothelial system, then targeting the healthy tissue, as you can see here in the middle. These are hepatic spheroids, which have been treated with a PD-1 checkpoint inhibitor, and you can see the infiltration of leukocytes. And ultimately, in safety, we would like to see, so what's the consequence of that? Is anything happening? And here you can see, for example, this is classic chemotherapy-induced apoptosis of intestinal organoids. So that required over the years quite a bit of work to test out and benchmark these different models. And as said, you've seen now over the course of this conference some of the examples that have been shown. For example, the work Nicole Czeczorewski presented earlier on, where we did establish such models to use them to characterize and select the best molecules early on. Now, the same type of systems we do now also want to use in the clinical space, but it comes with additional requirements. So here, first, a bit of an outline, what kind of effects we're dealing with and where actually human tissue models can really be of help. Not everything can be solved with human tissue models. Cytokine release syndrome is still like a big problem. Somebody mentioned earlier the Ticinero case. That's quite some time ago. Whole blood cell assay has been set up as a consequence of that, and this is being done. Nevertheless, it has some limited predictive power, and still in clinic, it's something we see very often, and is still something that remains to be solved. Off-target effects is relatively rare for these type of molecules, and one can think of like peptide arrays and other things. Autoimmunity is a big issue there. We're very interested in T-cell repertoire sequencing that we do with patients. What I would like to focus on are so-called on-target off-tumor toxicities, because there you have already a bit of an idea based on the expression profile of your target, where it will hit, and this is where it has been used for example, for T-cell bispecifics, but also cell therapies, obviously. So this is where human tissue models in the preclinical space, but also in the clinical space, have been useful. Now back to the clinical example and what has really brought us to think about how can we make use of MPS for a personalized approach. This is one of these reports here, which shows us quite astonishing and also concerning data. So what you can see here, these are non-small cell lung cancer patients which have been treated with a checkpoint inhibitor. And the graph here shows survival and the probability of experiencing severe side effects. So different types of side effects, pneumonia, hypothyroidism, and so on and so forth. And what you can see here is a significant difference between patients that have immune-related adverse events versus patients that have no immune-related adverse events regarding survival. Yeah. So, of course, when a patient experiences immune-related adverse event, there's chances are that we have to take those patients off treatment if they really become severe. But as there seems to be this link between survival, we want to make use of MPS to try and address that. So how can we keep those patients on treatment while at the same time managing the toxicity rather than just taking them off treatment? So we first had a look into our own trials without going into all details. So these are just two examples of clinical trials with a Roche checkpoint inhibitor called atezolizumab. These are these two colored bars, immune-related adverse events in two trials that we ran at Roche. So you see quite a high incidence of adverse events, almost, it's a bit more than 30%, most prominently skin, but also GI and endocrine. And actually, as you can see on the right-hand side, we could confirm that the occurrence of immune-related adverse event was linked to better survival. So the team went out and analyzed those patient data, made a lot of genetic work and compared these values, the genetic data with the UK Biobank genetic resource, where they were looking for predictive genes that are associated with the risk for autoimmune type of diseases. So in this case, the examples were taken for autoimmune skin type of diseases like psoriasis or vitiligo. And so they built what is called a polygenic risk score. So the combination of different genes that pose, that can explain a risk for developing these type of autoimmune diseases. And actually they could see that a higher polygenic risk score, yes, it does correlate with autoimmune type of, in this case, skin toxicity. And as you can see on the right-hand side, this higher polygenic risk also correlates with longer survival. So people that are on the treatment for cancer and at the same time are carriers of risk genes for autoimmune type of diseases, they are more likely to experience adverse events, severe adverse events, but they also have higher chances for survival. So this has all been published. by our team in PNAS some years ago. And that was really the starting point for one of the projects that we are now kicking off. So we thought about, okay, if this is true, so can we use patient cells before they are treated? So as they are enrolled in a trial, to be able to predict if there's a risk for immune-related adverse events, and thereby, in a proactive manner, implement early on mitigation strategies, and at the same time addressing the tox, so while maintaining higher efficacy. So can we really solve that directly? So when you think of that, so if you want to test patients which are already in a clinical setting, so then you need to think of other aspects that become key and important next to the scientific criteria for, in this case, an organoid, a patient-derived organoid, whether you believe the data it generates is true or not. You also have to deal with minimal starting material. So you cannot just take like large amounts of biopsies, and patients will not agree to that. It has to be quite a straightforward, easy workflow. It has to be small, scalable approach that fits into a typically decentralized clinical trial setting. So when we think of a phase 2 trial, typically such a trial is not taking place in like one clinical research center, but we have patients decentralized over several places, because sometimes it's really difficult to recruit those patients. So you need to think of, okay, how do you organize all the logistics so that in the end you get fresh samples and you can process them into microphysiological systems. Furthermore, of course, as the clinical trial will not wait, the clinical trial will move forward. So it's important that the whole process is very short. So from biopsy to assay ready results where you can take decisions on. So this is not feasible if we're talking about several months there. So it really has to be short. Last but least, you need to bring that into the clinical protocol early on. And for, of course, the informed consent, which you may or may not get. So for some of these biopsies, we're talking about skin or also colonoscopies. So we're talking about roughly 50% or so of patients that would agree to such things. So you need to factor all of that in next to the tissue model that has to work, which is sometimes quite a bit of a challenge, and this is what we're currently working on. We do that together with the team from North Carolina. This is a startup called Silis. They have established a machine that is relatively small and is able to generate microphysiological systems, organoid type of very, very small mini organoids within 7 to 9 days. And we do that with different types of organs, and would like to see, so how well do now drug treatment data match with the patient outcome? So these are studies that are currently ongoing, and I hope to be able to report back maybe next time we meet here. So this is all in the making. The goal of this is, of course, that ultimately, if it's successful, that if patients enrolled in a trial can all be proactively tested, and then if we see that the organoid yields in relevant results, that we can define individual treatment, and also for safety define individual measures. The whole idea here is that when we look now at how personalized medicine works in a clinical setting in oncology, so this is from the MD Anderson Cancer Center. So what we would like to achieve is something like the molecular tumor board. So this is something that has been introduced over the last years. So this is a series of about 20-30 mutations that are routinely assessed for every cancer biopsy, which is then part of the treatment decision for a personalized approach. So these are the targeted therapies. And as you can see, we're actually well advised, or physicians are well advised to make use of that. This is from a recent review showing that actually a physician, if the team follows only the physician's recommendation for treatment, actually the chances for survival of the patients is significantly lower as compared to what the Molecular Tumor Board is proposing. So if we're successful here, we would like to have a seat on that table and add patient organoid data so that this then in kind of a holistic assessment of the individual patient can contribute to more tailored approaches. So this is what's currently ongoing. Now for the last minutes, I would just like to switch gears very quickly for another very quick example around genetically defined systems. So as some of you may know, HLA types, they very often predispose for certain drug-specific adverse events, which also have a regional pattern of distribution. So on the left-hand side here, you can see some of the most famous examples. Abacavir is probably the most famous one, leading to hypersensitivity reactions in such a severe manner that it's actually required to test patients for that particular HLA type before a decision can be made whether the patient receives the drug. This is also something which has been found in association with other types of treatments, like here for Crohn's patients that receive these types of drugs. There's an HLA association with the development of anti-drug antibodies. So we had kind of a similar problem here. This is data from a phase 2 trial where we saw a number of patients that showed ALT elevation. So no severe DILI, otherwise the program would have been stopped. But quite a high number of ALT elevations, and pharmacogenetic analysis revealed that it looks like there's an association with a particular HLA type. And also on top of that, as you can appreciate from the graph there on the left, this HLA type is associated also with a certain ethnicity. So we asked ourselves, can we recapitulate that? Can we model that in an in vitro system to see whether actually, is it confirmed, first of all, an increased risk? Can we better understand it? And can we take measures? Because this program is moving forward into phase 3. Longer treatment, and also patients that come with significant comorbidities. So if we have already a risk factor, and for example, then patients have already, for example, inflamed livers, there's a risk of potential high-low type of severe DILI. So this is what we want to avoid. So we would like to use organoids to identify those patients early on. And we do that together as part of a collaboration with Custom in Cincinnati. They have established a very nice system which allows us to generate those genetically defined liver IPS models, which we're currently working on, where we hope that this will help us to initiate closer monitoring, maybe think of co-medications, altered dosing, or in the worst case, excluding patients from trial. So this is what I wanted to share with you. This is the overall concept. In summary, we're interested in looking both efficacy and safety at the same time to generate kind of an in vitro therapeutic window, use that for clinical decision-making, assessing that window, defining optimal combination therapies, identifying patients at risk, identify patients that will benefit most, not forgetting that, optimizing trial design, and hopefully also reduce our development cycles. Some of these, for those who are interested, some of these thoughts and concepts have been recently published together with colleagues from Novartis, bringing together the whole genetic field with patient-specific microphysiological fields, use that for patient stratification and aiming for individual treatment. And with that, I come to an end. Would like to thank those people, both from preclinics and clinics, that are working with me on some of those projects, as well as some of the collaborators. Thank you very much.
Thank you very much, Adrian. I'm sure there's a lot of questions, but for the sake of time, we have to refer them to the breaks. So we're going to move forward to the next speaker, who is Julia Raggi from Alveolix in Switzerland. She's going to talk about a patient-derived lung-on-chip model for immunotherapy safety assessment. Forward.
Thanks a lot. Thanks. Good morning to everyone. Actually, these are not my slides. I don't know. You can maybe check on that. Okay, yeah, there we go. Thanks. Okay, so thanks a lot for having me here. I'm Julia. As they said, I'm working for Alveolix, and I will present you today how we can use our patient-derived lung-on-chip model for immunotherapy safety assessment. And actually, it will be a presentation of a tool, actually, that could hopefully solve some of the questions raised by Adrian just before me. So, as you might already have heard previously in this meeting, in the talks of my colleagues, at Alveolix, our main expertise are the lungs and lung models, and in a specific case, the air-blood barrier models. So what we want to mimic here is this very thin air-blood barrier that is in the lungs, which is the functional unit. And if we want to really simplify, the main key cells that are playing a role there are epithelial cells facing the air and endothelial cells facing the blood side. And these two are separated by this very thin basal membrane. So in order to recapitulate this in vitro, we came up with this system, which you also might already have seen if you pass by our booth in the previous days. So this is called AX12, and it's our consumable chip. And it's 12 because we have 12 independent units in the central well. In each of them, you have this very thin and porous membrane, which is also very elastic and soft. And actually, it's very easy to capture epithelial cells and endothelial cells on it, on the two sides of the membrane. And when you do that, you get to a very tight co-culture where the two cell types are very close in contact. And the thickness of this co-culture is approximately 10 micrometers, which is not so far from the in vivo dimensions of the human lungs. So here we have just a very brief introduction about the whole lung-on-chip system we have. Here, oh, sorry. Okay, sorry. So here you have the AX12 plate, so where you do the cell culture, as I just explained you, and then you will place it on this docking station, which is the interface connecting it to the two controlling units that we have. On this side, you have the exchanger, which is used to control the actions you have to do in your normal cell culture, like medium exchange or measurement of barrier tightness. And on this other side, you see the breather. And as the name says, it's the controlling unit that creates the mechanical stretch so that we can use it for mimicking the breathing motion in the lungs. So I will show you now. So the main frame in which we work is actually the drug development process. And as we all know, actually this is very time consuming and costly. And it's also not so cost effective if you think that among about 400 compounds that are tested in the preclinical phase, only one of them will become a real drug and reach the market. So as we know, microphysiological systems are already used and more used in the preclinical phase, and this is where they can help. They can help, for example, in the lead optimization or in candidate drug selection, and specifically also in safety and efficacy testing. And this is also where at Alveolix we've produced some data so far. So actually, to wrap up our vision, and I think we share this with a lot of you, is to try to accelerate and shorten this whole process, and so that first the patients can get earlier, can benefit from safer drugs and more efficacious drugs, and also so that hopefully we can avoid some unnecessary animal testing. So, but how do we do that? How can we provide predictive data so that we have a direct impact on the clinics? So what we try to do at Alveolix is to collaborate very closely with, on one side, industries and pharmaceutical industries, and on the other side, the clinics, so that we can understand better all the requirements and the needs of both sides. And this should help to clinically validate our model so that then it can be involved more in this process. For example, with industry, we already worked a lot with different pharmaceutical companies, and where we tested some drug candidates, and the data created there were already used in 4 IND submissions. And a bit more about how we try to get close to the clinics. So as also was mentioned by Adrian, it's very important to work with human materials so that we can also replicate patient variability. And this is also important a lot for us. That's why we try to establish very close collaborations with tissue providers and hospitals so that we get access to human tissue, and we can isolate cells and seed them directly on the chip and then treat them with the candidates. Not only that, we also can consult the pathology institutes and get some insights about the diagnostics. And we can also get some tissue specimens in case we want to test for biomarkers, for example, by immunohistochemistry. Also, what we can do, we have access to, or we can do genotyping and biomarkers checks, for example, in the blood. For instance, we did, for example, some HLA typing that was also mentioned by Adrian before me, which is raising in importance. And also, very importantly, we also get access to clinical data and history of the same donor. So you see how this setup that we try to establish can be helpful and to at the end provide something which is predictive and clinically relevant. And actually, one of the advantages of having this setting is that you can run many donors on the chip, depending on how much time you have. But and then this way you can simulate a small clinical trial on chip. So meaning that you can check for the proportion of side effects and the variability you have, as what also mentioned before. And you can also try to find predictive biomarkers defining why some patients react more than others. On the other side, you can also focus on more patient-specific responses, and this is more towards the personalized approach and treatment. And this way you can also study underlying mechanism of toxicity. So I will present you now a very specific case that we did on the chip, and where we evaluated the safety of a folate receptor 1 TCB. So this molecule is a T-cell bispecific antibody. These are antibodies that are engineered to recognize 2 different things on their arms. On one side, they have a anti-CD3 place of binding, so they bind T cells. On the other arm, they recognize a target, which in our case is folate receptor 1. This receptor is overexpressed in many solid tumors, for example, ovarian or lung cancer. And that's why it's a suitable target for cancer immunotherapy. However, and the way how it works is to bring together very close the T cells to the target cells, and this should form a functional immunological synapse, so that this leads to target cell killing. However, since this receptor is also expressed in healthy epithelium, for example, in the lung or in the kidneys, this kind of antibody has a high risk of on-target of tumor toxicity. And actually, this antibody was already tested in preclinical animal studies, and despite the high efficacy that was proven, and you see here how strongly it inhibited tumor volume in some patient-derived xenograft mice models. When they tested it in monkeys, they could find quite severe lung toxicity in terms of cytokine release in the respiratory tract and also leukocyte infiltration. So since they observed this molecule was stopped in the preclinical development and could not go further in the clinics. So what we wanted to do with our chip was to try to reproduce this toxicity so that we can kind of clinically validate our model and show that it can predict this toxicity. So in order to do that, we used our alveolix lung-on-chip system that I showed you before. We cultured on it patient-derived primary alveolar epithelial cells from different donors. And since they have a very crucial role in the mechanism of action of these molecules, we also included immune cells in this model. And actually, from here on, I will call the folate receptor 1 TCB only TCB. And in our study design, we also had a negative control to compare with, and it was a non-functional TCB variant. So first of all, we checked for target detection in the chip compared to the tissue. And as you can see, after culture on the chip, the primary cells still expressed folate receptor 1, even a bit more compared to tissue. And this makes sense because we isolated epithelial cells. We then proved the presence of the protein in the tissue by immunohistochemistry, and on the chip after cell culture of some days by immunofluorescence. After this, since we were sure that the target was there, we treated the chips with the TCB and the immune cells. And then we checked for different functional readouts. For example, we checked for barrier function. And as you can see, at the highest concentration, we had a 70% drop in barrier tightness, which means that the alveolar barrier got leaky. We also checked for pro-inflammatory cytokines release to check what was going on in terms of inflammation, and we could see that many of them were released a lot. For example, interferon gamma, TNF alpha, granzyme E, which are very, very typical markers of inflammation. We also checked for cytotoxicity. We did some live imaging with Caspase-3.7 probe. And after image processing and analysis, we could quantify that there were more apoptotic events in the presence of the TCB at the highest concentration. And actually, when we checked for membrane damage by measuring the LDH release, we also saw a trend of this molecule increasing this membrane damage, the cellular damage in the epithelial cells. We then wanted to analyze a bit more the immune cells, activation and recruitment. And actually, we could track them on the chip. And thanks to some image processing, we counted how many of the immune cells were still attached to the epithelium at the end of the treatment. And as you can see, there are many more PMCs remaining attached. So they are recruited and really interacting with the epithelium only when there is the TCB. So this proves the specific mechanism of action that we could reproduce on the chip. Moreover, we then harvested these immune cells from the chips at the end of the experiment, and we analyzed them by flow cytometry to check for activation. And we could find both CD4 and CD8 T cells that were activated. And we even had some degranulating CD8 T cells. So actually, this was a specific case study. And then we tried to zoom out a little bit to try to get a bigger picture and to understand a bit what's going on in terms of kind of population effect. So we pulled all the different data that were run in all the donors on chip that were channeled with the same TCB. And actually what we could see was that in 70 to 75% of the donors, we could observe vascular leakage and inflammation, but cell damage only in part of it, part of this donor. So this is a bit some kind of data that, as also Adrian mentioned, proves that we can capture the donor-to-donor variability in this system. And this, in our opinion, is something that we should take into account. So now, towards the end, so we proved that thanks to this model, we can reproduce the toxicity of a molecule that was not accepted in the clinics because of its toxicity, right? So let's imagine now we would use this model to assess the safety of a new drug candidate. And let's say it would fall here. So at a first glance, you could be happy because it's quite far away from the toxic response, right? But actually you miss a bit of reference because you still have this part, which is quite a large window. And how can you know if the candidate is safe or is not safe? Where is the threshold? And for us, what we try to do lately is to include additional controls in our study designs, which we call safe controls. And with this, I mean a molecule that was clinically approved, or even better if it's already on the market, so that you know that it was accepted. And this way, this can help to define better a kind of safe zone, so that you know if your candidate falls there, it will probably be safe in the clinic. So it helps you to assess the safety of this molecule. Yeah, so and we did some preclinical data about this on the chip, and you can see that we were able to distinguish the safe versus the toxic controls. And what is also interesting for me is that, for example, here at the cytokine secretion, you have already some levels of cytokine secretion given by the safe molecule, and this can really help you to know what is the acceptable level in the clinics. So just to conclude and to wrap up what I showed you, there are many advantages of working with primary human material. For example, you can simulate small clinical trial on chip and donor-specific responses, and you can correlate the in vitro outcome with the clinical data. Then I showed you a case study with the patient-derived chip model, and where we were able to recapitulate the toxicity of a molecule known to be toxic. And I hope like this, I convinced you that our immunocompetent model is suitable for safety assessment. And finally, you also heard a bit about our strategy for clinical validation in terms of including these safe molecules also in our study design in order to have something more predictive and a tool that helps you to define better the safety of your molecule. So that's it from my side. So thank you all for your attention. I would like to thank all my colleagues at Alveolix and also Laurent Cabon and team at Roche, since we did a lot of data with them. Thanks.
All right, thank you, Julia, for this very nice presentation. Are there questions?
Thank you for the nice presentation. I have a question regarding the configuration of your chips. You have a line microvascular endothelial cells underneath. So
Yes.
the PBNC are recruiting through and crossing the border and killing the epithelial cells, correct?
Actually, for this model, we added the PBMCs directly on the epithelial side to model the worst case scenario. So to model what would happen if they manage to cross because there is leakage and they would reach the epithelium. We wanted to focus on what happens if the immune cells reach that point and what do they cause to the epithelial cells.
And you apply mechanical stretching in the process during the experiments?
In this in the case study I explained, no, there was no mechanical stretch.
Can I make a challenging question?
Yeah, sure.
So what will be the difference between this and a transfer?
Well, we already see, it's a good question. We already see differences actually compared to transfer because already our membrane is very different, it's much thinner. So the cells first are in very close contact to the endothelial cells, while in a transfer, the membrane is much thicker, it's almost 3 times more. So this already makes a difference in building up the barrier and the model. And also the stiffness of our membrane is very soft compared to inserts is softer. So the cells should be more physiologically similar to the physiological situation. Then yes, the next step will be to add also the stretch and see what changes. But so far we started with a static setting.
All right. Maybe one short question from me. You're using multi-primary material. Have you looked into IPS-derived cells?
So not for this specific application. It's also one of the things we are looking into at Alveolix, to work with IPS and then derive the alveolar cells, but not only, since we are starting to do also to apply our technology to different organs. And yeah, this is also an option for sure, which is interesting.
All right. Thank you very much again.
Thanks.
Our next speaker, Summer from University of Pittsburgh Drug Discovery Institute, will tell us a little bit more about patient-derived iPSC microphysiological liver system as innovative precision medicine platform for optimizing clinical trial design of non-alcoholic fatty liver disease. Welcome.
Thank you. Thank you for the introduction, and thank you for the organization committee having me here. Good morning, everyone. I'm Summer. I'm a second-year PhD student from University of Pittsburgh, and now I'm working in Dr. Taylor's lab at the Drug Discovery Institute. I'm very happy to present our work today. So our goal is to develop a precision medicine platform using patient-derived iPSC liver microphysiology system for optimizing clinical trial design of non-alcoholic fatty liver disease. Yep. Non-alcoholic fatty liver disease, or NAFLD, is a multifactorial metabolic disorder. Many factors contribute to the development of this disease, such as environmental factor, metabolic syndrome, demography, gut microbiome, and the genetic variants. Due to the complexity and the heterogeneity of this disease, NAFLD is recently proposed as metabolic associated fatty liver disease, MAFLD. So the starting point of our project is focusing on one of the most common variants that have been identified in the gene PNPLA3, as highlighted here, which I will talk about it more in my later slides. So now let's take a look of the disease progression of the NAFLD. So NAFLD usually started with the accumulation of the lipid droplets in the liver. The accumulative of the lipid toxicity will trigger the pro-inflammatory cytokine released from the immune cell. And it will further lead to the activation of the astrocytes for the fibrosis genesis. So the disease will progress from the early stage of NAFLD to an advanced stage NASH and to a later stage cirrhosis. So a good disease model should be able to recapitulate the disease progression, as I mentioned here, and as well as the key disease phenotypes. So as you can see from this map, NAFLD is a very common disease globally. And the high-risk variant of PNPLA3 is also a very common variant. And you may also notice here, You may also notice here, this high-risk allele is more common in certain population with a higher prevalence of NAFLD, such as the Hispanic population or the Latino population. Thus, the prevalence of this PNPLA3 high-risk variants accounts for a large fraction of the inter-ethnic variability in the NAFLD susceptibility. So our initial strategy to address the heterogeneity of the NAFLD is to stratify the patient population based on their genetic variants. So in this case, it's this PNPLA3 high-risk variant. So we will stratify, we will group the patients by the genetic variants for a more effective and more targeted treatment. So far, we have recruited more than 200 patients through UPMC Flow Clinic. And we have genotype and identified different genetic variants from our patient cohort. So you can see list all the variants list here or has been identified in the GWAS study showing association with the NAFLD. And they have a range of different effects. And PNPLA3 high-risk variants is the major one with, is the most common one with a major effect. And you can see this is a C to G mutation. And the C is the major polymorphism and G is the minor one. And you can see from on the right, we compare the genotype frequency to the different disease condition. We have the healthy individuals, we have the NASH group, we have the end stage of the liver disease here. And the minor homozygous are showing in the yellow box, and the heterozygous and the major homozygous are in pink and purple respectively. And you can see compared to the other variants, the genotype frequency of PNPLA3 is increased along with the disease progression. And very interestingly, on the very right, you can see the prevalence, the presence of this polymorphism is not necessarily related to the development of type 2 diabetes. Now you may be wondering, what is the biological function of PNPLA3? PNPLA3 encoded a protein that is highly associated with lipid metabolism. In the normal condition, this protein is located on the lipid droplets, and it functioned as a hydrolase to facilitate the metabolization of triglyceride and the fatty acid in the hepatocytes and the retinol in the stellar cells. However, the mutant form of this protein will block this process, which lead to the lipid accumulation in different cell types. So we used human primary hepatocytes to examine the intracellular lipid content with or without this mutant. And we used NAILRAD to detect the lipid content. NAILRAD is a dual probe. It will turn fluorescent green in a lipid-rich environment. Otherwise, it will stay in red. So you can see from the bottom image, which is the mutant hepatocytes, it shows a significantly higher intracellular lipid content. And the quantification here indicated that the normal human hepatocytes carrying this PNPLA3 high-risk variants present significantly higher content of intracellular lipids. But as I mentioned before, NAFLD is a complex disease resulted from the interaction of different cell types. That's where our model come into the story. We have built the lipid acinos microphysiology system, or LAMPS, to recapitulate the major structure of function, major structure and function of human liver. The liver acinos is the functional units of the liver, which consists hepatocytes, Kupffer cells, stellar cells, and the sinusoidal endothelial cells, and the cholangiocytes. The blood flow in it create the oxygen gradient zonation. Based on this physiological structure, we build our LAMPS model. LAMPS is a 3D microfluid device. The primary hepatocytes co-cultured with the key non-parenchymal cells, as I mentioned here, and we will culture them for 10 days with a flow exposure. It can be ran in either zone 1 or zone 3 oxygen level. To recreate the physiological and the pathological environment, we designed 3 medias, starting with the normal fasting here. Oh, sorry. Starting with the normal fasting here. The normal fasting media is to recreate, to mimic healthy liver condition with a normal level of glucose and insulin. And then we are able to drive the disease to the early metabolic syndrome, EMS, with increasing level of glucose, insulin, and the addition of free fatty acid. And next, we're able to drive the disease to late metabolic syndrome, LMS or NASH, with the immune stimuli such as LPS and TGF-beta. We also develop a matrix to quantify the disease-related phenotypes such as albumin secretion, hepatic steatosis, oxidative stress, inflammatory cytokine release, stellar cell activation, fibrosis, and etc. And all the measurements here are consistent with the clinical data. Sorry, it's frozen. Okay, yeah. To build a patient-specific liver MPS system required the collaboration from clinical side to the bench side. This is a multi-PI work funded by NIH. We have Dr. Bihari. From the UPMC Flow Clinic. We have Dr. Suto Gutierrez from the Pathological Department at the University of Pittsburgh. And we have Dr. Taylor's team from the Drug Discovery Institute at the University of Pittsburgh. So in our first aim, we will construct the liver MPS system using primary liver cells with or without this PNPLA3 mutant. We will drive the disease progression in our model. And the data came from here will serve as the fully adult reference for the later iPSC-based liver MPS. And we will build our model using iPS, using patient-derived iPSC cells, and the result we will compare the results to the first aim by comparing to the primary-based liver MPS. We were able to answer the question: to what degree this iPSC-based liver MPS is able to recapitulate the adult liver function and the disease phenotype. Okay, in the end, we are going to use our patient-specific liver MPS for clinical trial optimization. Regarding of the primary cell-based liver MPS, now currently we are able to evaluate our first set of human primary cells with or without the PNPLA3 mutant. So as you can see from here, we examine the steatosis phenotype by quantifying the intracellular lipid content. As shown in the graph, compared to the wild-type model in the blue, the mutant model in the red show a significantly higher intracellular lipid accumulation in the disease-induced environment, showing in here EMS and LMS. In addition to the intracellular lipid content, we also measure the secreted triglyceride. As you can see here, we compare the wild-type model and the mutant model. We see a significantly higher increase of the secreted triglyceride level in the late metabolic syndrome media. So taken together, our preliminary results indicating that this PNPLA3 mutant may introduce a burden in a disease-induced condition, and it may further accelerate disease progression. Now we are working on more lots of the human primary hepatocytes to confirm our initial results here, and more characterization matrix are carrying on to evaluate the disease phenotypes such as fibrosis or cytokine release. Oh, sorry. Is it frozen again? Oh, okay, sorry. Yeah, regarding of the iPSC-based liver MPS, we are going to generate the liver, the iPS-derived liver cells from patient PBMC sample. In our initial step, we will integrate iPSC-derived hepatocytes, stellar cells, vascular endothelial cells, and the cholangiocytes into our liver microphysiology system. In the future, we will also integrate the iPS-derived immune cells in our model, such as macrophage cells. The iPS-based liver microphysiology system is still at a very early stage. We have tested a range of different conditions and parameters and developed an optimization protocol to maintain the iPS-derived hepatocyte viability and the maturity in the LAMP model after 8 days with flow exposure. As you can see here, the IHABs co-cultured with non-parenchyma cells in the LAMP model for 8 days with flow exposure. And the image showing here represent the area of interest on our chip. And the GFP expressing IHABs found a nice and homogeneous distribution on the chip. And at the bottom image, you can see the red signal is HNF4 alpha, which is represent the adult hepatocyte marker, indicating the maturity of the IHABs. We are able to maintain about 70% of the maturity of our IHABs in the LAMP model for 8 days flow exposure. Meanwhile, we also developed disease media to induce the fatty liver in our model, and the fatty liver disease is induced by the addition of free fatty acid. Compared to the control group, we see a significantly increased of the lipid content here. We have DAPI here showing the nuclei, and the red signal is from lipid staining. And from the quantification, we can see the lipid accumulation is threefold higher in the disease-induced media with the similar nuclear with the similar cell amounts showing the nuclear count. And in this study, we use biosystic platform to establish and evaluate our reproducibility of our model. And this platform is very useful. It provides tools for us to evaluate and review our experiments with different settings. So our IPS-based model is still in development stage. So here is showing one example that how we use this platform. And the lines here you can see is from three different IHABs batch, and we collect all the data and put into the database. And you can see you can see highlight here our reproducibility is getting better and better. And now is the summary of my talk. We have recruited and genotyped more than 200 patients for this study, and we have established the protocols for the major liver acinos iPS-derived cell types. We have evaluated the first set of human PNPLA3 low-risk and high-risk hepatocytes in the LAMP model and found differences in the intracellular lipid content and triglyceride released between them. We're working on more measurements, including fibrosis and cytokine release panel. iPS-derived hepatocytes have been demonstrated to maintain the phenotypic integrity and the maturity on the flow for at least 8 days. So all these results have paved the way toward our final goal, to optimize clinical trials using patient-specific liver MPS system. So last but not least, I would like to thank our wonderful group and the great collaboration, and also thank NIH for the financial support. And that's all my talk today, and any questions or comments are welcome. Thank you.
Thank you, Summer. Wonderful talk. We have time for one question. Okay, I will take it then. In the projects that you are involved in, you have a couple of aims. Aim 1, where you assess the disease phenotype in the primary cells, and in aim 2, you're actually generating iPSCs. Are those iPSCs coming from the blood of the patients from the A1?
For M1, we are using the cryopreserved primary hepatocytes. In the patient-derived iPSC in the M2, we will using the patient that we recruit in the clinical trials that I mentioned. Yeah.
Those gonna be different patients,
Different
not
patients.
this 200. Okay.
Yeah.
Thank you very much.
Thank you.
All right, thank you very much again. And we're coming to the last presentation of this session, but last but not least, we have Sarah Shelton from the MIT giving a presentation on microvascular model incorporating cancer-associated fibroblasts and immune cell perfusion. Looking forward to the talk.
Thank you. Thank you all for being here, and thank you for the opportunity to speak today. I'm Sarah Shelton, and I'm a postdoc at MIT and also at the Dana-Farber Cancer Institute under the supervision of Dr. Roger Kam and Dr. David Barbie. So today I'm going to tell you a bit about my project that really is involving vascularizing patient tumor microenvironment models in order to do immunotherapy studies. So the overview of our approach here is to develop patient-derived models by taking patient surgical tumor resections and then disassembling that into the different cell types that we need and then reassembling them in our model. So starting with taking a surgical resection from the OR, we mince it into small pieces and then sequentially filter it first through a 100 micron filter and then through a 40 micron filter. And so that gives us 3 different size populations of cells and spheroids coming from this single patient sample. So first, the intermediate size fraction from 40 to 100 microns, those are our PDOTs, or our patient-derived organotypic spheroids. They still retain tumor cells, stroma, extracellular matrix, and immune cells. So we call them organotypic spheroids instead of tumor spheroids. And so the Barbie lab at Dana-Farber in conjunction with CAM lab has already done a lot of work characterizing the PDOTs and also comparing them to MDOTs, or murine-derived organotypic spheroids, and looking at response to immunotherapy, immune checkpoint blockade by doing live dead staining. We've looked at the populations of immune cells in the PDOTs relative to the primary tumor and found that these immune populations match. And so I really wanted to take these PDOTs and then put them in a vascularized model. So I think for doing immunotherapy studies, it's great if you can put your PBMCs or your T cells with your tumor spheroids, but I'm really interested in the extravasation and the role of the vascular biology in this process. So we need T cells, and I'm getting that from the surgical resection as well by expanding the T cells in that single cell small populations using IL-2 to expand the tumor infiltrating lymphocytes. And so then we also need the cells in order to create the microvascular networks. So the large size fraction that's not the immune cells or the PDOTs, I take that and further enzymatically digest that, and I'm getting out of that the cancer-associated fibroblasts. So that's the stromal cell population here. And then originally we were aiming to use the tumor endothelial cells, but we weren't able to isolate those in sufficient numbers. So we went ahead and started using the HUBEC in this model. And so I'll show you what happens when we start to build up this model from these individual components into the sort of full tumor microenvironment model. So if you're not familiar with our methods for generating microvasculature, we use the vasculogenesis model, and it works pretty simply by combining endothelial cells and stromal cells, typically fibroblasts, in a fibrin gel, and the fibroblasts support the connection, elongation, and luminization of the endothelial cells into perfusible microvasculature over the course of about 4 to 7 days. So the perfusible microvasculature ranges in diameter from about 2 on T to 200 microns. And we can co-embed tumor spheroids into this model at the time of seeding as the microvascular networks are forming, or you can preform microvascular networks and then add tumor spheroids to a port in the model. So the microfluidic devices for this model are very simple. So you need a single channel for your hydrogel containing the cells that forms the microvascular networks. And then we have 2 flanking side channels for fluid flow in order to support the culture with tissue culture media. Or once the networks become perfusible, that's how we introduce T cells and have them perfused through the system. So since I am using cancer-associated fibroblasts in this model to generate the tumor microenvironment mimicking model from these patients, I think it's worth discussing a little bit what a cancer-associated fibroblast is and where they've come from. So the sort of most simplified version of what a cancer-associated fibroblast or CAF is it's a normal fibroblast that's been activated. And so typically the most well-known mechanism of activation is TGF-beta secreted by the tumors, but there are also a number of other inflammatory and mechanical signals in the tumor environment that can activate fibroblasts and produce those CAF. So CAF from different tissue and from different patients are heterogeneous. There's definitely a number of different CAF subsets, and they are often categorized as myofibroblastic CAFs, inflammatory CAFs, even antigen presenting CAFs. So what this means is there's not really a single CAF marker that you could sort of pick out and identify this is definitely a CAF. But they should definitely lack epithelial, endothelial, and immune cell lineage markers. And they have elongated fibroblastic morphology, but you want to make sure they don't have mutations so they're not EMT cancer cells. So again, several of these markers exist. They're not exclusive, but they are useful to sort of confirm that you are looking at CAF rather than normal fibroblast. So PCR on average on this cancer-associated fibroblast population found that there's no big change in vimentin expression when I compare that to the normal fibroblasts. And vimentin is a sort of a pan-fibroblast mesenchymal marker. But there are upregulation in several CAF markers, including fibroblast activation protein, caveolin-1, alpha smooth muscle actin, and fibroblast specific protein. So here it's worth mentioning that the normal fibroblasts that I'm comparing the CAF to are not from the same patient. We didn't have enough tissue to sort of isolate normal versus CAF from a single patient, but I'm using primary fibroblasts from healthy donors. And so most of my cancer specimens are from head and neck cancers. I have several oral cancers, oral cavity cancers that will be discussing, and also thyroid and a subset of lung cancers. So in order to use matched primary fibroblasts, I also have lung, thyroid, and dermal fibroblasts to compare to. So starting off building this model, we were sort of building it up piece by piece and began with building microvasculature, combining the HUVEC endothelial cells with the cancer-associated fibroblasts. So now I'm going to be showing you maximum intensity projections of the microvasculature in these models. And in this case, these images are perfused with green fluorescent dextran, so you can see which segments of the microvasculature are perfusible. And so right away, it was really obvious that there was a lot of variability from patient to patient. So everything was seeded in identical conditions and cell numbers, and we see really broad differences between patient to patient CAF and how they support the morphology and the generation of microvascular networks. So you can see from one extreme in the top left where we have sort of minimally perfusible networks to the other extreme in the bottom right where we have really wide flattened networks that have sort of lost their cylindrical cross sections. But the majority of the samples lie somewhere in the middle. So I took a subset of all my patient-derived fibroblasts, and I compared that to the normals, and I started looking at different aspects of the vasculature, including the morphology and the permeability. So to look at the geometry, I used a program in MATLAB called AutoTube in order to mask and skeletonize the vessel projections in 2D in order to quantify the mean diameter, the number of branches, and the branch length. And so there was a significant increase in the mean diameter in the CAF networks compared to the normal. It was a small increase of about 5 microns. And additionally, a significant decrease in the number of branches while an increase in branch length. And so from the morphology parameters, we can also get functional parameters such as permeability by, again, perfusing with the fluorescent tracer like dextran, and then measuring the change over time of the intravascular to vascular to extravascular signal intensity of this dextran. So here I'm showing you 2 maximum intensity projections from the normal fibroblasts, since I haven't shown those yet. So here's a typical image of a lung fibroblast network and a thyroid fibroblast network from healthy donors. And I haven't shown you the dermal fibroblast networks because in the identical conditions to form all these other networks, those networks were not perfusible. So in the normal lung and normal thyroid fibroblast networks, permeability is fairly low. And pretty consistent. However, when I compared across fibroblast types, there's a lot more heterogeneity, a lot more variability, and also higher permeability. And so this is in line and consistent with the literature about the permeability in the tumor microenvironment and enhanced permeability and retention effect in tumors. And this is due to the fibroblasts alone, rather than the typical thought that the tumors are secreting VEGF and contributing to permeability. So yes, as you might imagine, we're wondering why these fibroblasts are promoting differences in the vascular morphology and function. And so our first thought was VEGF, vascular endothelial growth factor. It's a pro-angiogenic factor, and we wanted to look for that in the system. But we were also curious of what else these fibroblasts might be secreting that would be impacting morphology and permeability in these vessels. So I did a Luminex assay and cytokine assay for looking for angiogenesis markers. And I think we're frozen. There we go. I also simplified the model in order to look at sprouting angiogenesis. So a lot of these features had already pointed to this pro-angiogenic phenotype as we expect in the tumor microenvironment. So I simplified the model down to the hydrogel with the embedded fibroblasts and now localizing the endothelial cells as a monolayer on one side. So as we apply the monolayer, the endothelial cells will then sprout into the fibroblast-laden gel, and then I can image the fluorescent fibroblasts and do image processing to crop and segment and quantify the degree of sprouting in each of the different conditions. So it's really apparent right off the bat visually that the CAF are promoting much more sprouting than the normal fibroblasts. And so we see longer, more developed, more abundant sprouts in these CAF models than the normal fibroblasts. And when we quantify this, so I quantified sprout area, density, and length, and I found significant increases in all 3, ranging from about 1.5 to 2.5 fold increases in sprout length, sprout area, and sprout density. And so again, we think that this is probably related to VEGF, and the cytokine array reiterated that fact that VEGF, the vascular endothelial growth factor, is about 5-fold increased on average in the cancer-associated fibroblasts over the normal fibroblasts, again, with some heterogeneity between the patients. And so again, we would expect VEGF to show up in this screen. It's expressed by cancer cells, fibroblasts, macrophages, a lot of the stroma. It's really well-known factor in the tumor microenvironment to play a role in angiogenesis, typically in response to the hypoxia that occurs in the tumor microenvironment and the HIF-1 alpha signaling. And of course, there are several FDA-approved drugs for VEGF and the VEGF receptor approved for combination therapy in oncology. So as the title of this slide hints at, the other factor that we found was CCL5, and this was a little bit less expected. CCL5 or RANTES was increased just over 2 folds change from the normal fibroblasts. And it's typically a T cell chemokine. It's expressed by activated T cells, although in the literature, it's also known to be involved with angiogenesis, typically through VEGF stimulated angiogenesis, but also related to endothelial cell proliferation and migration. And then there's also a great deal of literature about its effects directly on tumor cells and in response and making them more responsive and to tumor progression and metastasis. As we saw, VEGF and CCL5 were both implicated in the system and highly expressed by cancer-associated fibroblasts. I started looking at whether or not we could tease out their different effects in the sprouting population in fibroblasts. So now I'm using cancer-associated fibroblasts and reminding you what those control sprouts look like here. And then if we block CCL5, we see a really noticeable reduction in the length, the number, the density of the sprouts. And then furthermore, if we block VEGF, it becomes an even greater reduction in the sprouting. And it looks pretty similar when we block CCL5 and VEGF simultaneously. And so this is all using neutralizing antibodies and a research grade bevacizumab VEGF. So quantifying this data, looking at 4 different cancer-associated fibroblasts and looking at the sprout area as our indicator of sprouting. And we see reductions from the control conditions to all of the conditions, anti-CCL5, anti-VEGF, and the combination of both. And interestingly, if you look at the blue and the orange bars, those are CAF 1 and 3, and those had the highest baseline production of CCL5 in my cohort. And that's where we see the greatest reductions in sprouting when we apply the anti-CCL5 treatment alone. So as expected, when you block CCL5, it's most effective if the cells are already secreting quite a bit of it. The 2 groups where there was no significant difference was anti-VEGF treatment versus anti-CCL5 plus VEGF. So this sort of reiterates that VEGF is the dominant effect here, but that CCL5 is probably having a synergistic effect in the tumors that's producing this great amount of sprouting compared to the controls. So we started thinking more and more about like, why are we seeing CCL5? And then we looked through the other factors that were increased in the cancer-associated fibroblasts. And we're thinking about like, a lot of these factors are overlapping in between promotion of angiogenesis and inflammation. So CCL5, I've already discussed, is a T cell chemokine. However, it also is known to recruit monocytes. And then CCL2, or monocyte chemotactant protein, was also high on our list of hits. And as it sounds, it's a monocyte recruiter, but it also can be used to recruit T cells. And as we look through the other markers in this list, and things like Ang2, CXCL4, and placental growth factor, and started looking into the literature, what really sort of popped out is that we were sort of looking at a dichotomy between T cell and monocyte recruitment. And so since we have perfusible microvascular networks, that's something that we can test directly. So we started looking at how angiogenesis and inflammation are connected in this tumor microenvironment model that's created using the cancer-associated fibroblasts. So in order to quantify immune cell recruitment in this model, we label primary monocytes and T cells with different color fluorescent dyes. In this case, this is from blood from healthy donors. And we perfuse equal numbers of both cell types, perfuse them for an hour under gravity-fed flow, and then wash out the unbound cells before quantifying the retention of and relative abundance of each cell type in these models. So here are the images, maximum intensity projections of typical microvascular networks with normal fibroblasts, or microvascular networks generated with CAF, with the same endothelial cells here from the HUVEX. And the monocytes are labeled in magenta and T cells are in green. And so you can see in the normal fibroblast networks that the number of immune cells is relatively low. We haven't treated this with any inflammatory cytokines in the media. It's just the contributions of the 2 cells in this fibrin gel. And on the other hand, when the CAF are in this microenvironment model, we see a lot greater infiltration of the monocytes in pink and the T cells in green. And when we quantify the numbers, both monocytes and the number of T cells were significantly higher in the CAF models than the normal fibroblasts. And interestingly, in both normal and CAF, we see higher numbers of monocytes compared to T cells in both model types. So getting back to the holistic model that we're trying to develop to have patient-derived models of the tumor microenvironment, we really want to be able to do immunotherapy studies on personalized patient models. So we have the CAF, we have the tumor spheroids, and we have the T cells. But again, if we want to use the microvascular networks, we really need an endothelial cell, and we need an endothelial cell model that's not going to conflict with our T cells and produce a graft versus host response. So we could think about doing HLA matched endothelial cells and have like a library of endothelial cells to match for each patient. But we took a different approach, and we were looking at trying to make the endothelial cells invisible to the T cells. So T cell recognition is the T cell receptor binds to the MHC peptide complex on the target cell, and the MHC class 1 has one fundamental building block, which is the beta-2 microglobulin, or the B2M protein. And so we knocked that out in our HUVEC cells in order to make HUVEC that would be invisible to these CD8 T cells. So we knocked out B2M from the HUVEC and from healthy fibroblasts in order to make immune invisible microvascular networks. And so we knock out B2M, and then they don't express MHC class 1. So we can form really beautiful microvascular networks with these HUVEC and fibroblasts that don't express MHC class 1 via the knockout of B2M with CRISPR. And so that is going to allow us to combine these fibroblasts, healthy fibroblasts, and these HUVEC with any of our patient models. So we have sort of a universal HUVEC now to go with all of our models. And so the full model picture is to combine these B2M knockout HUVEC either with our cancer-associated fibroblasts or with our B2M knockout fibroblasts. And we could also combine those two in different ratios to look at the impact of the CAF on the microenvironment. And when we combine those endothelial cells and stromal cells, that's what generates our microvascular networks. And within those microvascular networks, we're embedding the PDOTs, those patient-derived spheroids. And then after about a week of culture, we have perfusible networks, and then we can flow the T cells through the system in the presence or the absence of a treatment, such as the immune checkpoint blockade. So the overall goals of this are to do long-term recirculation of these lymphocytes in order to image their extravasation and migration through the tissue to the tumor spheroids. And then downstream of that, with treatment and with and without treatment, we want to do cytotoxicity monitoring of live dead. And we can also sample media from the system to do cytokine and look at increases in things like granzyme B, TNF alpha, interferon gamma. So the model is still in development, and we're building it in the piecewise fashion. So on the left side of the screen, you're going to see melanoma, patient-derived melanoma spheroids labeled in green. These are in a microvascular network generated with those B2M knockout HUVEC and B2M knockout fibroblasts. And so you can see the microvasculature forms nicely around the small spheroids. And then panels B and C on the right, we have microvascular networks formed with the B2M knockout HUVEC and in combination with cancer-associated fibroblasts from one of the patients. And then I've perfused T cells from the matched patient sample. And so T cells in this model flowed through the model. They adhered to the endothelium, they were trapped, and they extravasated. So in the panel on the far right, you can see the T cell in red extravasating from the microvasculature, and then one farther down in the image that has already migrated into the interstitial space. So in conclusion, I want you to all to see that the cancer-associated fibroblasts support vessel formation in this vasculogenesis model. And this resulted in slightly larger diameter vessels, changes in the number of branches and branch length, as well as increased vascular permeability to 70 kilodalton dextran. And so while we saw a range of geometry and phenotypes in these cancer-associated fibroblast networks, most of them live somewhere in the middle here with nicely perfusible vessels that look quite similar to our normal vessels. So these differences are probably due to the secreted factors expressed by the cancer-associated fibroblasts that induce both angiogenesis and inflammation. There seems to be a lot of crosstalk between those two. And the CAF resulted in much greater sprouting than the normal fibroblasts, which was able to be inhibited by blocking CCL5 marginally, and then more completely by blocking with VEGF or VEGF and CCL5 in combination. Finally, the CAF networks retain more of these immune cells, as we saw in the images and quantifications there. And so as we put all of these model pieces together, we're really trying to build these patient-specific models of the tumor microenvironment, where the microvasculature is a fundamental component in order to model T-cell extravasation, migration, and homing to these tumors, in order to model more of the components and the steps that are involved in response to immunotherapy, and then hopefully to be able to look at patient-specific responses to immunotherapy in these models. I'd like to thank the CAM lab and the Barbie lab at Dana-Farber and all of my collaborators.
All right, thank you very much. Since we're over time, we don't have any time for questions, but I want to thank all the speakers from the session again, and I'm sure you'll find them in the breaks if you have questions. Thank you all, and I'll see you in the next sessions.
Nicole Kleinstreuser. Her talk today is going to be on MPS Core, a global working group applying open science to tackling a pandemic. Thanks, Nicole.
Thanks, Jason and Joachim. Do we have a timer? So I know, it's fine, I have a watch. Okay, so yes, my name is Nicole Kleinstreuer and I'm the acting director of NICEDM. I realized that I probably should have put a slide at the beginning to tell you what NICEDM is, so I'll just tell you verbally that we are the US federal resource for alternatives to animal testing. So we're housed within the division of the National Toxicology Program at the National Institute for Environmental Health Sciences, but we provide support to a committee of 17 different US federal agencies that either require or consider chemical safety testing data and are interested in moving away from outdated, low throughput, potentially uninformative animal-based testing methods and moving towards more rapid, more human relevant alternatives to animal testing. So we have a very broad program of work that covers pretty much every ISID you can think of in the alternative space. And usually what we do is really focused around toxicological testing and regulatory decision-making and coming up with alternatives to animal testing to support those processes, including MPS. So this is a little bit outside of our wheelhouse in terms of more of a biomedical research area and doing infectious disease research. But I think as every scientist in the world felt at the beginning of the pandemic, we wanted to do something to help. And so that was the motivation for starting this global working group. And I have to make a special mention of Anthony Holmes, who's a senior scientist at the NC3Rs, which is the UK 3Rs organization, because he co-chairs this working group with me. So the founding members of this working group, as I mentioned, Anthony Holmes is the co-chair, and then we have representation from NCATS, obviously due to their leadership in the tissue chip space and the tissue chip consortium. And then from NIAID, which is the Allergy and Infectious Disease Institute in NIH, we have Mark Williams, and then we have two representatives from the Department of Defense, Tyler and Kyle, who are doing really excellent work. And you may have seen some of Tyler's posters at the poster session last night. And they're leading a lot of work in the tissue chip space, particularly as it relates to infectious disease research. So then we started reaching out to the community through the networks of our founding members. And one of our key objectives was to involve as many regulators as possible and folks in government agencies so that we could really start to have these conversations about what are sort of the readiness criteria, what are the contexts of use, what is going to help move MPS into the forefront as far as being acceptable for forming part of regulatory submission packages. And obviously we see COVID, as a disaster, but also an opportunity, partially because the animal models for COVID are quite bad. They don't get sick the way humans do. In fact, basically the closest one is the golden hamster. I don't know what that says about us as a species, but particularly the non-human primate models are very bad. And so there's this really strong opportunity for tissue chips to kind of step up and provide human relevant insight into the mechanisms of the disease, the pathophysiology, and maybe provide platforms for testing new therapeutics. So we have representatives from divisions, branches, or centers of all of these organizations. We have, I think, every biotech company that was here at the World Summit, or almost everyone represented on the working group. And again, if you're from a biotech company and you don't see your symbol represented on the slide here, we should remedy that. That's actually one of the objectives of the presentation today, is to continue to spread the word about the working group and recruit additional members. And then we have obviously some large pharma companies represented. These are the folks who are really leading the field in developing therapeutics, and so are quite interested in identifying relevant testing platforms that they could use to test their lead compounds. And then by far the largest representation that we have is from academics. So I think that's where, as we've seen over the last several days, so much exciting work is happening in the research space in developing new MPS models and platforms. And so we have so many different universities and groups from universities. This is actually just the first slide. And here's the second slide of all of the academics and MPS networks that we have represented on the working group. So really, what are the detailed objectives of MPS Core? It's to create this shared space. So to have this forum that is intended to be, a very neutral forum, but it wants to kind of facilitate this engagement and this interaction among international collaborators. We do have very heavy representation from the US, of course, but we have many, many groups in Europe, in Asia, in South America, elsewhere in North America that are part of the global working group, and we're always looking for more members. And we're really trying to facilitate connections to help researchers troubleshoot, help them talk and learn from each other's experiences and share resources and share advice. To really try and support the application of these novel technologies. And as I already mentioned, we are really engaging with regulatory authorities from around the world to try and understand what are the regulatory needs and decision contexts, and how can we help move the field forward in terms of the assessment of these novel systems and characterizing their performance and their readiness criteria. And then, of course, because my organization and NC3Rs are really leaders in the alternative space, obviously the 3Rs opportunities to reduce, refine, or replace animal testing are fundamental to this effort as well. There have been countless publications that have come out over the last several years on COVID disease mechanisms, many of which are being done in microphysiological systems. And actually, one of the resources that we have on our website is a survey of the literature on MPS and COVID. It's current as of the end of last year. And at the end of the presentation, I'll show you a link to where you can access that. So there's absolutely no way that I could possibly highlight all of the publications that have come out of members of the working group. So I've just chosen to focus on 2 publications. One of them is actually an overview that myself and Anthony wrote that was a feature article in Drug Discovery Today that came out at the end of last year, that described the working group, but also gave the results of a survey that we did of our membership to understand what are the challenges that MPS developers and researchers are facing. And so these range from scientific challenges, such as access to well-characterized cell sources, incorporating immune components, standards and performance criteria, access to tissues, and then also practical challenges. So things like resources and access to BSL-3 facilities, et cetera. And so we've had a number of very productive discussions in the working group on how to address some of these challenges now that they've been identified. The other publication that I'd like to highlight is from some members of the working group who also happen to be some of the organizers of the MPS World Summit. Then we partnered with them and researchers from NIAID to highlight really the critical necessity of pivoting the work on COVID-19 mechanisms and understanding of the mechanisms to a reliance on using microphysiological systems. And here this was illustrated for the brain. So using the brain organoid models that have been developed at Johns Hopkins at the Center for Alternatives to Animal Testing, they were the first group to show that there is infection of COVID-19 in the brain. And this paper includes, again, a survey of the literature and the major findings, and then also all of the neurological symptoms that have been observed across many large cohorts of COVID patients and then also postulates sort of the mechanisms of neuro effects from COVID-19 infection, which include things like cytokine storm and systemic homeostasis imbalance and hypercoagulation, ischemia, leading to many of these outcomes that you see on the right-hand side that range from actual physiological to neurological and psychological outcomes. So a couple of ongoing projects that are actually funded under the MPS for COVID Research Working Group are work that's being done at NIAID. So historically, the Integrated Research Facility, or the IRF at NIAID, has been very much an animal shop. So they have a very strong non-human primate program and many other animal models. And in part through discussions with the MPS core working group, their leadership has really pivoted a little bit and expanded their program to now include MPS models. So they have brought some of the emulate systems in-house into their BSL-3 and BSL-4 facilities. And then the other project that's funded under MPS Core is to add some supplementary funding to the funding that NCATS has largely provided for development of the MPS DB, which now has a new name. And I will talk about both of these in just a little bit more detail. So the NIAID IRF program is also being done in partnership with the DoD. So Kyle and Tyler's group are providing lots of advice and training. And we are paying for the chips and helping with study design. And basically, this is looking at some novel therapeutics, including synthetically derived antibodies, testing those in MPS models. So starting with the lung chip model and comparing that with preclinical data and clinical data when it's available. And so they're using the golden hamster model, as I mentioned, and then they'll also be comparing with human data from clinical trials. So these are antibodies that are already approved and gone into clinical trials in Europe. And so it's a really, really nice opportunity to have, kind of a cross system comparison and stepping up in complexity from just your typical in vitro kind of vero cell based model to the MPS, to the animal model, to the humans, and seeing sort of what is the value of information that's provided by the MPS. And then, as I mentioned, the MPS database is now called the Biostatistics Analytic Platform, and hopefully you visited the booth or saw one of the excellent talks that referenced this. This is a really phenomenal resource for the MPS community. So this is, you have the opportunity here to upload your data. There's varying levels of security, so you can choose to have the information that you upload be completely confidential and secure only to yourself or to your group or to a group of external collaborators that you define, or eventually you can make it open and publicly accessible. So that's a really nice feature. And then one of the other really nice features is all of the different tools and workflows that they have as part of the system. So you can do things like reproducibility analyses, you can compare across similar experiments that are targeting the same biology. You can look at ADME, you can do PBPK modeling. So it's really fantastic. And if you haven't checked this out, I highly encourage you to do so. What we specifically supported is the development of a COVID-19 disease model portal. So this includes lots of information on disease biology, different experimental models, direct links to vendors for specific resources, that can facilitate COVID-19 research in MPS. And it's really designed to be this sort of central repository for COVID-19 experimental data, not just confined to that coming out of MPS models, but also from preclinical and clinical work to try and, again, facilitate those comparisons. And there is a private portal for MPS core members. So if you are a member of the working group, then you can upload your profile and upload information about your expertise and your platforms and your interests and availability of things like BSL 4 and 3 facilities. And then that is, again, designed to encourage interaction and connection and networking among the membership. Something else that we've just added some additional funding for is the development of an IPSC portal. So obviously IPSCs are a tremendous resource in this field, and there's really a need to try and find, so to have a repository or a resource where researchers can go and find information on IPSC models that are available and what is their maturation status, their mutation status, how to obtain them, different protocols, et cetera. And so at the moment, the information that's included in the IPSC portal is shown on the screen here. So it includes things like differentiation characteristics, patient profiles, what is the source of the cells, what are the protocols, and are there any established applications either in the healthy or the disease model spaces. And then we have a number of resources available online. So we had last year, as a satellite to the NCATS Tissue Chip Consortium meeting, we had a virtual workshop, and it was really a fantastic meeting. We had presentations from a number of our membership on the research that's ongoing in their groups, and that was recorded and is publicly available on our website. We also have webinars that are not recorded and are not publicly available that are intended as a forum to share unpublished or ongoing research information and ask questions and help troubleshoot among our membership. And so we have those every few months. Obviously, one of the resources is simply having those points of contact. So we have distribution lists. My group is always happy to provide study design support. We have many experts in validation of alternatives as part of the group, not only in our organization, but in organizations like CAT. And then, we also try and provide information on funding mechanisms wherever those are available, and have dialogue with regulatory decision makers to understand their decision frameworks and their context of use. And one really cool thing that we started doing at the end of last year was, in response to the emergence of variants of concern, we have a direct line to the WHO. So Simon Funnell is part of our group, and we call emergency meetings of the MPS core work group when there is a new variant of concern. And then we can identify people that have, models set up in their lab ready to go, and they can receive the variant from the WHO distribution centers and start testing them and start trying to understand, what are the potential evolutions of symptoms and presentations of the disease that are emerging with these different variants. And then I already mentioned the Biocystic resource, and they have just a really fantastic series of training videos that's available online. So I'll stop there. I think I'm right on time. And we have about 130 members at this stage, and we're always welcoming new members. So please let me know if you'd like to join. You can also just go to our website there and find out more information. See we have the downloadable Excel sheet that has a survey of the MPS and COVID literature, current as of the end of last year, and we're updating that every few months. And that's also where you can find things like the recordings of the workshop. So thank you very much.
Yes, thank you for your presentation. We have time for one or two questions. Are there any questions? So maybe I start. Do you also plan to open that platform to other diseases and viruses or something like that?
Yeah, it's a good question, right? Because in our sort of statement of purpose, we're called the MPS for COVID Research Working Group, but it was obvious that unfortunately, this is probably only the first of several pandemics that we can expect over the next several decades. And so part of the objective was to create this community so that the MPS models, we can learn from COVID, and then we can then apply them to other infectious diseases that are already a problem or will become a problem in the future. So that's definitely one of the objectives as well. Thanks.
So Nicole, have you thought about you've got iPSCs also potentially adult lung organoids?
So there's information on the MPS the Biocystic, I still haven't gotten used to the new name, platform about adult lung organoid models, but in terms of like building a repository of information, that's something else that we talked about, not just for lung, but for many different types of tissues. We haven't, created a dedicated repository for that like that we have with the iPSCs, but I will say that a lot of that information is readily available through the platform. Okay, thank you.
Yes. Thanks again. So I'd like to ask our next speaker to the stage. Our next speaker is Lance Taylor from the University of Pittsburgh, and he will give a talk entitled A Complete Platform for Preclinical Trials of Non-Alcoholic Fatty Liver Disease, including a patient-specific human biomimetic liver microphysiology system and a portal to the Biostatistics Inc. analytical platform. Yeah, stage is yours.
I want to.
Yeah, sure. Nope.
Thank you. Well, it's a pleasure to take part in the first MPS World Summit, and I'd like to congratulate the organizers for making it a very successful first time out. My title got changed because one of our students that you heard from earlier this morning, Summer Z, actually her poster got selected for a talk. So I'm now talking about toward precision medicine and patient digital twins for NAFLD with a human biomimetic liver, MPS, and the Biostatistics Analytics platform. So this is the list of what I'm going to tell you this morning. First, structured liver MPS are important in the continuum of fit-for-purpose experimental models. Non-alcoholic fatty liver disease is a complex disease with no approved drugs. And the quantitative systems pharmacology, or QSP, and MPS approach has characterized NAFLD complexity, predicted and tested drugs to halt NAFLD progression. And then patient-derived liver iPSCs for NAFLD clinical trials on chips are in development. And 5, there's great potential for creating patient digital twins for precision medicine together with the Biostatistics Analytics platform. Why biomimetic structured human liver MPS? Why are they important? Well, first, liver is more than a metabolic engine. With the hepatocytes, liver is the primary or important secondary organ in multiple types of diseases. And the liver involves multiple cell types that communicate with one another in their microdomains, which in the liver are the different zones, as well as communicating with other organs. So we've set out over the last number of years to recapitulate the liver acinus as the functional unit. And it's characterized by having a vascular component, the acinus, which is lined with specific liver sinusoidal endothelial cells and contains resident macrophages, the Kupffer cells. And then the hepatic chamber contains the hepatocytes and stellate cells, as well as cholangiocytes and a few others. But we have focused on the first 4 cell types in the building of the initial model. The other important characteristic, as you do create microdomains with the flow of blood through the acinus, where the oxygen tension is distinct at the front end and much lower as the blood enters the central vein. So there's different physiology and pathophysiology that occurs in the zones. So biomimetic structured liver MPS are critical for mechanism-based knowledge. So where are we going with MPS and liver-based diseases? I think it's fair to say we all believe that in time we'll be using stem cells so we can make these precision medicine tools. These cells can be obtained either directly from the organ, and we make organoids that way, or you can access through the skin cells or blood cells and generate iPSCs that then can be driven to specific, in this case, liver cells. They can then be combined and allowed to self-assemble to make organoid MPS, or physically layered and or bioprinted to make structured MPS, which is what I'm going to focus on today. Either one of these types of models within microfluidic devices can then be used for predicting disease progression and response to drugs. And very importantly, it's going to be a tool, many of us believe, for improving clinical trial designs before drugs ever see patients. You can study individual organs on chips by adding components to that chip that would arrive from other organs, or you can physically couple them to other organs, which we do in both a Nordis platform that we use the lamps on, as well as the vascularized lamps that we do with the microNit platform. This is a summary of our human liver MPS-based programs, and the focus for today, which is not all of them. We are testing therapeutics for metastatic melanoma in the liver. We have an SBIR with DILIsym Solutions, which is a computational modeling company where we're looking at biologics-induced liver injury. We are finishing the paperwork to start a bioprinting grant for the V-lamps for NAFLD. We have a type 2 diabetes drug discovery program that also obtained some funding to look at COVID. But today I'm going to focus on the NAFLD drug discovery program, which easily couples to our program in optimizing clinical trials for NAFLD, and then coupling that to what is now the Biocystic Analytics platform. NAFLD is a progressive disease, and it's a growing and global challenge, with 25% of the world population fitting somewhere in the continuum of NAFLD disease, characterized early on by an increase in fat droplets within hepatocytes or steatosis. A percentage of patients go on to exhibit non-alcoholic steatohepatitis, which is immune infiltration. Percentage of those patients go on to develop increased fibrosis that can end in cirrhosis, which is an end-stage disease. And some patients actually go on to hepatocellular carcinoma. One of the problems is this isn't a linear progression of disease. Different patients have different rates and can actually skip over different stages. So our initial goal in going after NAFLD using chips is to repurpose drugs and or drug combinations that can halt or potentially reverse the disease phenotypes and genotypes. And we decided to start with a focus on stage 3 fibrosis and earlier in order to develop therapeutics. To add insult to injury, NAFLD has recently been proposed as one of the components of the metabolic syndrome that includes obesity, lipidemia, diabetes and insulin resistance, and hypertension. And many in the field have suggested renaming NAFLD to metabolic dysfunction associated fatty liver disease. The prevalence of NAFLD is increasing worldwide. And in fact, if you look at the red, that's equal to 30% or more prevalence in the population. So there are two major problems. How do pharmaceutical companies optimize patient cohorts for NAFLD clinical trials? Because all of the trials so far have failed to meet endpoints. The second is, how will clinicians identify the right drug for the right patient? And obviously, this is a component of precision medicine and will be required to address the heterogeneity that's due to genetics, the environment, as well as the lifestyle of patients. Some patients are at increased risk of progression, for example, the PNPLA3 polymorphism, and that's something that Summers talked about earlier this morning. Patients have significant phenotypic differences due to the environment and lifestyle. And patient disease progression rates vary widely, and only a fraction of patients respond to specific treatments. hence the failures in clinical trials. Well, a key a parallel approach to traditional target-centric high-throughput screening and animal models is something we've been working on for a number of years, and that's the use of quantitative systems pharmacology, or QSP, and human microphysiology systems as a powerful combination for drug discovery and development. As a reminder, QSP is determining the mechanisms of disease progression and mechanisms of action of drugs on multi-scale systems through iterative and integrated computational and experimental methods. So you can have an experimental platform, generate data, start building the computational model, make predictions from the growing model, test it back into the model, and do this iteratively. And this can increase and optimize the development of therapeutic strategies. Well, I don't have to tell this crowd about human MPS, but the key is that if you combine human MPS for any disease, we're particularly interested in the liver, with QSP, it is a powerful approach for drug discovery development, clinical trials on chips, and the path to patient digital twins. So this is a simplified pipeline of what we do applying QSP and MPS. We always, we always start with patient material. In this case, in the study I'm describing, 182 patients that underwent bariatric surgery, and samples of their liver were taken for doing RNA-seq. That data can be used to infer pathways of disease progression. That data can then also be used to predict drugs and targets by machine learning and systems biology approaches. And then you can generate patient-derived primary cells and iPSCs and test the predicted drugs and candidates in human biomimetic liver MPS. And again, you can start building a computational model in this iterative process. You can improve things before you even start doing specific drug testing. In addition to the experimental data, we also use validated published knowledge. This combination is then uploaded into the Biostatistics Analytics platform. And because QSP is a systems biology approach, you expect emergent properties, which can include novel targets, biomarkers, repurposed drugs and combinations, the starting point for novel therapeutics and therapeutic strategies. All of this knowledge then can be fed back to patient populations. So we have a human NAFLD liver MPS model that can be progressed from normal. And then we've predicted drugs and drug combinations to halt the disease progression. And we've started with primary cells as a reference. And now we're building the patient iPSC preclinical trial platform to make it personalized. These two models that we use, one is the LAMPS, or the liver acinus MPS, based on the Nordis chip, and the vascularized liver MPS based on the MicroNit platform. There's pros and cons to both, and we use both of these platforms. The LAMPS model is very simple to make. It's very reproducible. You can control the oxygen, for example, and hold it at either a zone 1 or a zone 3 level. The VLAMPS is more complicated, more sophisticated. It has a vascular channel as well as a hepatic chamber. So in the vascular channel, which is lined with the liver sinusoidal endothelial cells, we can actually pass drugs and cells, including immune cells, that can then enter the hepatic chamber when activated. The first studies have been focused on primary cells, and as Summer mentioned this morning, we are developing the iPSCs for the others. A key element of this is we've built a panel of phenotypic and functional and genomic metrics to characterize the chips. I won't make you sit through me reading this list, but there's multiple components of the secretome. We have fluorescence-based protein biosensors in individual cells, And we have additional imaging metrics as well as endpoint measurements, including doing RNA-seq on the model itself. All of this data is uploaded into the Biostatistics Analytics platform, and I won't dwell on this because it's been talked about multiple times at the meeting, but we have found that it's critical because when you vary the cell line, the media, the matrix, any of these variables that we have in MPS models, and 6 months later, a year later, you want to go back and see how things changed, you can do all of that analysis within the database, as well as using it as a starting point for the computational modeling. So we've quantified the progression of NAFLD using these models. This particular data is based on the LAMPS model. Summer, for those of you who were here earlier, described the media where we can progress from the normal liver with kind of physiologically relevant glucose. We can drive it into early metabolic syndrome by the addition of more insulin and free fatty acids, and can drive it into NASH by further addition of immune activation factors. This is a summary of some of the main metrics, and I'm not going to repeat all of the data that Summer showed, but we get steatosis increasing over the disease progression, oxidative stress, inflammatory activation. We get glucose, insulin resistance. Sorry, there was a little bit of COVID fog for a moment. Here are some specific measurements. These were all done in zone 3, what we've reported here, because these major characteristics of fibrosis, steatosis, and immune activation are actually maximal in zone 3 compared to zone 1. This work was led by Mark Meadle in the group. So this is kind of the QSP part of this. And we've done computational analysis of NAFLD patient RNA-seq data. And we've been able to define the molecular disease states and then use that to predict drugs for experimental testing within the liver MPS. This is obviously a complex image, and you can't see it well in the back. But what we did was to take the 182 patients, which are shown in this heat map in the columns, and then pathways within the KEGG space are the rows. And then we applied analytics to unbiasedly correlate all of these components. And then they sorted just using the data, again, in an unbiased way. And that produced from this pathway characterization 3 components: a quote normal steatotic group, a principally lobular inflammation group, and a principally fibrosis group. The metrics here are blue shows a lower than normal pathway expression. Red is above. And with this unbiased analysis, we can now use this data and build a pipeline of computational and further systems biology tools to get to the point of predicting drugs. And I won't take you through all of those details, but we use 2 approaches to prioritize the drugs that we've determined out of Drug Bank. And we now have 40 that are highly prioritized that we've started investigating. This just shows the proof of concept from doing this. This is just 2 of the drugs out of the top 40. And alone, they only partially change the steatosis, lobular inflammation, and fibrosis. In combination, pioglitazone in this case, with verinostat, a HDAC inhibitor, actually suppressed all three, and under concentrations of the three drugs that have been used in other applications. So now we have started testing each one of the top 40 drugs and their combinations with the goal of finding the top combinations that we will take to the clinic, and also use the knowledge gained here, the knowledge gained here to do new drug development. Summer also talked about the genetic side of susceptibility, where PNPLA3 has a high effect on susceptibility for NAFLD. There are others, some that have a small effect. We decided to focus on PNPLA3. It codes for a protein known to have hydrolase activity towards triglycerides and retinol esters. Importantly, and this is a philosophy that I think is important, before doing iPSC-derived studies in CHIPs, we think that you need to define the adult reference point using primary cells. And so we've started by identifying in primary hepatocytes that were genotyped, those quote, low risk and the high risk PNPLA3 to do experiments on. So we've essentially set up chips that are in normal fasting media, early metabolic syndrome media, and then driven to the later stages of the disease. And in fact, just looking at the difference being the presence of the high risk component, we're able to see that intracellular lipid is increased when you compare the low risk in blue to the high risk in red, where you're looking at, in this case, total lipid tox that represents the amount of lipid. And you can also do the same thing with the secreted triglycerides. So this is a major component of the genetic impact. So we have a large NAFLD patient population that we follow in the UPMC Fatty Liver Obesity Wellness Clinic that supports the NAFLD clinical trial on patient CHIPS program. We start by taking blood samples from these patients where we have all of the clinical data on each one of them. Using their blood cells, we generate iPSCs, and then we take those and drive those to the individual cell types that we want, and they can be put into the chips to build that individual on a chip. So far, we've have 246 subjects with NAFLD-NASH that have been enrolled in the program, and they've been genotyped. And this is just through April of this year. And again, we're identifying all of the variants, but experimentally, we're focusing on the PNPLA variants. We have the high risk variant and the low risk variant, and that's what we're focused on now with the early work on the iPSCs. Since we just talked about the Biostatistics analytics platform, I won't dwell on it other than to say we can't do most of what we're doing and are planning on doing without a one platform that has a complete data pipeline from discovery to clinical trials all the way to building patient digital twins, from very simple things to more sophisticated things. So we think people, whether it's this platform or something else that will do something comparable, it's time that we start managing all the data so we can know what we've done from week to week, month to month, and year to year. An exciting new venture is creating patient digital twins. And we're doing that starting with the NAFLD Flow Clinic patients and the liver MPS that we're building. And with patient digital twins, you integrate clinical data from each individual patient. Obviously, the goal is to have a chip for each individual patient or for multiple organs that are coupled or whole body. It would be more sophisticated chips over time. That data taken together can computationally be used to create the patient digital twin for precision medicine. We think this is the future. If we think the mountain to climb to optimize iPSCs is a challenge, this is a massive challenge. But just like there were naysayers a decade ago about the potential value of MPS, I think I've heard some of that complaining since we've started talking about this. I would say wait 10 years, and I think we'll see that it's having an impact. This is just the same list that I started off with. This is what I told you. I have a disclosure to make since I'm involved in biocystic. And this is the acknowledgements, great team within the Drug Discovery Institute, and we have our appointments in computational and systems biology, and then great collaborators across the university, both engineers, clinicians, and other kinds of investigators. This is our funding, and I have to advertise that we have positions available in the Institute for research assistant professor and a couple of postdocs. Thank you.
Any questions for Lance?
Yeah, Martin Golubieski from Germany, from HITS. Thank you for this very interesting talk. And I was wondering, you showed in the beginning the value of coupling the computational modeling approaches with the MPS technology. Is that also covered, or is there a plan to be covered by the analytics platform that you develop? Because I mean, you can develop a system that integrates the data that is then feeding the models, or is that approach more an integral approach that also spans over the modeling?
Great question. And our philosophy is that if you're using an MPS in any way, you want to capture all of the data within the database, which includes the computational and systems biology analyses. So yes, it's being incorporated.
Yeah, I have one question. For the QSP work that you're doing, is it just one component? Because obviously NASH is a multi-organ disease. So how are you taking to affect the different components?
So,
and one additional point is then, do you think you should have a multi-organ system? As a number of talks over the last 2 days, 3 days.
Two good questions. What was the first one? Let me answer the second one first. We're already coupling organs for our type 2 diabetes study, which also includes MAFLD. We have pancreatic islets, the liver, and we're about to add white adipose tissue. So we see subsets of organs being the most useful in the near term. Whole body on a chip, it will come at the right time, and we're seeing some great progress there. So yes, what was, now I have forgotten.
Yeah, so the QSP model, was it just a one component, and how you're factoring in?
Oh, so the data was from patients. So all the analytics were on RNA-seq from patients. So that's everything. But we've also, and I didn't take the time to show it today, we've done RNA-seq on the chips that have remained, quote, normal fasting, early metabolic syndrome, and late metabolic syndrome. And we get about a 70% overlap of the pathways that we've identified between the patients and the chips. And that's not even involving all of the cells yet. So it's already pretty good representation of the liver.
Fantastic. Thank you again. As Thomas is coming up, there was just a reminder after the end of this session that lunch will be served outside. So yeah, obviously Thomas doesn't need any introduction. So Thomas Hartung is going to be giving a talk on from good cell and tissue culture practice to good in vitro reporting standards.
So, hello. You have witnessed over the last 3 days that we are doing a lot to promote microphysiological systems. I'm going to talk about a parallel initiative in which we, over the last 8 years, involved far more than 100 people, which is good cell culture practice. It's all about quality of microphysiological systems and cell culture in general. I have to mention some conflicts of interest. Here, especially relevant are my involvements in the microphysiological systems area. So please take everything I'm saying with a grain of salt. You have seen the revolution. We are part of it, which is taking place through 2 workshops, which were organized by Uwe Marx and Adrian Roth, both of them in Berlin. We have been trying to find consensus and promote the acceptance of these methodologies. We were very happy that the 2nd workshop led to this science perspective paper more recently, but I don't need to preach to you about MPS and their aspects. As I said, it is all about quality. And I would like to talk about mainly the first 2 steps, which is good cell culture practice, which is about how to do cell culture and in specific microphysiological systems. I will talk about the starting project of in vitro reporting standards. And ultimately, this is all leading to something which is somehow qualifying and validating these systems for actual use. Irreproducibility is a problem. And we don't get to good systems if we are putting bad cells into whatever fancy bioreactor or whatever we're having and call them MPS. And there's a lot of aspects to it. I will be very brief on this. We have to assume that still about 25% of the cell lines in academic use are not what they're supposed to be. It is consistent over time. The last study I know of is from 2015, which found pretty much 25% misidentified. The second point, we still live with mycoplasma infections. Again, something between 15 and 25% is likely infected. This is something which is easy to control, but it's not controlled everywhere and is impacting dramatically on our outcomes. So there's genetic instability. This problem is a bit less with iPSC, but not gone. We have genetic instability, and we have to be careful how long we are propagating our cells, something we have to tackle. This was for me, eye-opening study where we found that 2 vials of cells, which came from the same batch from ATCC, probably one of the best cell culture banks in the world, had dramatic differences in their behavior. And when we took materials, again, from the same batch, without even sawing it, we found dramatic differences in 6 chromosomes within these same vials from the same batch. So there's still some quality control to be done, how we produce our vials. This is another example showing you the problem of these cell lines, the MCF7 cells, again, like in the previous study, which have been given rise to more than 30,000 scientific papers. And in this paper, analysis clearly shows they're not even typical for breast cancer. It is comparing the MCF7 cells with actual materials coming from patients. So it is very, very problematic what we're often using. And this is not only the MCF7, this is the HeLa genome. In the end, it shows it is a Franken cell. It has some chromosomes completely missing. There is reshuffling of essentially everything. It is really dramatic. The MCF7 cells had lost 10% of their genome. About 40% was duplicated up to 40 times. Yeah, so it was really an enormous sack of genetic materials only. Here, another example. These are commercial cell lines. You can pay a lot for this. Supposedly, the only difference is that one gene was inserted, a P450 enzyme here, and you can see they are very different, complete chromosomes, again, not identifiable in one. This is something where you would interpret your findings by saying, yes, this is with and this is without P450, but it is much more which has changed. So altogether, irreducibility is the problem. Cell culture artifacts. Our use of antibiotics makes our life easy, especially with complex systems like an MPS, but they are producing a lot of effects. This is just the gene expression which is induced by the use of standard antibiotics. We have a situation where we culture normally our cells by like pan-fried eggs, sunny side up. That's how our cell culture looks like. And we are jointly trying to move this to three-dimensional situations, which also give us then finally cell densities, which are tissue-like. So not all our factors being diluted, percentages of the cell-cell contacts which normal tissue has. So there's an enormous advantage, and MPS are for this reason alone a very important step forward. But there's more artifacts in standard cell culture. The sudden change of the environment by changing our media, suddenly replenishing all of the nutrient and oxygen which has been used and removing the waste. This is a situation where a cell has to stay adaptable, and being adaptable is the opposite of differentiation. It is a very important step that we are introducing homeostatic conditions. And this does not hold for all research. Our own organoids, we are changing the media every second day. It is not by moving to an organ that would have overcome this problem already. It is really important that we consider moving more to homeostatic conditions. And there's many more to mention. We have obviously a problem with bad cells in a device. But the use of primary cells, which are increasingly becoming commercially available, the use of stem cells is helping us at least to get better human cells available. But is then the lack of organ interactions? You know, we have seen it, body on chip type of solutions. That's what is really the future of allowing us not only to have the tissue physiology and cell physiology reflected, but also some of the actual physiology of the body. I believe that the 21st century science requires 21st century cell culture. And this is where all of these MPS come in. This is what it's all about, to really improve the quality. Already in 1996, I started an initiative which is called Good Cell Culture Practice. At the time, organizing a symposium for the German Cell and Tissue Culture Society. This led 1999 to a call on the scientific community to create GCCP, and I had the privilege to lead the task force for ECVAM before I later became the head of ECVAM. And in 2005, we published for the first time a document through the Commission on good cell culture practice, which has been reasonably cited since then. It is talking about what you should know about your model. It's talking about the assurance and quality of all materials and methods you are applying. It is talking about documentation. So what is the minimum documentation necessary if you describe such systems? It talks about hazards. It talks about the laws and regulation and ethical principles. And it talks what should be the education which is advisable to do high quality cell culture work. So these are the 6 principles of GCCP. But 2005 was before even iPSC were described. So it is very meager on stem cell work. It is not at all mentioning anything which is bioengineered microphysiological systems. So in 2015, we held 2 workshops on both sides of the Atlantic. These are the organizations which teamed up at the time. And these 2 workshops produced workshop reports and were the starting point for revision of Good Cell Culture Practice 2.0. Good cell culture, these are the respective reports. Good cell culture practice is not alternative to good laboratory practice. It is to some extent overlapping, but it is meant for those people who are cannot apply good laboratory practice, because good laboratory practice, for example, requires that every person is educated before running the experiment. In an academic lab, this is not the case. We are learning or training our students while they're doing things which we still want to publish. So it's more a minimum standard we are trying to achieve here. Good laboratory practice would require that each experiment has one dedicated workplace. Our hoods are used for tons of projects. We cannot afford a standard of GLP, but we can work in a spirit towards GLP, and this is what good cell culture practice wants to be. It wants to give guidance to journals and funding bodies, and this is what we try to achieve. And finally, in January, we published the new guidance document. The draft had been published exactly a year earlier, and we received 350 comments from more than 100 stakeholders, which were discussed and worked into this by the drafting group, the steering group, which you can see here. So I invite you to look for this document, make use of it. We are entering now a dissemination phase. We are trying to get this to the respective stakeholders. We hope that funding bodies will consider requesting GCCP standards for execution of work, because we feel that is something to promote our work. Interestingly, a parallel development with some overlap in personnel led the OECD to adapt their good laboratory practice guidelines to in vitro. Because good laboratory practice had been developed originally around the animal experiments which were done in OECD. And they call it good in vitro method practices. And interestingly, it is copy pasting a lot from GCCP 1.0, and it's in full annexing the first of the workshop reports we did, the only thing which was published at the time of their publication in 2017. So this is a very important development, and we are continuing to crosstalk with them about adaptation. The second element, which I think of our work, which is not known to most of you, is we are interested in bringing evidence-based medicine to toxicology. We call this evidence-based toxicology. I started this also at the time of the commission. We held the first conference on evidence-based toxicology in 2007. Since 2009, I have the privilege of a chair for evidence-based toxicology at Johns Hopkins. And since 2011, we are hosting the Secretariat for the Evidence-Based Toxicology Collaboration, an initiative which involves the FDA, the EPA, the European Food Safety Authority, and others in the Board of Trustees, and many, many activities. More than 900 people have worked on the evidence-based toxicology collaboration. I'm mentioning this because there's again a tremendous overlap in quality assurance, the desire to come to good data, to good, to high quality things, and come from good data to good conclusions. This was a paper, one of the outputs of this collaboration, which was guidance on assessing the methodological and reporting quality of toxicology relevant studies. And it shows what are the frameworks to assess whether in silico, in vitro, in animal data, human data, epidemiological data are of high quality. So I invite you, because this is something which is critical and identified as one of the needs. We don't really have enough for evaluating in vitro studies. The ToxR tool, which also came out of the evidence-based toxicology movement, was the only one really suitable for qualifying papers. So an ongoing project is the IV-CAT, the in vitro critical appraisal tool. This is again a tool which wants to judge on the basis of a publication, is this good data? Is this a system which is suitable for working? So this is an ongoing process out of the EBTC, which I see very much in parallel to these activities. The second point I would like to make, and this is a project which has started in 2019, where we started with this white paper on good in vitro reporting standards, because it's not only to do the work well, but also to communicate it well to others. This goes beyond documentation, which is internal to the lab, which GCCP covers. It is about what we are doing in order to quality assure our publications. And I believe that a lot of the reproducibility issues we have in science are actually miscommunication issues. We are not talking well enough to each other on how to reproduce an experiment because we still limit ourselves in the way we are describing the models, and we do not systematically apply reporting standards. This is an example where we developed a template for cell-based toxicological test methods. Similar templates could be developed for others. I think it is really important to set standards here and help a journal to have checklists of, is this system adequately described? Many papers are useless for the scientific community just because they miss important information on what has been done, what has been blinded, which passage of cells has been used and others, the metadata around our experiments. This is where we need to improve and to build on. And you see, from my point of view, this is something where we really have a piece where simple cell models should be quality controlled by GCCP. The microphysiological systems now covered in the 2.0 version. The in vitro standards from OECD come in if this is for regulatory type of testing, but there's reporting standards now to be produced. It is about the quality storing of the publications we do from this. And in the end, this ensures that an information need is actually satisfied. And with this, I would like to end. I cite always or almost always in my presentations John Maynard Keynes, an economist, because he said something very wise, which is the difficulty lies not in the new ideas, but in escaping from the old ones. I believe that for our escape from the old ideas, from the systems of the past, quality assurance will be critical. And I hope that GCCP and GiveRest will be important contributions to help the community. Thanks a lot.
Yes, thank you for that talk. Are there comments, questions from the audience? Yes.
Yes, Thomas, thanks for addressing this important topic. And I was wondering, are there any specific plans or there specific approaches for training editors and chiefs of the major journals or program directors of the funding agencies so they can train the peer reviewers to appropriately comment on these needed issues?
Yeah. We actually are postponing, for evident reasons, our planned workshop with journal editors already several times. We had such a journal editor workshop for evidence-based toxicology, so we have some experiences with this, and we see that this is really helpful to get a core of them together and also to understand better how we can serve their needs. And we want to do exactly this. At the moment, I hope to have this in fall of this year, because it is about journals agreeing on implementing this and setting a signal. But we want to do better than, let's say, in the field of animal experimentation. The NC3Rs has done very important work with the ARRIVE guidelines, but nobody is using them. 4,000 something journals have accepted them, but they're not implementing them, as we have unfortunately seen. So we hope that by combining journals and funding bodies, I hope that we can get further. You get as scientists only when it's about money, yeah? and that's why I think it is not only the journal's job to implement these things. Yeah.
Maybe a question from my side. You mentioned the cross-contamination issue, which is like 20 years old, but you did not mention the substances we use with batch-to-batch variations. Maybe you want to comment on that.
I mean, the guidance is talking about all of this. Obviously, it is each and every component in our work is problematic. Yeah. I mean, no chemical is clean already. There's always contaminants inside. Many people just believe that the amount in our Sigma vials is actually, which is printed on, is identical, and they just put some solvent into the thing. And very often this is a dramatic mistake already made in producing our standards. The batch variation extends then obviously to all plastic materials. We only address this when we have a problem, when something suddenly does not work properly. But I'm not aware of many people who are consistently doing batch control here. And the batch control of cells, I showed you one example that it can even be the same batch of cells. The explanation for this is probably that these tumor cell lines have different subpopulations. They have over time, for example, the MCF7 cells seem to have one subclone, which is about 20 to 30% of the MCF7 cells we had, which has lost entire chromosome 7. But it's only about 20 to 30% of the cells. And probably they have different sedimentation behavior that when filling these vials, you do not get the same mix all the time. And this is the explanation why there is within batch variation for this. This is at least my only explanation I have found so far.
Thank you.
I have rather a comment than a question. So I have been attending a workshop a couple of years ago from EMBO, the European Molecular Biology Organization, and it was on standardized reporting on cell lines. And I think that might be helpful for your dissemination plan, because I think EMBO also as a publisher might be quite interested in
no, this is, we need to be fair. There's a lot of different areas which are trying to move towards good practices and guidances. The white paper which I showed on good in vitro reporting standards cites, I think, a dozen attempts on which we can build. It is more really about trying to find a consensus and getting many of these elements together. I believe there's still need for something which is a point of reference for us, and which is also going beyond the scientific community by addressing in a dissemination stage funding bodies and journals, because that's the way of getting it in. Otherwise, it is a piece of paper which is not worth the paper it is printed on if it's not being used.
Yeah. One additional comment, Thomas, you mentioned the RIVE guidelines, that there's the RIVER guidelines. So NC3Rs, I'm part of their, they've got an ongoing working group to come up with that. So I'd be happy to link you with that initiative as well.
Yeah, I mean, I know that they're very much aware of our initiative. I was a bit surprised that we were not invited to join, but this is I mean, the good thing is if a lot of brains are picked and mobilized, and in the end are contributing to the overall need of these type of standards. Yeah.
So just to address the comment about funding agencies. So the NIH peer review process over the last several years has already incorporated requirements for authentication and best practices before funding applications. Yeah. So it's making its way.
No, this is, I mean, I really applaud a lot what the NIH has been doing for increasing reproducibility of research, making things more relevant. But I think something comprehensive such as GCCP, and still to an extent that it is still readable, or you can refer to it, or can make it a basis for training on quality assurance for practitioners. I think that's something which is needed and has to go beyond select things. I'm not aware of requirements for mycoplasma testing, for example, but authentication is really an important issue. I'm very happy that this was tackled. Thanks for the comment.
Thank you. I think we have to move forward. Thank you again, Thomas. So our next speaker is Daniela, and I would like to ask you to say your second name by yourself, because I'm pretty sure I will make a mistake there. But I will just read the title of your talk, which is industry perspective on the challenges and opportunities in developing, selecting, and applying advanced in vitro models to drug development in the context of data-driven decision-making. So you are from Roche. Daniela, the stage is yours.
Thank you. It's actually Ortiz Frangiuti. So, so you know where's the fast to click? Is it this one?
It's this one exactly.
This one?
No. So you go forward.
Super. So, hello everyone. I'm here representing my group at Roche. We are working in investigative and immunosafety in the preclinical area. And I'm here to talk to you about how we're using basically the systems that we've been exploring and seeing in this conference, and how do we use them to do decision making, basically. So what are our challenges and where we're going? And I'm really excited because all that we've been talking in this week is steps that we're all together doing, climbing a ladder to change the status quo. And this has happened before. So I want to tell you a quick story about how you change the status quo. So a long time ago, people thought the Earth was the center of the universe. But some of the data wasn't really fitting. So the planets were doing these weird motions, and they try to like make sense of it and make models and theories about it. And it didn't really fit. And so some people came with innovative ideas. And one of them was Nicholas Copernicus, who said, like, how about we change the focus? Let's put the center in the center, the sun. And it's just really elegant, you know? But of course, nobody believed it because there was data missing. So it took actually centuries to collect this data. And one of the key players was Tycho Brahe, who collected through the whole of his life with very advanced instruments for the time, the movements of the planets. Now, this data could have died with him because he wasn't really good at sharing. But luckily, one of his students or collaborators in that time, Kepler, knew where to find this data, and he could then take that data after Tycho Brahe died and come up with new ways of modeling the data to prove that innovative idea that Nicholas Copernicus had before. And then after him, when he said, okay, actually the theory works if you just make it like an ellipse, and if you this change in mindset, and then came Galileo and Newton, and the rest is history. So actually, it's very important, that step in the middle of when we collect that data, because that is what allows then the new ideas to become accepted. So where are we today in our climbing of this stair? We need to do better, basically. So our translation rate is still quite poor. There's a very low likelihood of a drug being approved in phase 1 to actually go through the whole process and get final approval and be there really for patients. So we clearly have to do better. And we already have some ideas. And it's a matter of saying, okay, what else do we need to make our case and to really come up with these better models to make those predictions? So I think here is a community that we are aware of these challenges. So what is making it challenging? One of the topics is obviously humans are not animals, and that's why we are all here working on it to make more human relevant models, aside of also the ethical issues of reducing and even replacing animal models. So this is not new to this community, and it's one of the challenges that we are trying to overcome. What are the other things that are making our life challenging? Is biological diversity. So why do we respond different to treatment? And we've heard a lot in the morning and throughout the whole week, personalized medicine, personalized making predictions. And still a lot of our programs rely on cohorts of patients where we have non-responders, we have people who respond okay, and people who respond really well. And what we need to start thinking is like, what do we learn out of these exercises? And what do we learn from what happens in these rounds of testing so that we can bring those learnings back and start with forward translation? So this is another one of our challenges. Now, it is also very difficult, especially for us in safety, because we have a growing arsenal of therapeutic modalities. So, and this is just one example. There's many therapeutic modalities, but for bispecific antibodies, and just to make the point, you can see that there's like a plethora of molecules, and they all have different biology that we need to replicate in an in vitro system to be able to actually achieve better predictions and be more close to what these antibodies do in vivo or in patients, basically. I was very happy to hear already that we're already aware of reproducibility and trying to change that. So we see that also even, for example, this is one of my favorite initiatives, a reproducibility project for cancer biology, where they assessed like, okay, from all these top tier papers in cancer research, which ones can we reproduce? And it's been a huge effort to do this. And it's quite disappointing, actually. So the reproducibility is very low. But what I like about this consortia is that they're not just focusing on pointing out the pain point, but they also are picking up the key learnings. So they are pulling what makes these papers that you see in blue here, what makes those papers reproducible. And it actually boils down to reporting standards, good documentation, very defined and detailed recording of experiments, materials, data, code, how things were analyzed, and then that this data is registered, that there is transparency, that you can go actually to the people who did it and have that access to that. And then also that there are these incentives for replication, which is, I'm very glad that this discussion was already happening. So these are also, it's not just pointing out the problem, but also like, let's look at the solutions and go through them. And especially for us, this is incredibly important because the learnings on translational power arrive years after clinical trials start. So if you see here, we have like a couple of drugs that have been already approved, and you can see in the colors are like when the clinical trial starts, so phase 1, until when it's approved. And you can see that it takes a long time. And on the bottom, I highlighted that. So it's really decades. And the learning, so you can see the publications here, they come even after that. And still, we are in the gray zone there in the beginning where we have to make decisions today. And this is where we are getting stuck. So nevertheless, we do put molecules in the clinic, and actually we do it without animal studies. This is a package where we enabled an entry into human in vitro only safety regulatory package. And this was actually a golden opportunity for us because it was a molecule, it's like the TCR-like molecule that binds to a peptide instead of a protein. And this molecule is working in the context of the MHC complex. So it's very human specific. And because of this, animal safety testing was just not possible because of the mismatches in the human animal MHC and also because of the peptidome differences. So here we have a huge challenge and at the same time, a huge opportunity to show that we can actually put a molecule in the clinic without animal models. So what you see here is, what we did is take all the critical organs and cell lines, and not cell lines, sorry, cell types for that we would need to screen to really come to like a comprehensive package to show we're doing the best that is in our hands to give an in vitro only package to the regulatory authorities. And what is interesting is that this is a huge endeavor. So we're including 2D cultures, organoids, spheroids, macrophysiological system, and we are working internally with our labs and our own internal development groups. At the same time, we also work with collaborators. We also work with CROs, so external providers. And this is a mammoth task to put together these packages. And obviously related to that, and because we have such a plethora of systems involved, we have very variable readouts. So we do cytokines, we do PCR, we do functional low dimensional readouts, we do microscopy, flow cytometry, omics. And you can imagine that if for each one of these, we're creating all this data, it gets big, basically. So, but let me go back to, okay, how do we even like start to choose which systems to use for this kind of packages? And we do this in very tight combination with the project needs, so what the molecule needs. So we look at physiology, that's obvious. We see like, okay, do we have like a good level of physiological relevance in the system? That's one thing, but we also look at target expression. So which model is basically reflecting the human physiological situation in the different parts of the organ. And based on that, we can compare among the systems that we have and make the best choice of the system that will help us answer those questions for each one of the organs. And then obviously there's also technical questions. So regarding like accessibility of the tissue and so on. I will not go into it, but it's not just that part, but we also need to basically put the thing together in physically the plane. So, so the other thing that we do is we have to do the model characterization embedded in the safety assessment. So basically we cannot wait 4 or 5 years until the model is characterized because we have to put the package together today. So what we do is we embed this characterization within the safety assessment. And actually this is like crucial because a lot of our data then, if we see noise, if we see something that it doesn't really make sense, then we help ourselves with that characterization to define whether this is something because we are working with the heterologous co-cultures or whether this is something related to the PBMCs that we used. So we had some fantastic posters in the session from my colleagues that are really pushing all of these initiatives. If you didn't have a chance to see the posters, I invite you to revise their abstracts and contact them directly so they can give you more information on this. And we do things like ranking our PBMCs, HLA matching, the heterologous co-cultures, and also revise the physiological relevance of the models that we're using. So no, it's probably not perfect. We are heavily working on it, but we need to do it also in parallel with the actual drug development. And actually by doing that is how we learn what we're still missing to know about the models. So you can imagine then we go ahead and we deploy all these models and we put our molecules in. And for each model, for each replicate, for each time point, for each readout, we generate a lot of data. So this is an example only for like kidney, renal proximal tubule epithelial cells. And you can see that we have all these different readouts. And this is just for one time point. So this is not yet aggregated, integrated, interpreted data, okay? We do this for all of that battery. And so the challenge then is just put in numbers. We take like all these different variables. We end up with literally millions of conditions. So we have multi-model, multi-readout, complex integration, complex interpretation even. And somehow we need to make sense of this so that we can say, take a decision, basically. And that's not all. We, on top of that, have the whole part of the metadata, which is not included in this number, about like which individuals we considered in the platform, the samples, the experiments, biospecimens. So all of these things are variables, actually, and somehow we need to make sense of it, and it's extremely complicated. And now, with this level of complication, and what I showed you before about the cycle of the data, which is sometimes even in decades terms, try to think about finding and understanding this data in 10 years' time. Think about your own experiments, like, could you make sense of it in 10 years' time? I would find it a challenge. So what we are resorting to be able to make sense of it in 10 years' time is to implement good scientific data management. And this relies on prospective fair data, so that the data that we collect is fit for purpose, already thinking of those 10 years from day one. And that's how you make then this data findable, accessible, interoperable, and most importantly, reusable, so that then we can start going in these areas of knowledge, insights, and for us, very importantly, decision-making. And in practical terms, what it means is we need to record what we're doing. So from day one, from the moment we're planning, from when we're sourcing our material, when we start implementing our experiments and generating the data, we need to know what we did and have identifiers so that we can trace back our results to specific individuals or specific variables that then we can say, okay, here is what is giving us predictability or not. And the other part of the coin is, once we generate all this massive amount of information, we need to synthesize it. So we need to process it, integrate it, and do modeling to then be able to get to this decision making. So go, no go, maybe we develop a de-risking strategy, maybe we prepare a regulatory filing. And really it's about ensuring the connectivity of those readouts so that when the molecules get to the clinic and we start producing also clinical data, that these two data sets match and can be connected. And that is the only way we can then start to increase confidence in our translation power. And we have internally also a huge initiative to verify, so to make this data fair in clinical trials, so that this is enhanced data and insight sharing program. And really the key is that in the center is data management, data integration, and having always a single point of truth when we can refer to where the data was generated. And this is what then allows this forward translation. And also when we go to the clinical setting to really bring that back and achieve that reverse translation. And we have guidelines and practices already implemented for clinical data. So we have our FAIR data playbook. We have guidelines on internal reuse of patient-level clinical trial data. We also have a framework for responsible access to internal data. We shouldn't forget that we are dealing with patient data, which is very sensitive data, and we need to act with it responsibly and have also the proper controls on the access to that data. And nevertheless, we need to be very careful with our data through all this process of planning, acquisition, processing, and release. And so what we're trying to do is, even if there's parts of our work where we are working in parallel within clinical data, that we both escalate this ladder together. So we have a layer of infrastructure. We have APIs, so programmatic access is incredibly important because this kind of complex integration can only be done programmatically. And then we have our scientific data assets, which are specific to the domains where we're working, whether it be clinical, whether it be animal studies, in vitro data. That's very specific to the domain. But then we need to add on top of it a layer of harmonized data that makes the bridge to other domains. And on top of that, we can create an integrated data sets, data marts, that we can then, on top of it, do analytics and have visualization tools that help us understand what is happening. And that's how we get then the transitional power. So it might sound even a little bit dry, the whole data, take care of our data thing. But actually, if we really want to achieve translational power, we need to climb this ladder together. So with that, I would like to thank all the fantastic colleagues. So this doesn't happen if we don't have the support and we don't have a lot of people and a huge community of colleagues really implementing and putting this data together and doing this amazing work, whether it be on the bench, on the analytics, on the IT side. It's really, we're all in this boat together. And I just want to thank all of my colleagues that we climbed this ladder together. Thank you. Questions?
Thanks, Daniela. Really phenomenal talk. I'm curious about the controlled terminologies and sort of the knowledge organization systems that, how are you choosing those? And how are you, which ones have you sort of zeroed in on to make those linkages between the different data streams?
Thank you. Fantastic question. So we have internally a specialized group, which is the Roche Terminology Service. And what they work is basically they're specialists in ontologies. So they actually have a database of ontologies, and we internalize also ontologies from other communities. So whether it be like, I don't know, MedChem or protein ontologies. So they keep track of all the different terminologies that are like in the outside world. And internally, what we do is we work on synonyms. So if there might even be like two terminology databases or standards that are overlapping. So then they take care of having all the synonyms for each one of them. And then we get internally even identifiers for the terms that we use. And basically, it's a federated system. So we don't collect all the data in one database. The database is fit for purpose for every step of the development. And these other databases that control the language have identifiers where they connect then the terminology that is used in each one of the databases. Because absolutely, this is key. So we need to be using the same terms that the clinical people are using, that the chemists are using in the very early development. And so we even have a dedicated team to do that. Thank you.
Thank you. In the absence of validation of many of the microphysiological systems, how do you see that this can be overcome to facilitate moving with microphysiological systems as alternative tests acceptable by regulatory bodies to move to the clinic?
Great question. So yes, basically we don't have those standards yet. I would love that they would exist, that this terminology would already be there. So what we do is, the approach is rather like, since we don't have it a priority, can we be extra careful about really registering all the details that we possibly can, Yes, so then in long term, even if those standards don't exist now, we are already controlling the terminology, right? So hopefully when the community also like matures and then we start having like more standardization, we already have the data. Then it's just a matter of connecting it to the standards that hopefully will be more and more created. And in terms of validation, I think it's very difficult to validate generically microphysiological systems because the variables are so large. But on a long term, what we're hoping is that even if, I'll give you an example, like we make an experiment today with a certain technology. I hope that in 5 or 10 years, this technology has already evolved. So those plates will already be in version 20, right? So even then, I won't have access to the plate that we had 20 years ago or 10 years ago, let's say, but I still collected all the variables related to those systems. So then we can at least assess, okay, which are the variables that are critical for a microphysiological system to have that we have predictability. So I don't think we'll be able to say it's this system because that system will evolve, but we'll be able to say, for example, fluid flow is incredibly important. That's giving us predictability. Or, I don't know, pick your variable. We don't know that. It's a little bit a shot in the dark, but our best shot at it is to have collected that metadata. So, and that's like a huge gap because often this is overlooked. You say I use that system, but what are the details of it,
Thank you for your nice talk. I have a question with regard to your animal-free risk assessment, which you showed. So in vitro talks, typically in the past did like hazard identification, and when you do like risk assessment animal-free, how about the exposure? How do you assess the exposure in order to do a proper risk assessment?
You mean the exposure of the drug?
Of the drug. I mean, if you only
Mhm.
do in vitro, how to, typically in the risk assessment, you do exposure assessment.
So I'm not an expert because this is an ADME question, but what I can tell you is we do a lot of modeling also like PDPK. So I cannot tell you the details because that's not in my area of expertise, but we have teams like of mathematical modeling. And then what we do is, even if we use like microphysiological systems, then we need to have that layer of like mathematical modeling and simulation to translate to what you would see then either in an animal model or even like in humans. But this is not my area of expertise. If you want, I can connect you with my colleagues who actually do this work.
Great. Yeah, thanks for all the questions. Thank you. Thank you again. So we're on to the last presentation. I was looking for the speaker. Yeah. Yeah. It looks like we may be finishing early. So there's going to be lunch outside, and so it looks like we're going to finish early. I just want to thank again for all the speakers and for the great presentations for this session. Thank you. Excuse me, for the up the back, are we going to play the video? Just hold on a sec, everyone. Yeah, we can't hear the sound. Okay, so everyone, this session, this talk will be available through the recorded sessions. So we're going to finish it early then. Thank you.
A wide variety of contexts of use, from target identification to drug safety and efficacy and more. This figure does come develop from
Hello, everyone. We're about to start our very last session of this exciting meeting. And I hope you will enjoy our discussions about what we are doing and what we have been doing and what we are going to do around MPS around the globe. And we will have a great panelist on the stage in a moment. From different countries, from different regions representing MPS. But before we talk to them and ask them questions, we would like to show you a short video where they represent their region, MPS activities in their regions, what has been done and what their challenges and opportunities in their regions. So please start the video and enjoy the movie.
Good afternoon. My name is Dr. Megan LaFollette, and I'm Program Director at the North American 3Rs Collaborative. Today, I'm honored to give a microphysiological systems status update for my region. As you may know, MPS use is rapidly advancing in North America. This is evident by the many special interest groups dedicated to various areas, including developers, end users, standards, the summit, databases, drug screening, and more. There are also many developers that are headquartered in North America, 20 of which that are involved directly in my group. We also know that many end users in our region are already using MPS for internal portfolio decision-making. These decisions span across a wide variety of contexts of use, from target identification to drug safety and efficacy and more. This figure does come from developers in both North America and Europe, but I believe it to hold true for North America alone. In terms of regulatory status, we know that USA regulatory agencies are generally supportive and receptive to MPS. And we know that MPS have successfully been used in IND studies for safety assessment. Beyond safety, we also know that the FDA has accepted label expansion using only MPS efficacy data. Despite this, no MPS have been formally qualified by US regulatory agents, and so gaining consistent and clear regulatory acceptance is a critical challenge for the field. In part due to this need for validation, comparative species of MPS are a high priority for development, although liver, neuro, and kidney MPS also rank high for end user interest. Now, just to introduce my group briefly, the North American 3Rs Collaborative is a nonprofit whose mission is to advance science, innovation, and research animal welfare through collaborative 3Rs efforts. Our MPS initiative seeks to increase adoption and regulatory acceptance of MPS to further the 3Rs. We're unique in that we focus primarily on MPS developers with commercially available systems combined with a few additional members, and you can see all of their logos here. And we focus on 4 key efforts: interfacing with end users, primarily the IQ MPS affiliates, advancing regulatory acceptance, creating a technology expo, and general education. You can find our resources on our website, including both previous educational and regulatory presentations. And also stay tuned for our 2 publications, including a commercial technology overview and regulatory guidance from the developer's perspective. Finally, I do want to highlight that we've created a user-friendly technology hub that features developers and currently can be sorted by tissue type. And with that, I look forward to the roundtable questions and invite you to email me with anything that remains after the session. Thank you.
Good afternoon, Jürgens. I hope you had a great week at the MPS World Summit. My name is Peter Loskill. I'm heading the Department for Microphysiological Systems at the University of Tübingen in Germany. And today I have the honor of introducing the European Organ on Chip Society, EUROX, to you, for which I currently serve as the chair. EUROX is a non-profit, independent organization which was established to encourage and develop organ on chip research. We launched the society in November 2018, and nowadays we have more than 500 members from more than 30 different countries all over Europe, but also stemming from all over the world. The society is governed by a board of 12 elected members, and we are supported by a regulatory advisory board, which is chaired by Maurice Whelan from ECVAM, and an industrial advisory board, which is chaired by Pelin Kandaralioglu from GSK. We also have an alliance with the International Society for Stem Cell Research, ISSCR, on our home journal, Stem Cell Reports. And all in all, we're trying to foster interdisciplinary and intersectional collaboration. What does that mean in concrete terms? We're working on implementing the Organ on a Chip roadmap, which was established as part of the Fed Open Project ORCID a couple of years back. It's a roadmap that was put together by a number of stakeholder workshops in Europe focused on the development of organ on chip research in Europe. I'm not going to go into detail. If you're more interested in that, I'd like to refer you to our website. One of the other things we're doing is our annual conference. Here we merged multiple conference series that were happening in Europe before we launched the society. The aim is to have a low threshold access, which is affordable for early career researchers, and to give plenty of time for interacting and socializing. We've organized 3 conferences so far. In 2019, in person, before COVID, in Graz, chaired by Thorsten Meyer, where we had more than 200 participants. In 2020 and 2021, we had to go online. We had to chair Christine Mamry and Maria Tanya, and we had more than 400 participants and very nice keynotes and also plenty of discussions, although it was online. This year, we have the upcoming conference in a couple of weeks, which will be in Grenoble in France on 4th and 5th of July. Next year, we will be co-organizing together the MPS World Summit in Europe, in Berlin, in Germany, and we're hoping to see all of you there. If you're interested to learn more about Europe, please visit our website or follow us on social media. Thank you.
Hello, my name is Ben Moz, and today I'm going to talk about MPS status and update in Israel. So before we start, a little bit about Israel. Israel is in the Middle East, about 15 hours flight from here in New Orleans. Population of about 10 million people, 9 universities, at least 20 research groups working on MPS, at least 3 companies, and numerous governmental initiatives. Recently, we established the Israeli Organ on a Chip community. It includes about 20 groups or a little bit more from 9 different universities, and there is a really good collaboration between the groups because some focus on sensor development, platform development, IPS, disease modeling, and it is very diverse and good communication. Now let's talk about how we can fund some chips. So there are numerous of governmental initiatives. For example, recently there was a $40 million initiative for bioconvergence, $10 million for biochip program, which I'm part of it, for example, and for example, alternative technologies for animal models. And there are some industrial initiatives, such as big pharmaceutical companies, such as Teva and other companies that actually provide sponsored research. Obviously, there are also VCs and nonprofit organizations that can also support such funding opportunities. So let's talk about the implementation of these technologies in Israel. And basically, mostly it is used for disease modeling, drug discovery, and recently it is also for basic research. The main end users currently are academia, although the big pharma companies are trying to implement and to create organs on a chip units in their pharmaceutical company, but currently they are mainly do it as a sponsored research. There is no regulatory agency in Israel for organs on a chip. And basically, obviously you can get these technologies from the groups here in Israel, but the commercial platforms are available we don't have local agencies, but we can obviously buy them directly from the supplier. It is available for the public either through conferences, or it is being published in open source. So how our group actually contributes. So as you all know, organs on a chip is a very complicated technology sometimes, and you need know-how. We want to make it that it will fit into standard lab tools. So what we did, we use a 3D printed chip that can integrate with the standard well plate. And therefore you can take a standard well plate and to create it as a chip. And then you can have a customized chip that can fit to all your needs. Last thing is the challenges on the international society, mainly standardization. We need standardization. And this is a global challenge, and this is one of our challenges as well. So thank you very much.
Hello, I'm Wu from China. I'm the Dean of School of Biological Science and Medical Engineering in Southeast University and the Director of the State Key Laboratory of Bioelectronics. I'm also the founder of Chinese Organoid and Organ-on-Chip Association and an organ-on-chip company. The startup of Chinese Society of MPS is a little bit late, which just started this year. But It grows very fast. By now, we have about 400 members in 100 different affiliations, including universities, research institutes, hospitals, and companies. The major founder of MPS are the group government, including Chinese National Science Foundation, Ministry of Science and Technology, and the local government. The support also comes from industry and the VC funding. And we are glad to see that more and more startup companies appears recently. Most of the research and the products are focusing on tumor, skin, and recently, brain has also attracted a lot of attention. MPS in China is anticipated to getting applications in drug discovery and disease model. Last year, Chinese FDA approved using microfluidic and organoid model for drug evaluation, which would be a big step for MPS in China. Technology and product standards are also being proposed and will be set up soon. The first proposed national standard in China is the skin on a chip, which is hoped to be funded for a 2-year development. The commercial resources in China are limited. Only a few companies, such as EverTarget, can provide. We also established a public database. As you could see, the website with underline, the address is www.organchip.cn. The major challenge is to develop international standard and push forward technology and expanded application. So we need to communicate and cooperate. In the last few years, we have organized 4 times international conferences on MPS. Dozens of members here also attended our conferences. I hope Chinese MPS society can have some interactions with the international society. And also societies in different countries can work together to share information, technology, and development the international standard. Lastly, we have some photos to share with you. We welcome you to visit Southeast University and join our next 5th conference. The conference will be announced later in June. Thank you.
Hello, my name is Youngjae Jo. I'm very happy to be able to introduce the current state of Korean MPS in this meeting. And I would like to apologize in advance for not being able to attend in person due to my personal circumstances. First of all, the overview of MPS status in Korea corresponds to the community interest groups level. The largest financial support for MPS is the government. Recently, various Korean governmental agencies are providing financial support for MPS research, and the key stakeholder is currently the Ministry of Trade, Industry, and Energy. This is the first representative project of 3D tissue chip in our country. Yes, sure. Training for new researchers is also very important in Korea. For example, the OK MPS Validation Center is going to create an institution to verify MPS produced in Korea. This center is also preparing some technical education program for MPS investigators. Now, special 3D tissue chip for skin and ocular model for irritation test has been developed in Korea. And about this question, as far as the R&D level, all of the areas were touched by MPS, but there is a scarce MPS used as the industrial level. Previous 2 tissue chips are certified as OECD TG by promoting international standardization with COCBAM under the Korean FDA guidance. However, it is still applied only to cosmetics. And fortunately, there are several MPS companies based in Korea. And at the same time, the products from leading foreign MPS companies are also being introduced to Korea. In our country, MPS data are mostly spread to the public as a part of R&D activities. And these 2 groups are the major Korean R&D group for MPS. And the challenging point related to MPS in Korea are various, but I think a diverse research team is collaborating for the same purpose of MPS. For example, international cooperation is also being emphasized in our project, and Professor Nuri John and I will be in charge of this project as co-PIs. This is the last question. And in our country, there are still many things that need to be overcome about MPS. And of course, having an international scientific society would be of great help. A similar symposium will be held for the first time in Korea soon. And I've already invited Dr. Taegul to visit Korea. And I'm looking forward to seeing you in South Korea. And thank you very much again for these great opportunities.
Hello, my name is Yasuyuki Sakai, working at the University of Tokyo, and also I am involved in the EMEDO MPS project and also the activity of the Japanese Society of Alternatives to Animal Experimentations. I'd like to briefly update MPS status in Japan. In Japan, researchers in industries such as pharmaceutical, food, cosmetic, and chemicals has begun to try to use MPS of both foreign and domestic companies' product, with their strong expectation to better prediction. Last 5 years, EMEDO set up a national project and where we developed 3 types of new MPS. And also we are now discussing about setting up the Japanese MPS consortium. And the last 5 years, we have been focusing on liver, small intestine, kidney, and blood-brain barrier. And this EMEDO project continues next 5 years, where we need to focus on the extensive evaluation of the developed MPS in terms of the COUs proposed by the users. In the past 5 years in EMEDO project, we have been focusing on pharmacokinetics and safety issues. However, still the MPS has not yet been qualified in regulations. But through the MPS project, we developed a user-friendly, cost-effective 3 types of MPS, and also iPS-derived small intestinal cells. I'd like to talk about it in the next slide. This slide shows the 3 MPS developed. First one is peristaltic plate type 1. This is very easy to use, and I have been focusing on the co-culture of enterocyte and hepatocyte. And second one is microfluidic type, and there's a membrane, macroporous membrane, and both sides there's a perfusable compartment where we can strictly control the shear stress. And this device is suitable for the co-culture of kidney and endothelial cells. And the third one is quite a unique one. It employs microstartup-based pumping on-chip perfusion system and also direct oxygenation from the bottom. And this is very important to enhance the function of the small intestine and liver cells. All these 3 are cost-effective and have high usability. Now we can supply 3 types of MPS and iPS-derived small intestine cells. However, still, the understanding of the regulation agency, such as PMDA, is not enough. To overcome this, we need extensive evaluation against the specific COUs. And also, we strongly hope the progress and the development of the MPS World Summit, and we are very much happy to be deeply involved in that activity. Thank you.
On the MPS state of play in Australasia. So there's no overarching society. I propose that the Australasian Society for Clinical and Experimental Pharmacology and Toxicology is a suitable society to base MPS activities in terms of an annual conference in Australia. There are other groups, significantly smaller, Medical Advances Without Animals, and the Associated Australian Centre for Alternatives to Animal Research. The focus of these groups is more strongly on in silico methods than on MPS systems. There's also the ARC Centre for Personalised Therapeutics Technologies, which runs out to 2024. As is common, government, philanthropic and industry sources of funding are obtained to support MPS activities. And yes, there is training involved with the training of postdocs and PhDs at the ARC CPTT, but the scale and duration of this effort is limited. There's a more long-lived training available at the Melbourne Centre for Nanofabrication, which provides instruction in microfluidic design and fabrication, but not in MPS systems as such. There's considerable expertise in kidney on a chip and a major collaboration with Melissa Little, CEO of the Novo Nordisk Foundation Centre for Stem Cell Medicine. There's a range of different MPS type systems in use, focusing on cardiovascular safety regeneration, on genetic diseases using iPSCs, on improving predictive pharmacology, and on physiological emulation through the control of dynamic control of media. There's also main end users being research and industry philanthropic collaborators, but there are no patient groups using MPS at this stage. There has been a company formed in Australia, In Vitro Q, but it's not operational locally. It's operational in Asia and Germany, and they're rolling out a product called OncoPDO, oncology personalised organoids for predictive pharmacology of commonly used chemotherapy agents. In terms of industry, the Schott Minifab is a supplier of MPS-specific microfluidic devices, and there are other companies involved, such as Trajan, which is assisting the ARC CPTT in refining and commercialising its multiplex dynamic media formulation technology. We also have some challenges, of course, as we all do. There's some skepticism as to the impacts of MPS, how large and what is the lag time. So this can limit resources. But I think on the very positive side, the International MPSS will provide a profile and a bridge to the local cognate society, ASCEP, to be able to further promote these activities.
Good afternoon. I'm Dr. Grave from Brazilian Center for Validation of Alternative Methods, and I'm talking about MPS in Brazil, state of the art and challenge. Brackman is still preparing a survey to be sent to laboratories, industries, universities, and any institutions that use animal in experimentation in order to map the use and need for using alternative methods. MPS in Brazil is starting to be implemented. So as I know, we have only 2 government institutions and 1 startup working on it. The 2 government institutions are Fiocruz, unit in Rio de Janeiro, and the unit in Paraná, that are 2 states of Brazil, and National Laboratory of Biosciences. And the main organ and systems that are being studied are related to cardiomyocytes, kidney, liver, neurons, and intestine. The main areas that are being developed in Brazil are basic research and other use for regulatory acceptance. Brazilian regulatory agents has no position about MPS up to now. I think that they are waiting for the fully implementation of this technology in Brazil. And we still need international supplies for our studies. Funding is the main challenge for the development of MPS technology in Brazil, since up to now we had only 1 public notice by the Ministry of Science, Technology and Information, and 1 project for the inner employees in Fiocruz. That was an innovation project using organ ownership. I think that an international society would be very helpful for Brazilian researchers, as well as an event like this one, since we may keep in contact with the development of technologies and people that are really using these technologies, and maybe start a partnership. So I'd like to thank everybody for being here in this Congress, and I'd like to say a special thank for my crew, for my group in Brackman, and for the collaborators that made possible this presentation. Okay, and you can find the email and the web page of Brackman. Thank you, everybody.
Okay, well, now we will ask all the panelists to go up on the stage and we will make a short round of introductions. And please, when you introduce yourself, also you add something you would like to add to the video you just showed. So I will start with me. I'm Lena Smirnova. I'm at Center for Alternative to Animal Testing in Johns Hopkins. I am chairing this session together with Dan.
Dan Tagley, welcome again to this panel. So we'd ask the panelists to introduce themselves and perhaps if you can provide one or two sentences, something that you wish you had said on the video that you weren't able to cover.
Yes, thank you, Dan. I'm Megan LaFollette from the North American 3Rs Collaborative, speaking for the United States. And I think, certainly learned a lot during this conference and have some updated opinions. Just one that I want to bring to the forefront is that I could maybe change in that video now is to not become overly focused on the so-called regulatory acceptance. Certainly it is important, but there is huge scope and potential and a lot of other challenges to work on in terms of qualification validation, internal portfolio decision-making. And as I did say in my presentation, the FDA is already accepting MPS data for IND and efficacy. So although this issue is important, I think we should not become too overly focused on it.
Hello, my name is Yasuki Sakai, coming from Tokyo, Japan. And as I said in my video, and also probably you already feel the same thing, but the great success of the first World Summit, MPS Summit, I very much, actually I personally studied a lot about not only the scientific part, but also the implementation, particularly pharmaceutical area. And as one of the representative of Japanese colleagues, I'd like to, I may say that Japanese researchers from now on strongly be involved in the world activity of MPS, including in the future, we want to host the World Summit in Japan in the future. Thank you.
Hello, everybody. My name is Otávio Presgrave. I'm the coordinator of Brackman, Brazilian Center for Validation of Alternative Methods. And as I told him in my presentation, Brazil is starting to use MPS. And what's good for us is that we have a structure for validation and become an official alternative method to have three entities related to this, which is the CONSEA, which is the National Council of Control of Animal Experimentation. It's responsible for officializing methods, alternative methods in Brazil. We have the BRACVAM, and we have the RENAMA, which is the National Network on Alternative Methods. So I think that the efforts of these three entities will push up these studies and development of MPS in Brazil.
Hello, I'm Alistair Stewart from University of Melbourne and director of the ARC Centre for Personalised Therapeutics Technologies, which is a training centre with 10 RHD candidates, and we look to sustainable training, and this is one of the challenges that is faced, that there isn't a national focused fund like the NIH NCATS. We were privileged to have Dan Tagle visit us and lecture and assist our training program at its outset in 2018. But we need a more sustainable approach. I would say with regard to the regulation that In Vitro Q presented a test case to the regulator in Australia called the TGA. And in that respect, the TGA was able to identify a category, a regulatory category for the individualized patient prediction of chemotherapy, which should not be used. That was their particular model, to use organoids to detect chemotherapy, which would be ineffective. Thank you.
Good afternoon, everyone. My name is Peter Loskill. I'm from the University of Tübingen, but here representing Eurox. And I think one of the things I didn't mention that much in the video is that we are a grassroots-driven academic society. We obviously also have a lot of people from pharma and are targeting the application of these systems in pharma. But one of the points I'm trying to stress all the time is to not only look at pharma for end users. I think the technology has a huge potential looking in biomedical academic research. This is where the majority of animal studies are run. This is where there is potential for huge impact. And this is also something where, let's say, regulatory aspects are not that big of a topic. So instead of focusing everything on regulators and putting all the pressure on regulators, I think we should put pressure on the scientists to use these systems.
Thank you, Peter. My name is Ben Maoz. I'm from Tel Aviv University in Israel. First of all, I would like to thank again the organizers for creating this amazing event and also for inviting me. Also, for me, it's very nice to be in this panel because I actually see there are people who had to take longer flights than I am to get here. So it was actually nice. I will not repeat what I said in the presentation over there. I will just say two additional things. That although Israel is a very small country, right? We are about 10 million people. I think that if you think capita-wise, we're the #1 in the world, the amount of groups that are actually working on organs on a chip per capita. Maybe the Netherlands is basically the same, but we have more than 20 groups that actually work on organs on a chip. We have a number of companies, I think at least 3, and we have a number of governmental initiatives. So organs on a chip is something that is nicely developed and in Israel, and I encourage you all to come, visit, and to contact. We established the Organ on a Chip Society, so if you need some contacts, a specific contact, please let me know, email me, and I'll be more than happy to help. Thanks.
Thank you. Hi, hi. My name is Nuli John. Yeah, actually, I was supposed to be here on the first day, but my flight got canceled, and I had to arrive a day late. But anyway, I'm sitting here in place of Dr. Cho, who is an MD, a practicing MD, so he couldn't make it due to some scheduling reasons. Well, first of all, I think Korea, has a very high number of microfluidics and organ-on-a-chip groups. I think as high as Netherlands or Israel, I think. We have a lot of few startups. One was here, and we have one company in Korea that was also acquired by M&A by a bigger pharma type of company. So we have a really vibrant community, and I think we also have a lot of enthusiastic medical MDs who are interested in this technology. I think one thing that we lack is the government support. So we've been doing our research on our own and using small individual grants, but I think hopefully we will, we just started getting bigger grants. But I think this type of collaborations with international, society and other researchers will be very fruitful and have a will lead to more collaborations and more success in this field. Thank you very much.
Thank you for a nice round of introduction. So what we saw from the video and also from your descriptions now, we see that there are differences between different regions and countries, and there are also things which are in common, obviously. So, but my first question would be about the difference. So what do you think the driver force of these differences? Is it the availability of the technologies, availability of fundings, or it's maybe just the focus of this particular country or the region in their focus on health perspective? So, Peter, do you want to start?
Yeah, I think, I can having worked in the US and in Europe, I can compare at least US and Europe very well. And I think it really depends on what is the starting point. And I think here in the US, you had the program, which started everything, and where you have a huge funding opportunity and driven by governmental institutions. And I think this is very unique to the US. I think and nowhere in the world we have that. In Europe, we never had a program really on organ on a chip. We had some countries like the Netherlands going really forward, establishing national societies, national institutions, and this then was driven to basically, as I mentioned, more of in a grassroots style. So the academics came together, we formed a society, and we're trying to thereby form the field. So I think that it really depends on what is the outer framework. If you have a funding agency like in the US, which is forward-looking, this will give you a totally different situation.
Yeah, I can tell my perspective about Israel in that sense, that I think in Israel it's a combination of two things. First of all, because it's a small country and everybody knows everybody. So people that are working with tissue engineering, they talk with people that do microfluidics, which they talk with people who do sensors. So the part of the integration is pretty straightforward. So when everything started, and people that are working for a lot of time with microfluidics, but they never had any experience with tissue engineering, they just go to the next university, which is 30 minutes by car, and they collaborate. So it was a very natural thing. The second thing is that also governmental initiatives started, both for alternatives for animals, but also for things related more to microfluidic chips. And the third thing is that you have a number of pharmaceutical companies that everything started. For them, it was a great opportunity to try also to invest instead of creating their own units in their own pharmaceutical companies, basically to invest in the academia to do sponsored research. And basically, it will help them to push the field a little bit forward so that afterwards they can have a benefit from this. So I think these are basically the three reasons, at least in Israel.
Well, I think that all factors mentioned are very important, and I think that they differ from country to country. It depends on the stage of development in terms of size and financial of each countries, and perhaps I would introduce another factor that is how much the NRA, for example, of a country believe or not in the power of an alternative method, or how much the NRA agree with or still believe that the animal experimentation is too important. I think that all factors I mentioned are very important, and I would include this also.
Okay, thank you. Ben actually answered also my second question about what is the effort to synergize the activity around MPS in the region. So it's basically everybody should know everyone. This is the answer. I will open to the podium, to the audience.
Questions from the audience to a particular panelist or the whole panel? Anyone? If not, we'll go to the next question.
Yeah, I have no questions? Okay. Then.
Thank you very much, all. How do you see that global collaboration on MPS would help to advance the acceptance of the technology as a possible alternative for future translational and personalized medicine? Thank you.
To the whole panel.
Yeah. Global questions.
Alistair.
Coming from the most remote and perhaps the second smallest country in population terms, I think we need the muscle of an international organization such as has been created to get leverage from our funding agencies and our government And so this global collaboration is absolutely key to raising the profile of MPS in Australia, such that the sporadic funding that comes is easier to secure.
Well, I think that MPS, although is in the state of advancement around the world, I think this is still a puzzle. So we need to collaborate each other countries in order to complete all this puzzle and maybe harmonize the methodology and processes. I think this would be an answer to your question.
If I may add just to what was just said, is that first of all, I completely agree that such events are crucial to make it better. First of all, the networking, but also to see what other people are doing and to create communities. So definitely this is number one. I think that there are two other parameters. First of all, that the companies that were presented, outside over there, they should reach all the other countries, right? So to make it more accessible, at least technology-wise, that not all the people will need to start and to invent the wheel. And they will bring this technology that it will be easier to use. And the third thing, I think that it's extremely important to see whenever you want to collaborate with someone, that you do it in a smart way, that it will be really a synergy. Because two groups that are doing the same thing to trying to bring cells from one side of the globe to the other side and to try to integrate the cells that they're doing with the cells that they're working, it's a bit challenging. So it needs to be really a group that one is creating a platform and the other one is doing the biology, or one is doing the modeling and the other one is doing the data. So you need to find the right collaboration and the way of collaboration. This is my input in this.
And I would just add that agree with everything the panelists have said, but also just having those key early adopters, wherever they are in the world, who are willing to take the leap, that are willing to put in the efforts, whether they're academics, whether they're pharma, whether they're companies. And the more that the international society can champion them, can lift them up, can help show them, because ultimately we're humans, we're social species. We want to see, we want to follow the trends, we want to fit in, and we're quite risk averse as well. And so the more that we can champion and learn and be realistic about where the challenges are and what was necessary to overcome those challenges.
Maybe adding one more thing. I mean, why do we need to be global? Because the challenge we're dealing with is global. So the health challenges, COVID was one of the perfect examples. We all have the same challenges. And also the end users of the technology are typically global big companies. And now I'm contradicting to what I was saying earlier. It's not only pharma as end user, but also the scientists. It's a global community. So it is absolutely natural to address this challenge also in a global manner.
I think we had one more question in the back.
Let's meet halfway.
I was dreaming. Thanks. Yeah, Claudia Gärtner from Micromobility Chip Shop. We are not doing such fancy research as you, that implementing the biology on chip or microsystems. We are a fabrication company, and there my questions come from who or how do you implement the easy engineering part to make your system afterwards happening? We saw this in a diagnostic world where the quantities of consumables are much higher. So how to address the fabrication? And the second question would be, we had this funding topic, and on the podium, we have 3 countries which we have partners in German funded projects where we have bilateral projects running. Are there other opportunities? Brazil is open, Israel is open, and Korea is open for such small funded project to broaden this way that we get small project for the small companies involved running. So 2 questions there, fabrication and small funded projects.
Okay, I will start. Regarding the second question, funded the projects. So Specifically, I can obviously answer for Israel. We do have projects that are, as you said, like Israel, Germany, for example. So you have the DEEP, the DevGay, you have the GIF as well. So you have many different specific opportunities, but you also have a lot from we have now in Israel. They develop a really nice concept of the innovation authority, where you can come for projects that are specifically between academia and industry, because they want to make more things that will become industry and startups. So you can find a specific group in the academia that you will try to do something that will become a product or IP or something like this, and you can get a lot of funding for these as well. So this is regarding specifically in Israel. I didn't completely understood the first question, I must say, but it was something regarding integration with the chips or integration with the biology in the chips, or if someone else.
I think it's microfabrication.
Yeah, exactly. How you have the application end, but also the fabrication. Is this of interest, and how to embed? For instance, we were talking a lot about standardization. Can we drive this, for instance, in easing fabrication?
So obviously there are a number of companies, and if there's something that you want to do easily, so obviously there is the company that you're representing. There are other companies as well that some of them are doing custom-made chips, and some they have their own standard chips. So obviously it can be something that people can use and talk with the international suppliers. But specifically also, if you want to have a custom chip, so there are specifically in Israel, there are a number of groups that are really good with microfluidics. So you can definitely talk with them and to see if they can fabricate something for you. But then the question is the quantity, right? Like if it's something that you need a lot from, then obviously it becomes a little bit more sophisticated or need to be addressed. Hope I answered the question. Anyone? Okay.
So regarding the funding, do you think there are opportunities to work with your government funding agencies to create a program across the globe similar to the Human Frontiers in Science, where investigators from different countries work together and are funded by their own respective country, but it's all synergized and done collaboratively?
Was it for something? Was it for me or general?
For any country that you seem that your government agencies or funding agencies are receptive to that idea?
So for me, the simple answer is yes, definitely yes. The government and the Ministry of Science and Ministry of Technology are extremely open for such initiatives. And they have recently they launched this $40 million initiative for bioconvergence and these kind of things. So for Israel, yes, for sure.
The same happens in Brazil. Basically, our funds come from the government, from federal stage, and sometimes from the states. And it's possible for us also to make collaborative studies with industries and other institutions. But basically, our fundings come from the Ministry of Science, Health and Information and Ministry of Health.
This is a shameless advertisement. In Australia, there is an R&D tax credit, which means that every dollar spent earns 43% back. And so this does tend to facilitate industry collaboration. There are also schemes in place for linkage grants from the Australian Research Council that leverage government investment. And we have a National Research Infrastructure Fund. And in that grouping, there is a therapeutics innovation group. And certainly I'm going to be lobbying that group to see if we can set up training and funding streams for MPS systems, because I think we do need something that's more systematic than the sporadic successes that happen at the open competitions in the NHMRC, the National Health and Medical Research Council.
As far as the international collaboration funding, I think I still have a grant with the previous person, Claudia, but it's Korea, Germany grant, but it's open over whole, bio spectrum or others. And then we were just lucky enough to get the grant and then use the funding to work on this. And that also kind of on this MPS project, but that also kind of gets into the question of fabrication, because not a whole lot of people have fabrication knowledge. And I think nowadays there are so many designs that's being made up for their own applications. But I think as the field matures, I think maybe as an international society, maybe we should have come up with a format like SBS type of format or something. Probably it'll be the web plate format, right? Because it has to be compatible with all the equipment that's out there. But in order to make those things, I think the largest hurdle for us was the cost of making custom chips with a polystyrene or injection molding houses. So I think we need to maybe, as a field, come up with some kind of put an effort into establishing a standard so that maybe it can be either plug and play or at least have some basic units that can be used across different areas.
Maybe I can add to this last point. It goes a little bit back from Dan's initial question, actually. But in Europe, we have started a process on getting a standardization focus group. And Dries van der Meer, I don't know if he's still here, is heading that. He's back there waving. And because this is an extremely important aspect. And I think this is something that the global community should do together to standardize. And to also define where standardization can be done is necessary and where it can be done without limiting innovation, because this is always the balance. But very briefly also to Dan's question, I think this is one of the biggest or most important things you can collaborate only if you have funding for collaborative projects. And I know, we had this discussion many times. I know all the efforts you're taking, and I think this we have to now support you as a global community. We have managed in Europe to have a good funding agency that funds all the different countries, and this should be something to try to do on a worldwide scale.
Hello, I'm Zorae Zadifar from Wyss Institute. I'm seeing a panel of all these leaders coming from around the world. I just came to my mind, there are a lot of diseases and health conditions that we see disparity among different ethnic backgrounds and genetic backgrounds. And seeing this panel, I think it's a great opportunity that there would be global collaborations. They have all these systems and you have access to your own population with the ethnic background. And we can, working together, we can make that database to have better knowledge of how our ethnic background is contributing to that. And I wanted to see, if, as leaders, how you see that coming along in the future. And if there will be funding, for instance, I don't see anyone from African countries here, but they are one of the populations that in most of the cases, they're more susceptible to a lot of diseases. And maybe at the moment, they don't have a lot of funding or the technology is not much established, but together, maybe there's opportunities to help kind of universities in those less privileged countries to also have access to the technology to study.
Yeah, I think this is an extremely important point. We are from around the world sitting here, but we're missing in big parts of the world. And I think this should be one of the big focuses to help these areas where there is not, and we've seen there's even in the representation here, there's a different level of research on, or the field in itself is in a different level. But we should try to get a global community, and this includes all the continents and countries that are not yet involved here. So I think this is one of the most important things. Also, and again, now I'm referring again to pharmaceutical companies. I think this is also something, talking to pharma companies, one of the challenges they are now more and more experiencing to see to not only develop drugs for Caucasian males, but to develop drugs that work all around the world. Yeah.
I think I want to build on the less privileged parts of the world. Maybe when we have a global society, could have an initiative to support some training for less developed countries. So trainees could start from there, the first train, they know about the technology, go home and then establish. And this should be like much of industrial, probably driven or funded. Thank you.
Yeah, I was just going to add and basically say that same thing, in that I think an international society could do great aims to have a, you know, whatever you want to call it, developing country, underrepresented ethnicity groups, whatever, and have funds for those individuals, for those countries, so that we are bringing them to the table, that we're not necessarily trying to speak for them, and supporting growth within their own country as much as possible. You know, we have to be realistic, but we should also be doing the best that we can. So again, thank you for the question asker for bringing that up.
If I can just add one more point, is that I completely agree that we need to push forward the technology, but we also need to keep in mind that the technology itself is expensive, right? So if you also don't have the funds to use it, it is also a challenge. So if we will come and we will have free training all over the world, and we'll give free chips for all over the world, it will be currently still more expensive than just to use regular well plates, right? So we also need to make sure that if we want that every lab will be able to work with organs on a chip, we need to make sure that the price is competitive to places that it's not, you know, that they don't have the funds to support this technology. So this is just, I think, a point we need to keep in mind.
Okay, we have last 2 minutes. I will add to the education question. We also need to think about to educate our youngest, the new coming researchers and young investigators. So somebody, maybe you can reflect what is happening in your region in terms of the education of new generation of scientists about MPS. Yes.
Okay, actually, I'm working at the university, so even in Japan, I feel every year students who are get interested in 3 arts are increasing, even in engineering school. So, but they are still young and naive and don't know everything. So that's why, I think we need, it's a good opportunity to show a kind of roadmap, a scientific roadmap. It's easy, understandable manner. It's very important for young students. So it's a very good opportunity because we have a very quick advancement of the MPS technology, and it's a very good opportunity to expand the in vitro system in the every sector of the society, and probably if we think about the future, such young student. It's very important, of course, it's very important, and we need to guide in a more scientific manner to show a kind of roadmap.
Yeah, I can just add that, at least from my knowledge in the United States, as well as many places, you know, training of young investigators takes place within the context of a specific project. So I do know there were at least 12 different universities that had invited speakers at this summit. And so we certainly have a lot of universities that have those programs, but I don't know how well those programs are known to the wider young population, you know, MPS, even to the public. Whenever I talk about what I'm doing or where I'm going, people have no idea what I'm talking about. So I think, you know, having the International Society focus, you know, on that student population, maybe having a place where grad student positions are being posted, maybe having courses, funding for the students as well. But also focusing some on that public outreach so people even know what these are, because if they don't even know they exist, it's hard to imagine. They kind of will just come by them by chance instead of taking that initiative. So public outreach, I think, is also going to be an important factor.
Anything to add? We are perfect on the top of the hour of this session. I will probably allow one more question from the audience if there are any. If not, then we will move to this closing ceremony in 5 minutes. Thank you very much for attending this session.
Yeah, thank you, panelists. Thank you.
Okay, so please join me for the last finish line we have to meet. And I promise you there's some interesting things awaiting you. We will talk about what is coming. We will talk about who won the prizes, poster and presentations. So please come in and listen in. So can I have your attention, please? Good. Okay, it is my great pleasure to get us started with Susie Fitzpatrick. Susie is the voice of MPS and FDA and has been instrumental in getting all of this started, as you have heard several times. She was our keynote speaker in the first of our pre-meetings, our virtual pre-meetings, and she's going to open our closing ceremony now. Can we have her slides, please?
Do I push this? Okay, first I want to say all of the science is really amazing. When I was in the commissioner's office 10 years ago, and they came in with a picture of a box with a funnel and an arrow going out and said, we want to develop a body on a chip. And all of us just in the commissioner's office just said, okay, all right, you know, and I wish Dr. Pilata was here today to see everything that he did and Dan Tagley did as the two real pioneers in pushing this technology forward. And it's really amazing, and amazing for a regulator to see all the potential new tools that we can have to get new products on the market. So I have the privilege of giving you the stats. We had a lot of attendees. Thomas was worried about it. Probably he shouldn't have been. We had 667 registered, 450 in person, 215 online. A lot of FDA people were online because we're barely getting to travel, but they didn't want to miss this. So they were all crowding together around computers and taking advantage of looking at this. 303 academic and government, and some of those in person, some online, 255 industry, some in person, some online, and 107 students, really the future of this whole technology. And I'm glad that Kat does so much to encourage students. The next one is, as you can see, we have them from all parts of the country, not only the United States, 413 people, and really we're just starting to travel too. So this is only my second time out of the office in 2.5 years. So, but I won't read all the countries. You can see it. You heard all of the partners in there, and I think people have, Arlena, they're going to have email addresses for people if they wanted to actually think about partnerships. I know we do a lot of partnerships at FDA. CRADA is where we bring your technology. We have lots and lots of chips in our labs to learn about chips, and research collaboration agreements. So if you've got a good idea, come give a seminar at FDA and introduce your chips to us, and and sometimes we adopt them. The abstracts, they had a lot of abstracts. Is this your part now? A lot of abstracts, many from young investigators, which I think is really neat, and 142 speakers, some invited, some selected from the abstracts, 164 posters and 24 posters online. That's a lot for a meeting that, you know, we're putting together, and it's a lot, and it's a lot of great data. And I think that's it. And again, I'll just say thank you on behalf of FDA for developing really great tools that we are anxious to use in our application. Thanks, Dan, for being a great partner too. So.
Okay, I have a privilege to announce our awardees of the best poster presentation, best oral presentations, and also, can I start slides? Yeah, first of all, before we go to that, I want to emphasize that we had some travel awards and travel grants, which we gave 22 domestic travel grants for students and postdocs and 13 international travel grants. And this was made possible by our grant, which we received from NCAT, but also Next Generation Humane Science Travel Award. And again, Best Poster and Oral Awards were also supported by NCAT. And I will call the names, and I will please ask people to stand up as you can get the round of applause. So First of all, I want to say that it was extremely competitive for the poster award because we had over 100 posters from young investigators. So it had 2 rounds of evaluation. First round was abstract, which was judged by at least 3 judges, and then the second round was poster presentation, was judged by another 3 judges. So every presenter got at least 6 people judged their performance. So, and I will move forward to our third place of the grad student poster award, which is Daulet Alan Eldin from Johns Hopkins University. And our third place postdoctoral or young investigator poster award goes to Julia Marti from University of Tübingen. Our second place best poster award from grad student goes to Emerali Salahi from Texas A&M. And our second place for the best poster award by young investigator presenter goes to Lauren Richardson from University of Texas Medical School. Okay, and now the first place. The first place for the best poster award from grad student goes to Alan Kim from Johns Hopkins University. And the first place for the best poster award by the postdoctoral trainee or young investigator goes to Alice Stanton from MIT. Okay, so that was posters. We also judged our oral presentations by young investigators, early career scientists, and we had also postdoctoral trainings or early career scientists or PhD students. So I will start with third prize for the PhD oral talk, which goes to Ellen Ken from MIT. Sorry, it's postdoc, I misread. I'm sorry. Now I'm going to grad students. So the third place for the best talk by the grad student goes to James Tronolon from Texas MI. Good. Okay. Second prize for the postdoc or early career presenter goes to Jeremy Huang from MIT. And the second prize by grad student goes to Raphael Yaga-Madior, University of Paris. Okay, and now the first place. The first prize for the best oral presentation, well deserved, go to Lauren Richardson from UTMB. And the very well-deserved first prize for the best oral presentation by grad student goes to Devin Meyer from Johns Hopkins. And we also had a competition which was supported by ESTIF. And ESTIF is European Society of Toxicology in Vitro. And there was an application process they were looking for researchers who are working in vitro toxicology and in silico methods to promote the field. And they also awarded 2 posters and 1 oral presentation. So ESTIF Best Poster Award for PhD candidate goes to Erika Ferrari from Politecnico di Milano, Italy. And Best Postdoctoral Poster Presentation Award goes to the same institution, Roberto Visone. And the Best ESTIF Oral Presentation by Early Career Investigator go to Carolina Romero from Johns Hopkins. So with that, I will conclude our awardee ceremony session, and we will move to announcement of our global society, MPS. Then.
All right, can I have the slides, please? I think you've heard through the global panel or probably throughout the meeting mentions of a professional society. So it is a great privilege to announce the formation of the International MPS Microphysiological System Society. You have the website there. Come and visit. It's not active yet, but hopefully soon. And let's use this as a forum to exchange ideas. Let's continue the collaboration. And perhaps we could also set up discussion forums, focus topic groups and things like that through the symposium, through the society. And then, so one of the things that the committee decided in terms of what to be all-inclusive in trying to define what microphysiological systems are, and so this is sort of like a word cloud that would encompass pretty much, I think, everything that people are doing in this organization or in this conference. The Society's vision is to accelerate the use of MPS to improve health, welfare, and the environment. Our mission, as you saw from the tagline of this conference, is to connect, exchange, and educate. And so I hope that the connection, the exchange of information and the education will continue beyond this conference, and it will hopefully will build up to the next conference next year. Let's not forget about the networking connections that we forge at this conference and continue those kind of interactions. The core values of the society would be excellence and integrity, certainly shown by tremendous scientific progress and just the science, just overall quality of the science presented. Diversity, equity, and inclusion. You heard last night from the reception, even from the host group, the New Orleans Chamber of Commerce, that it was so impressed with the number of women, number of people of diverse background being represented in this conference. So let's continue on that track. Innovation and impact is certainly another core value. Respect and accountability, connection and collaboration. And connection and collaboration, as you already saw and experienced, can happen also in very fun environment. So let's not just be scientific geeks. Let's have fun doing science. And education and career. I mean, the training is certainly an important focus, not only of the conference, but certainly I think it should be perpetuated by the society. And of course, professional and career development should also be a core value with that. In terms of the structure, this is sort of like the proposed structure of the society. There will be a governing board. There will be regional chapters. And right now within the regional chapters, we have Europe, Americas, Asia, and the Pacific region, including Australia. And then, of course, there are going to be interest groups. There are thematic interest groups, ADME, DMPK, PBPK, disease modeling, toxicology, drug discovery, and then personalized health. We will have an interest group also on policymakers and regulators, NGOs, as well as industry. I think it's your turn.
Yeah, and as Don already mentioned, it was a work of a committee which we established in preparation toward the society, and this was MPS Society Organizing Committee, and I would like to thank all these people who were working hard with us on creating the vision, mission, core values, working on our structure and how we can implement our thoughts in the action. And so these people will work hard, and they will continue work hard this afternoon. We have a satellite meeting for us. It's not over. And so this committee proposed members for Board of Trustees, which was then elected by our steering group, which represents 52 organizations. So we are now finalizing the Board of Trustees, which will then, from this meeting, will take over from the organizing committee and will work the next year to come on bylaws for the society, registration for the society. It's a lot of bureaucratic work around the society to kick off that. And then the Board of Trustees will be replaced by Governor Board, President, President-elect, and so on. And how it will be elected, we would like to have obviously members in our society. And so this board, Governor Board, will be elected by the members. And we would like to invite all of you online and in person to become a founding members of this society today. And I would like to get a round of applause if you agree. I will not ask for a round of applause if you disagree. Move forward. Yeah, we will have individual members, but also we are looking for corporate members, organizational societies, and also donors. And stay tuned. Our site is under construction, but it will be up probably next week, where you will find more information, also our Board of Trustees and our plan for the next year and years to come. Thank you.
So good. We have a society and you're all members. Isn't this great? Good. And the best thing is there's no fee yet. Okay, let's come to the next highlight, which is Berlin is coming. And with Berlin, I asked Uwe Marx, one of the 3 co-hosts of the Berlin event for next year, to tell us what they have up their sleeves.
Thank you very much. First of all, I would like to thank Dan and the NIH for making all these sponsoring the first 3 congresses, and the entire committees which you have seen, and everybody who has been here to set the bar at that high here in New Orleans. And then I would like to thank you that you decided to have the 2nd Congress in Europe, and specifically would like to thank you that this will be in Berlin, because ich bin ein Berliner, and I can pronounce it like John F. Kennedy. Ich bin ein Berliner. Who of you in the room knows that phrase from the historical date? No, not everyone. We have quite young people. That's why I'm asking. But probably not everybody, even of those who know the phrase, know the speech. But the date is important. It was June 26, 1963, as you see there. 1 year later, I was born, exactly 1 year later, in Berlin. So my mother was so impressed that, okay, let's do it. So we will come back to the date a little bit later, but maybe one of the incentives to make it in Berlin is because in Berlin we have already a certain background in MPS, and that background comes from the fact that I spent nearly all of my conscious life in Berlin, and finally was able in 2010 to acquire a multi-organ bioreactor program from the Ministry of Education and Research in Germany, 10 million for 10 years under 2 conditions for my vision to build humans on a chip. The first condition were come back from the industry, my own founded companies, and go back to any of the universities, build that thing in the university, and then and the second one is make it available in medicine and commercially viable. So that were the two conditions, and I decided and find a wonderful environment at the Technische Universität Berlin, at the Department of medical biotechnology of the Institute of Biotechnology, where we started in 2010 and immediately founded the company in order to really make that second condition happening. Now, since the 10 years of development, or in addition to the success which we have had in research and which we had in building the company and the business and the interrelationship and the society and all our workshops in Berlin, my successor, Professor Roland Lauster, the head of the medical biotechnology department, was able in parallel to team up with the Charité, with a professor in the Charité, and the Charité is one of the largest university medical centers in Europe. And they decided along that path to come up with an interdisciplinary research center exploring MPS technologies and modeling and AI technologies. And they called it der simulierte Mensch, the simulated human, to revolutionize cancer therapies, first in the Charité and then abroad, shift the paradigm in transplant therapies, and reduce and replace animal tests in basic research. So along our developments with the MPS, along the MPS were coming from other parts of the world, also to Berlin. It is a research hub in Berlin. That decision was made by the city, sponsored also by the government, the German government, to build for 34 million this building here. And we really hope, and I hope you cross the fingers, that it is available for your visit at the Congress time. So that was maybe one of the incentives to take Berlin, but a much more, maybe even important incentive is obviously that Berlin is probably the only city which experienced for the last 100 years such a tremendous historical changes, with such a tremendous impact on what is happening with the citizens. That shaped our current next year city very much. And you might probably, or not all, but many remember that 100 years ago, we started in the 20s with the so-called Goldene Zwanziger, the golden 20s in Germany. So that was a period from 24 to 29 where we connected, where we exchanged, where we educated, and a prosperous life was running up in Berlin. And then for a while it became darker and darker and darker, including the Nazi era, after the end of the war, it immediately turned into the Cold War. And the most traumatic then situation for the Berlin citizens was the wall which divided the city into two parts. I schematically put these two parts here in blue and in red. So at that time, nearly no connection and no exchange was possible with the eastern part of the city, and that is really a traumatic, and I would say humanity a threat, and a threat for the population there. And there were two important dates and events where the Berliners were fully emotional in a positive way. And these two events were the June 26, 1963, when John F. Kennedy was in Berlin and give his historical speech, giving hope to the Berliners that all can end. And finally, the end of that period in November who knows? Yeah, okay, so I can write whatever. I know it's 89, and it is written here. So that was showing that even Germans Berliners are emotional and can really be happy. And with that, situation, those of you who have been conscious at that time already and have been part of that thing, you know, the golden age returned to Berlin, I think, to the country. And that's why we thought, why not exactly 60 years after the speech of JFK to start our MPS World Summit in an environment which connects, exchanges, and educates. Due to this history, Berlin has made a huge jump, even in Germany, within Germany, toward a multicultural, inclusive city. It is called Kleines New York, a small New York, and we do have a lot of signs of empathy. Now when the Ukrainian war starts, many of us had direct people at home and taking them. So we changed totally, and we have a wonderful city. And that is probably the more even important part of why Berlin would be the right selection for that second World Congress. And we have a small video here. If that works, you can see that you also can have fun in Berlin.
Tell me all your secrets. Fill me in on your wildest moments. Colored trees, your yellow leaves move me. Dancing moves, your painted red shakes the room. When I get there, I'll let my feet roll in your arms. I'll lose control. When I get there, I'll know the answer. Let me know when I get closer. Dancing land, you're my sailing ship. Guide me when you head for your trip. Right out of this wicked black and into bright. Straight toward the warm sun and into his hands. I'll let my roll. I'll break in your arms. I'll lose control. When I get there, I'll know the answer. Let me know when I get closer. Dancing land, you're my sailing ship. Guide me when you head for your trip. Right out of this wicked black and into bright. Straight toward the warm sun and into his hands. I'll let my roll. I'll break in your arms. I'll lose control. When I get there, I'll know the answer. Let me know when I get closer.
Okay, with that, you have a feeling that we have science there, we have a history there, and then we will be able to have fun, as you done already ask us to make sure. The next one I wanted to thank you, I call it the Berlin Preparation Group, which is working already like 5 months, I think, right? Somewhere about. We already fixed the venue. We found the best in Berlin venue right in the heart of Berlin, which is the Berlin Congress Center. It's the Alexanderplatz. What means heart of Berlin? This is a little bit about the venue. And here you see it is just on the Alexanderplatz with the television tower here, right there. And who knows Berlin knows that this is our city hall area right in walking distance. It is a nice place with a lot of different kinds of hotels being around to accommodate. And we think that this was a very first step to secure a right environment for that our our next summit. So with that, I would like to invite you to become in the next year, at least for 1 week, a Berliner. Me and my co-chairs, Marcel Leist, sorry for that. How can I go back? Marcel Leist, sorry. Somebody from the University of Constance is representing CAT Europe, and Peter is here already, Peter Loske from University of Tübingen, who is the European representing the European Organ on Chip Society, which joined the effort. And that is the idea which we hope will then later, when the summit is coming back to other countries than US, will also get it as a message to the other countries, try to combine the geographical local activities with that international World Congress and make it better and better. Also, the bar is high. And if we are able now, so we will meet you with the best ever German empathy and enthusiasm you can have. I mentioned the two dates where we were empathic and happy. And if we can do that, I don't know whether you guys can help me. If we can open that link, you will have 20 seconds of John F. Kennedy and the response of the Berliners to his speech. And that will be our attitude 26th of June next year, 60 years after our fathers and mothers had that attitude. But I'm not sure whether we can really jump in. Otherwise, you go, how many chance? Any chance? Okay, yeah.
Keep talking.
Okay. So I will, okay, I hope that all of you will be there. I hope that the sponsors which have been this year will repeat their engagement and that we find much more
All
people.
free men, wherever they may live, are citizens of Berlin. And therefore, as a free man, I take pride in the words, Ich bin ein Berliner.
This is our attitude next year.
Good. Okay. Uwe gave me another midlife crisis. I realized that on this day, my mother was highly pregnant. I was born the 2nd of September this year. Okay, so I'm enthusiastic about getting this to Berlin as the second place on a journey, which hopefully will continue. Can I have my slides? At the very beginning of this conference, I made a promise to you, and I used Queen. I said, we will rock you. And actually, I think, yeah, we have rocked you. So consider yourself rocked. I think you are now moved into a society, whether you want it or not. But obviously, you can opt out. Yeah, it was for me something quite natural to think about music, not only because of New Orleans. I'm a big fan into hi-fi, as Lena will be sometimes is annoyed about the new headphone, the new amplifier, and whatever I need. And I think our interest here is jointly in hi-fi, because hi-fi stands for the reproduction with high fidelity. This is exactly what we are about. Hi-fi is used mainly in the musical field, trying to produce the best music experience. But what we are trying to do is trying to do the best representation of biology which there is. I'm very much aware nothing compares to the original, as you can see here with Emmeline, if you can touch and have the real thing, but we are trying to come as close as possible to these experiences. We all have different types of musics we like. We have different areas we are using, but it is important that we come together and have a joint interest in hi-fi. Branford Marsalis, he was not born here, but he was raised in New Orleans and is one of the great jazz musicians of our time. He said once, there's two types of music, good one and bad one. And I think we can very similar say there's two types of science, good one and bad one. There's not applied science and fundamental science or medical science and others. It is all about quality. And I believe that in a very important part, mainly in the area of human cell and tissue culture, but also in extension, obviously animals and ecosystem, what we are doing is really game changing. It is about replacing the human cells, which were difficult to get in good quality and quantity, the tumor cell lines of the past, these HeLa cells and whatever, to replace them by something really better, stem cells, bioengineering, the microphysiological systems which bring us together. It is a slide I used earlier today when talking about quality of cell culture. We really have to move away from the irreproducibility of the cultures we had so far. The traditional cell culture is simply something which is not serving as soon as we have something better in our hands. And this is the tissue architecture, the organ functionality, the thing we are aiming for, the 3D, the co-culture and perfusion, which is really helping us to overcome many of the shortcomings cell culture had so far. Science is about the type of hi-fi. It is about separating signal and noise. It is really getting some things where we can see better because we have reduced separated signal and noise. And I believe it is in total the investment which is necessary now to produce less noise, to produce more important, more relevant articles, to increase the reproducibility, the relevance of our systems. And I think starting with hi-fi models of the human body and similar, this is a starting point for better science and helping also to get to more relevant things. It is about quality on the way. I'm making a shameless plug here again for good cell culture practices, which was a very important product for us parallel to the development of this conference series and the society. More than 100 people involved in over 8 years developed, just published in January, Good Cell Culture Practice 2.0, because I think this is what we all should be striving for when establishing high fidelity models. Yeah, it takes a village. It takes a ton of people. You see the happy co-hosts here. Susie is here. Don Ingber gave us a beautiful keynote lecture to get us started. I think we all are smiling today, very similar to the photograph here. But the work really was done by others. I praised Lena already several times as she had her finger in every pie possible, which led to this conference. But there's a team behind her. Camilla was from the beginning the partner in crime for getting these things done, and more recently joined by Anwin and Tracy. This is the CUT team, which was doing most of the work from our side inside. But yeah, there's tons of people who believed in it. The organizations, I can only say it once again, 52, 34 individuals on the scientific advisory committee, and one of the committees closest to my heart, the fundraising committee. That's why I put Jay, our committee chair, here up, because they made it happen. They believed in it and did hook others to come in. We had important pre-meetings. These pre-meetings gave visibility. We had more than 700 people attending these two meetings with excellent speakers. These are the chairs of the committee again. It is a larger group of people who help with these programs, and I don't have the time to name them all, but it was a fantastic way towards this conference. We had a local organizing committee, and here the two engaged members out of our group, Ben Capielle, David Bode. David has not only contributed to organizing the conference, but he gave it also the swing necessary, starring twice in his fantastic Benz. And I think the macro party was not a macro party, this was a giga party, wasn't it? So there was a committee behind getting this all done, who composed the program. And again, here, Lena, who chaired the committee, but also Magdalena and many others were involved in getting this committee to produce the program as it is. And all of our events here were grassroots, in the sense that everything was done bottom up. There was nobody saying, we have to go into this direction. It came out of proposals of the membership in the various committees. Everybody contributed. Everybody has ownership to the overall result. And I hope that we can keep this spirit of working together. And I think a lot of the enthusiasm and the heart blood which was spent, you feel in so many aspects, comes from this. Unavoidably, some things will not work perfect if so many cooks are in the kitchen. But I think we managed with very, very few flaws. And I'm very proud and I'm so thankful for at least 100 people who were involved in getting us where we are now at the end of this conference. Yeah, and those who came and lived it, you saw the statistics. We are shy of 700 being involved in this conference, but we will make our content also available after a certain period, also freely available, so that anybody can follow up what was said here. Already now we had some 200 people listening in to the live stream, among them 65 from FDA, yeah, which I found really remarkable. So you were heard. And these people will have prime access to our recordings, but we will, as I said, make the recordings available at some point freely, that anybody from whatever part in the world can actually access them. So thanks for coming here and living with us 4 exciting days here in New Orleans. Yeah, and then there were those who paid for it. I think they deserve an extra round of applause because they believed in this product and they made it happen. Yeah. So you see, I think the industry exhibition, which was nicely welcomed by all of you and populated, I think this was really what we could give in return. Prominent places all over our programs, and I hope that this was appreciated and we see you all supporting Berlin in 2023. This is my last slide. You see, Berlin is waiting for us. After I photoshopped this a little bit, the 2023 in Berlin, I promise you will be another exciting party. It will have a different touch, as it has to have. I'm a German, so I'm trying to bring all my empathy. Uwe, you can count on this. It will be the next step for our series of conferences. I'm excited that with the society we just created and which will be formally implemented in Berlin, we also have a sustainable strategy how this conference series will be let free, because we plan to spin this off. This will not be a cut event for all time. It is meant to be handed over after the first 3 years, which we want to help as a midwife to get this organized. So IMPS, the conference, the society, is going to be the organizer then for the future meetings. And I'm looking very much forward that we do this in the same spirit of exchanging, connecting, and educating each other. Thank you very much and see you in Berlin.
Thank you, Dana. Well, we're pushing up all the time.
Of course. Yeah. Well, you're my partner.
Yeah, I know. I will talk to